and a lot of issues that are actually environmental related, and especially that are triggered by changes in climate. And we have talked about this over and over again. And from yesterday's presentations, even to this morning, there's been a lot of emphasis and a lot of talk about the environmental shifts that are causing a lot of problems in many areas, in ecosystems, for bees, for plants, and also for this case, our viral friends who are maybe becoming even more dangerous, more epidemic, and that is what we shall be looking at today. And out of this, we are seeing a lot of issues to do with expanded vector zones. We know that most of the viruses that we have are carried by vectors. We are seeing mosquitoes extending to areas where they were not thriving before. We are seeing ticks and other vectors spread even wider, and then we shall have a look at a brief look at this. But this is because of the extended environmental shifts and rising temperatures and change in climate that has caused a lot of problems. And we have seen increased outbreaks, even in diseases that were not a problem before. We've seen more frequent outbreaks. We've seen even severe viral disease outbreaks as a result of the expanded vector zones and as a result of the environmental shifts that we've described. And that means that we cannot help but talk about One Health, where we are combining our human discussions. We are talking about animals, and we are also talking about the environment and the ecosystems, all of whose health is important and are connected deeply in enhancing the outbreaks that we are talking about here. And just to remind us, I know this has been talked about several times and by several presenters, talking about the components of One Health. Here we are talking about one side where we have the different sectors. We also have on the other side the different societies. And what One Health does is that it enhances communication, it enhances what we call coordination and collaboration. And this has been talked about over and over again during previous presentations. And the aim and the goal of this is to ensure that there is sustained animal health, there is sustained ecosystem health, and there is also sustained human health. So we are at a place or as a space where all these have to be looked at for us to be able to ensure that human health is also sustained. Now, what are the drivers of emerging viral outbreaks? So we have several viral outbreaks, as we shall see, but what is driving most of the viral outbreaks that we are seeing today? As we have said, number one is rising temperatures, and this is creating a lot of problems because we're having, like, for example, the dengue fever and the chikungunya virus epidemics that were mainly on the hotter and lower zones, now even going to highland areas and causing problems in places where they were not a challenge before. And this is basically because we are having a lot of increase in temperatures, and this causes the mosquito vector breeding to be dislocated to areas where we did not experience before. Now, we're having a lot of floods and droughts. So it is after a drought, then a flood. After drought, it is a flood. And this definitely causes a lot of problems in the water systems, which cause disease spread, even of diseases that were not a challenge a few years ago, like hepatitis B. Now it's hepatitis C, sorry, it's now becoming a challenge. It started in China. We thought it was a Chinese problem, but now currently we've had a lot of issues in South Sudan, in Uganda, and some spillovers even to Kenya. One of the presenters yesterday talked about deforestation from Central Africa and land use change, where you get a lot of conflict between animals, between wildlife and humans. And what this causes is zoonotic disease spillovers. And we have seen a lot of Ebola cases coming up lately, and also SARS-CoV-2, which was a big problem just the other day. Issues to do with urbanization and climate stress. So we are seeing a lot of our urban areas getting congested, creating a lot of crowding, which also causes a lot of stress in our environment. And when the environment is stressed, it means that it will have a ripple effect on our on disease or viral disease transmission. It is common knowledge that climate change actually amplifies disease, and it's not only viral diseases. We are talking about issues to do with bacterial diseases. We are talking of issues to do with sexually transmitted infections, because we have done quite a lot of studies and we have seen that even sexually transmitted disease follow climate change patterns. Where climate is becoming more severe, you find rainfall, drought, you find a lot of, or a spike in even sexually transmitted infections, cholera, and many other diseases that are not necessarily viral. But one of the aspects that is important is vector distribution. Climate change is affecting to a great extent the distribution of vectors, and especially mosquitoes, plus other vectors, where you find, like, as I said previously, dengue and chikungunya viruses previously were found in very hot areas, but now we are having cases that are being reported even in highland areas as a result of increase in temperatures and so on and so forth. So, spillover risks. So we are having higher spillover risks of infections, of viral infections. Like COVID, we saw COVID just the other day. Ebola has been a case where after every year or after every 2 years, there will be an outbreak or a spillover of Ebola virus or Ebola virus disease. And that is as a result of increased interaction between man and most of the carriers or most of the vectors of these viral diseases. And we're also having diseases that are more severe than they were. That is outbreak severity. Someone told me yesterday that, you see, previously this common flu was a flu, you'd get sick, get well, and move on. Nowadays, you might be hospitalized for 2 days or for 1 week, and almost everyone is getting a cough after every other week. And so that means that there is outbreak severity even of very common diseases, or very common viral diseases that we were used to. And it could be exaggerated by flooding, like the hepatitis E virus that we talked about, that during a drought period, there is low hygiene. During flooding, there is fecal contamination, and hepatitis E is easily transmitted. And we have seen this case in China, in many other countries in Europe, and now it is a case here at home in South Sudan, in some refugee camps in South Sudan, and also in Uganda. And some of the cases have also spilled to Ethiopia and also also in Kenya. One of the major important things that we have to note is that there is a very strong correlation between viral characteristics or viral sensitivity characteristics and climate change. The two of them go hand in hand. For example, you will find that there are higher mutation rates in most of the viruses, especially our RNA viruses, as a result of climate change. And what usually happens is that as the virus tries to adapt to increased heat or increased cold or increased wetness, then there is upregulation of some of the genes that cause mutations, that cause the problems that we are having today. Problems with HIV, or with, drug resistance or antimicrobial resistance. And so high mutation rates has been as a result of climate sensitivity, as our organisms or microorganisms try to adapt to changes in the environment and especially in climate, then they develop high mutation rates, and we shall have a look at that. Then the other thing that is a challenge is the broad host range. You find that some of the viruses will affect humans, they will affect monkeys, they will affect a particular amphibian, and then they will still affect humans. And this is actually a great driver of the emergence and reemergence of many viral infections or viral outbreaks. And what it does is that this particular case makes viral infections to be easily spilled over. Like, for example, there is one that affects a buffalo in the forest, and then there is cross interaction between humans and buffaloes in the forest, and so the zoonotic spillover risk is increased as a result of host range, or the broader host range of these organisms. The other thing is environmental stability. You find that there are organisms that are stable at certain environmental conditions. And so with a change in climate, like for example, if you look at the hepatitis E virus, it is very stable in wet conditions. And you find that there is more rainfall. Whenever there is more rainfall or even flooding or soils are even wetter, the hepatitis E virus will be more stable. And definitely vector-borne dependence, as we have talked about the shifting vector patterns, which have also been talked about in the course of presentations here. But we also have the latency and reactivation. There are some viruses that can go latent in an organism or in an environment, but then they can be reactivated in times of stress, like for example the herpes virus strains. They can go latent for a long time, but then when there is stress, like for example there is increased heat, there is immunosuppression, there is food insecurity, malnutrition, these particular viruses will be reactivated and they will become an outbreak. And so all these are climate relatedness of some viral strains. And what we are seeing currently is that we are witnessing a lot of viral disease outbreaks as a result of combination of many of these factors. And just to remind us, I know we know this, but there are some global pandemics that have come up recently that are very related to climate change. COVID is one of them, COVID-19, just the other day that made us just shut everything and caused a lot of havoc in the healthcare system, in economies. Mpox, which has not been a problem for many years, is now a challenge and is actually going beyond where it was endemic and affecting almost every other country, especially in East and Central Africa. We had an outbreak of Marburg virus just the other day in Tanzania and in Equatorial Guinea in 2023 as a result of climate change and climate dynamics. Ebola, hepatitis E that we have talked about that has not been a challenge for long. Dengue fever and chikungunya also becoming a problem, as well as other avian H1N1 and H5N1, among many others that are now a bigger problem. And there were reports about some few months ago of H1N1 showing possibilities of being a better infector in humans. So developing receptors that make the virus to infect humans even better. And all these are directly linked to climate change, and some of them, like the influenza viruses, could have an indirect link, but then you cannot ignore the climate aspect of it. For East Africa, we have talked about Marburg virus in Tanzania, Mpox, Ebola virus, Rift Valley fever that we had cases reported in Marsabit just last month, and which had gone down, but then we're having sporadic outbreaks in different areas, and hepatitis E that we have talked about widely in South Sudan and parts of Uganda. We have also seen quite clearly that climate change also impacts on antimicrobial resistance, or AMR. And we have seen a lot of increase in AMR in E. coli, in S. aureus, and many other pathogens as a result of increasing or rising temperatures. As this particular pathogen tries to adjust to the increases in temperatures, there are also other negative effects, and one of them is AMR, which is the talk of the day today. Environmental pathways getting clearer and better for the pathogen. Cross-sector challenges of AMR, where we're having AMR from animals or from vet into humans, and from crops also into humans, where the ecosystems are speaking to one another in terms of enhancing antimicrobial resistance. And this means that there is an urgent need for One Health approach in AMR prevention. But then that is not enough. We have had neglected diseases that were not a challenge for many years, but now they are springing up as very important infections. Chikungunya virus and dengue virus as one of them. They are really, even in Europe, in Asia, in the US, where they were not a challenge, now you are having traces of those diseases as a result of climate change. Zika virus, Lassa virus, Rift Valley fever that we have talked about, with a current outbreak, possible outbreak in northeastern Kenya, and hepatitis E that we talked about. So these are diseases that did not have a problem, that we did not have a problem with, and they were actually neglected. But now they are springing up, and we really have to have a look at them in the face of climate change. So really, climate change is changing a lot of issues, especially in the health sector. There was that which was not a concern, but now it is a concern, and especially in sub-Saharan Africa and East Africa, where the healthcare system is not well streamlined and is not well structured. These diseases are becoming more and more of a problem as we proceed. Now, just to remind us that, in summary, what happens is that, and we have had this over and over again, that the vectors will be used to amplify the epidemics, and then you pass these epidemics to humans. But now having our friendly animals interacting with even the wild animals makes it even worse. But then we are not left without a solution. There are things that we can do that can help us in mitigating the spillover of viral infections, especially from the environment. And that we have tools that we can use currently, and most of them are tools that are under study or are being developed. GIS tools, climate monitoring tools, genomic monitoring tools that African BP is very good at, early warning systems, as well as data collaboration. So getting data, world data, and data sharing. And I finish. We call ourselves to action in mitigation and preparedness for climate change, because I can assure you it will only get worse. We don't expect it to get better. In the US, they are struggling with floods. Elsewhere, they are struggling with drought. So one of the things we need to think about is integrated climate adaptation mechanisms, where climate adaptation is being linked to health policies. We are doing that as a country, and there's some work we are doing with AMREF International University in integrated climate mitigation or adaptation into health policies. But all of us have to be part of it, especially as scientists. Number two is to strengthen One Health surveillance, where we look at all the ecosystem. We look at the animal, we look at environment, and we look at human health as one and the same thing. We also need to invest very well in genomic epidemiology, and the good thing is that African BP has really been on the forefront, trying to bring this to the forefront. We need to expand our access to vaccination. We saw what vaccination did to COVID-19, what vaccination is doing to many other viral diseases. Most of, especially the viral diseases, are neglected diseases. Even if they have vaccines, their vaccines are not readily available to those who need them. So we really need to expand our access to vaccines as we promote multidisciplinary collaboration. We need to talk to the bee people, to the amphibians, the genomics people, microbiologists. Everyone needs to speak to one another. And finally, we need to fund our research. We need to increase research funding in low and middle income countries. Does not mean pouring a lot of money into it. It's having forums like this where we bring in more collaborators, more people talking about the same thing, making it cheaper and making it easier. And we did this sometimes early this year where we did a workshop that was supposed to be very expensive, but we brought bioinformatics people, we brought Technical University of Kenya, we brought other Kenyatta University to be able to carry out a workshop using the AfricaBP forum that was able to train over 32 people in antimicrobial resistance and also in phage. And with that, I stop and I welcome questions. Thank you for listening. And thank you for the opportunity, sir.
More appreciation, Dr. Kingo, and thanks for the wonderful talk. Now, I will invite the speakers to the front for questions. Professor Rebecca, Dr. Kingo, and Karani. So we have the online. We can start in case we have online questions. We are going to have only a max of 2 questions per speaker. No online? Oh, yes, Jean.
Thank you very much. My question is for you, Karani. Thank you for your presentation. You presented models of extinction, and I would like to know what are the parameters that are built into these models. You talked about extinction of frogs, and it's alarming to see that in all your models there is extinction quite soon. What are the parameters, and what is the effect of climate change, of change of habitat, encroaching by humans, etc.?
So thank you so much. So first of all, I used Vortex to model extinction probabilities, and one of the parameters that I used is inbreeding. Inbreeding, you understand what I mean by that, and then used the population densities that I had showed earlier, of about 1,200 individuals per hectare. That is within the breeding site of that, and then also there was lack of genetic data, so I used some dummy value for that. And then modeled for a century, that is 100 years. What else? Yeah, mostly those, if that is adequate to provide that. But then when I was beginning, my major intention was to incorporate genetic data, that is from the diversity values, using data from NCBI to see whether the species that have some level of genetic diversity known have similar patterns of extinction compared to those that have nothing at all, that we do not know anything about. Yeah, and then about climate change, I am this person who people call pessimist about it, but I believe that some of the things that we talk about are probably not necessarily climate change, but rather our livelihoods, our means of production, because for the species that I modeled, like the Kenya jewel and the silver bladder reed frog, you realize that more of habitat loss is more of habitat loss rather than those stochastic events resulting from what people call climate change. Because these frogs depend on wetlands, and our wetlands are continually being encroached into. The pesticides we use, destruction of forests. For example, the Kenya jewel is fully forest dependent. It cannot be found outside the forest. Probably unless you have some level of canopy, that species you will not find it there. So the things that we do, rather than climate change itself, I would say are the result of some of the extinctions that we are seeing, especially in this part of Africa. Yeah.
Great, thank you, Karani, for the answers. And in case of anything, can touch base with Jean later on. One more question, specific to Karani.
Yeah. Thank you. That was a great presentation. I'm a champion of, life and life science, plants, animals, and all that. But you see, with population increase, you realize that the riparian land, the forest, and everything are being bottlenecked by growth of population. By 2050, the population of the world will be increasing. Is it possible for us to create artificial swamps, like the way we have national parks and all that, whereby these endemic species, or rather these important species who are getting extinct, can be put and be protected, rather than trying to tell people not to build houses in riparian lands, telling people not to cut down forests? We just create artificial areas whereby we can create artificial swamps and all that. Is it possible? That's what I'm asking. Yes.
So thank you so much for that good question. I believe in passion for nature and love for humanity. And one of the things that I keep telling people is that by 2100, most of the lands that we use for agriculture and for other uses and for urban development are most likely to be returned back to nature, which is a very odd thing, because that requires policy, that requires consensus, and that requires people to appreciate that some of the things that we do, like agriculture, also do not need to be done in the scale that we do them. A simple plot of land can feed an entire community. So we do not need the rest of the land that we keep cutting down forests to plant crops for use. Last week I was doing a baseline survey for restoration of a wetland stream, and a stream in Masai Mara. We have a restoration site there at Mar Trust, and we are in the near future we are going to restore an entire stream that is originating from Mar Trust. That is being able to put damming points, being able to restore the vegetation that was there before that stream was destroyed, placing strategic access points for wildlife and for livestock, because you realize that is land of the Ma community. So we have a lot of livestock that is uncontrolled. That is the access to water points is key, but then needs to be managed in order to restore those wetlands. I believe that it's possible to do most of those restoration works, and research has shown continually that restoration increases biodiversity. And from the presentation that I showed an example from New Zealand, you've seen that starting from soil bulk density, soil moisture, all the way to microbiomes and plant diversity were all had changed as a result of wetland restoration. So thank you so much for that question.
Great, thank you, Karani, for the answers. Then we move to Professor Rebecca's. Question to either to Kingo. Question to Kingo. One question from here, Kingo, and one from online, yeah?
To professors, doctors, and all protocols observed. Good morning.
Good morning.
It's quite a heated one, huh? 3 powerful presentations this morning, but none touches down Dr. Kingo, because it's a main challenge that's facing globally. So my key question is, because I can see how you're presenting your work, it's really something that's spanning in you also and everybody. So I'm asking, which is the quick fix that you've come up? Because we know these are the things that we're gonna be having. I mean, this is our home. We can move nowhere, and it's only one way we can try and see if we can make it better for each and everyone. So what's your take? What do you think is the quickest way we can start already, so that we can be able to curb all these issues? Thank you.
All right, thank you, sir. So, quick fix. Unfortunately, there are no quick fixes. We have to think sustainably. So, our discussions and our thoughts are supposed to be sustainability. But one of the things I would really advocate for is African grown solutions to African problems. We have heard a lot, like for example, if you look at HIV, which has been a menace, we're depending a lot on drugs from out, a lot of research from out. And the time when we started doing our own research and creating our own solutions to our problems, we have to a greater extent been able to manage HIV with all the challenges that are there. So talking of viral infections, we need to think of local solutions, but then when you think of local solutions, you have to think of sustainability of the same. A quick fix for today may not be a quick fix for tomorrow. So we need to be sure that that which we bring up today, or that which is the solution today, will benefit even generations to come. The other thing is definitely talking One Health, which is really important. Previously, we have thought as that human disease is human disease, and so human disease researchers speak on their own, veterinary people speak on their own, microbiologists, environmentalists speak on their own. But when we start such a discussion of human health is linked to every other health, the health of the environment, the health of animals, and so on and so forth. Really, that's a beginning of getting it fixed up. Number 3 is collaboration. We need the way, here we are several people from different, we're sitting with beekeepers and amphibian breeders and so on and so forth. So really getting to talk one language is important. And then finally is capacity building, especially for our young people. And I really appreciate AfricaBP for being very specific about the young people. And that's why the room is full of more young people than the older generation. We need to invest in the young people, that which we did not get when we were growing up, that you get a chance to get a chance to research. And finally is to link whatever we do to policy and to public health. Public health people have been speaking alone again, policy people speaking alone, but we are at a space where everyone is now interested in what is happening. We are talking to parliamentarians, we are talking to policy people, we are talking to public health people, and there are several initiatives that I'm involved in, where we're talking to nurses and clinicians about AMR, about use of bacteriophages for against AMR, about viral infections, which was not the case. So really we have to have all this multidisciplinary thought for us to be able to move forward. Quick fix, maybe a vaccine. Economic status is also a challenge. But then if we combine all the aspects together, we'll be able to proceed and proceed sustainably. This call is for sustainability. Thank you.
Wonderful, wonderful. Thank you, Dr. Kingo. Do you have questions for Professor Rebecca Karanja? One question online and one in here.
Good morning, everyone. My question is to Professor Rebecca. You said that you found like 20 mentions for the eDNA pollinator studies. What were the search parameters for the databases so that we can look also into it? And also for the eDNA, what are the materials that you use to get the DNA to maybe investigate on pollinators that visit the plants when you're not in the field doing the work, maybe during the nocturnal or the rare species during the night?
Thank you very much for your question. On the first question, The parameters I used were methods, eDNA, pollinators, pollen, nectar, propolis, beeswax, hives. Then for the materials that we would use to detect if pollinators had visited plants or flowers in the absence, when it's not the observation window, you can use the hairs. The bees are quite hairy. You can use any shed materials like scales. You can use fragments, skin fragments, wing fragments, and even the pollen that you may find on the flowers. It can have some materials from the pollinator. Thank you.
Online, yeah? Okay. Someone has their hand up, eh? Maybe you can unmute them to ask the question. In person, do we have an extra question? No?
Okay, thank you very much for giving me the chance to ask the question. My question is to Professor Rebecca. Again, it is a really wonderful presentation. My question is like, how do you discern or identify which DNA is from which species, or are you like bulking up all the DNA and identified by different species? Because like it's not a little bit clear for me because the pollinators, they are not necessarily bees, and there might be like other insects and also other mammals like bat or others. How do you discriminate that? If you have developed a protocol, I really love to learn. Thank you.
Thank you. That's why I said we are at pioneer stages. This has not been done, even barcoding. But if we could use references that have been done elsewhere, that would make our work easier. Because as I said, when plants are visited at night, at times you are not able to tell. But if we collaborate and use databases from other places, we'll be able to detect. Plus, I know that fragments of either the skin, the hairs, they are unique to species. Human hair is different from hair from other animals, so we can be able to detect the bees. Then once, if we could have the barcoding of what is already in the databases, it would make it much easier. Thank you.
Thank you very much.
Thank you, Karani. Thank you, Professor Rebecca, and thank you, Dr. Kingo, for the wonderful presentation. Let us appreciate the speakers.
All right, so we're ready for the next session, for which Professor Anne will take us through.
So thank you very much. I feel like that first session for today deserves another round of applause. Can we appreciate them? Thank you. So now we're moving on into a very, very exciting part of this session. I mean, all the others have been very exciting and very informative, and I think you'll agree with me. But the reason why this session is very, very important because it's for the first time that we'll showcase the genomes that African BP has sequenced. When we published in 2022, we said we're going to sequence 100,000. Yeah, very ambitious project. It's taken us 3 years to, get the pipelines going, to get the workflows decided on, to get the samples in some cases, to get the funding. Funding took some time. And now we can actually showcase the beginnings of the 100,000 genomes. So to set us off, we will have Dr. Ansia Van Coller from the South African Medical Research Council. She will be speaking about the genome assembly of the indigenous
African plant species, the Hylichrysum from Western Africa. And Dr. Ansia, for her and for us, this particular genome is the first time it's been sequenced. It's the first time the Oxford Nanopore is actually being used to sequence a plant genome, at least on African soil. And as you can see, Dr. Ansia is from the South African Medical Research Council. They also were very happy to be given this delightful work of venturing into plant genomes. And we are very excited to hear from you, Dr. Ansia, and you have the mic. And for the next 15 minutes, we will delve into your plant genome. Welcome. Thank you.
Thank you. Thank you so much for that introduction as well, Anne. Yes, so I'm very happy to be able to share with you guys some of the work we did at the South African Medical Research Council in collaboration with AfricaBP to do a de novo genome assembly for this African plant species. So this is Helichrysum odoratissimum. It grows very abundantly in southern Africa, and in some of our local languages we call it kooigoed or impepo. It's also known as African incense because it has quite a strong perfume smell. It's also used as an insect repellent and a perfume for that reason. It is of great cultural significance in South Africa. It is often burnt to kind of ward off evil or to bring good fortune. It also has lots of medicinal uses, so it has been shown to be successful in the treatment for heart disease or relief of chest pains, cough, fever, microbial infections, and it also has like calming effects when ingested as well. It is one of the most frequently harvested and traded plants in South Africa, and it has great potential for commercialization. So there's been no assembled genomes published for any species of the Helichrysum family, but there have been studies looking into their genome size and their chromosome number and the ploidy, and there's been lots of variation identified in these elements, and it has shown that polyploidy, for instance, is a very important evolutionary driver for the diversification of the genus, leading to broad ecological adaptability. So within the same species, you might see a diploid individual in a certain habitat and then a hexaploid individual in a habitat a few kilometers to the side. So given that no genome has been published for any of the Helichrysum species, we then sought to sequence and assemble the genome of Helichrysum odoratissimum using Oxford Nanopore Technology long read and high-C sequencing. So just for a little background on de novo genome assembly. So if no reference genome exists for an individual, it needs to be constructed de novo. And so this process relies on alignment tools that aligns the reads to each other in order to identify overlaps and identify contiguous sequences, which can then be linked together in a process known as scaffolding to generate a consensus sequence. So this is a very computationally intensive process and can take quite a while, but long reads such as PacBio or ONT are especially useful for de novo assemblies because it can be easier to resolve, complex or repetitive regions of a genome. So we sampled a plant in the Western Cape, not too far from us, on a beautiful wine estate. And then some of my colleagues were doing all the lab work, and we were able to extract really high quality DNA and RNA from a fresh plant. And then we did ONT sequencing on our ONT promethion P2 solo, and we did the high-C and RNA sequencing on the MGI-DNB-seq G400 platform, and these are located within our facility in Cape Town. This is the general workflow that I followed for constructing the genome. I won't go into too much detail just for the sake of time, but in general, I took the ONT data, did high quality base calling, basic QC and trimming off low quality reads. I did some ploidy estimates and genome size estimates using this data as well. I then integrated the Hi-C data to do a de novo genome assembly using Hi-Fiism. And thereafter, I aligned the Hi-C reads to the draft reference and pulled out all the relevant Hi-C contacts, which are used for scaffolding. And then we did manual curation of the genome up to chromosome level. And then we integrated the RNA sequencing to annotate the genome, to describe what is inside the genome. So I used smudge plot and genome scope to estimate the ploidy of this plant, and it said that this plant was hexaploid, and you can see there are 6 peaks of the K-mers here. So that's telling us this is a really complex, very heterozygous plant, and it had a very large genome size estimate. Just for a little more detail on the Hi-C protocol, as I'm sure many of you might not be familiar with this approach. So Hi-C is basically a high throughput technique to capture the 3D conformation of chromatin. So in a cell, the DNA is kind of coiled up on itself. So bits of DNA that are not necessarily linear, they do interact with each other. And through the process of cross-linking, we can attach these pieces of DNA next to each other in a process known as proximity ligation. And then when you extract the DNA, you end up with fragments that contain information from different regions of the genome that are within close spatial proximity. And you can sequence this DNA library and generate a Hi-C contact map like this, where you'll see where a specific region of the genome interacts with another part, and that can help you to arrange the genome in the order of chromosomes, because DNA that's closer to each other will interact with each other more. So it helps to generate chromosome level assemblies. So here's some of the findings from our assembly. I've shown here the draft assembly and then after scaffolding with the Hi-C. So there's quite a large number of contigs, as in separate contiguous sequences that make up the full genome, but it is quite a large genome, so the fragmentation is not too unexpected. It has a large N50, which means most of the fragments are really large, or 50% of the genome is made up of really large fragments. And you can see that in the L90 number as well. So this essentially means that majority of the genome, 90% of the genome, is made up of 24 contiguous sequences, where the rest are just kind of small, unplaced bits. And we had a really good BUSCO score. So this is the percentage of conserved genes that we identified in the assembly. We then moved on to manual curation using the Hi-C data, and this was done with the help of Sadiq, and he will be presenting after me as well, so you'll hear straight from him as well. And then we were able to anchor those contiguous sequences into 7 very large chromosomes, and then several more unplaced smaller scaffolds. So this is a manual process, and it was quite lengthy, especially due to the size of this genome, the ploidy, and all of the repetitive elements present in this genome. But here you can see the Hi-C contact map for the genome. So I then moved on to annotation, and we identified 342,000 gene transcripts using the RNA data. And then we also saw lots of repetitive elements. So for instance, you can see here about 54% of the genome contains transposable elements. And I've tried to illustrate it on this circus plot here as well. We can see the repeat density spans across all of the chromosomes. So this could be associated with the survivability of this plant and its adaptation to its natural habitat in the mountains. So yeah, just to conclude, we were able to construct a very high quality chromosome level assembly for Helichrysum odoratissimum, and this completeness and the quality was shown through all these quality matrices we used, and you can see using the high C contract matrices that it clusters nicely into those 7 chromosomes. And yeah, this genome will contribute to conservation and sustainability. It will help with bioactive compound discovery and like bioprospecting, which will assist with the commercial development of this plant. And that's all from me. Thank you so much for your attention. I'd like to also just thank the team at the SAMRC and at AfricaBP. It has been a big project, but yeah, I've learned so much working on this. And thanks everyone for your attention.
Thank you very much indeed. It has been exciting to begin this sequence, and I know you're at the tail end of the final analysis and publication, and really look forward to reading that paper. And now that everything is standardized, I'm sure we will just quickly start churning out those genomes. But please hang on online so that we can have the Q&A after. And next, we're going to hear from Sadiq. Sadiq is going to talk about the genome assembly of the African star. Sadiq is an active member of the African BP in the subcommittees that deals with sequencing and annotation. He's a bioinformatician, currently a postdoc researcher based at Ramsted Research Group in the UK. He focuses on tree and crop genome, but we've been using him a lot for all the sequencing and annotation that's happening in the African BioGenome Project. And we're very happy to listen from you. And so please go ahead and make your presentation. Thank you.
Thank you, Professor Han, for introducing me. Just to make sure, can you see my slide? I'm not sure that I did the right slide.
Yes, we can hear you and we can see your slides.
Okay, good. Good. I'm really happy to be here for in this great workshop, and I'm also really excited to present this unique work particularly. So my talk is going to be about chromosome scale assembly of the white star apple genome. A little bit intro about white star apple. White star apple is a forestry, an indigenous forestry, widely distributed in East, Central, and West Africa. And it's primarily cultivated for its nutritious and sweet fruit, which is almost the size of apple size. And then it's highly valued for its local market value and widely utilized because of its nutritional value. And then it has well known for local medicine due to antioxidant and anti-inflammatory role. And besides being utilized directly, it are also is also used to as a processed food product, like in a gel, jam, and marmalade. And so somehow it can be processed into some sort of product, and showing and how it can be processed in the versatility in its utilization. And beside the food role, it has also industrial role. It can be used as wood protectant, resins, and the seeds of the fruit can be used to make vegetable oils. So basically every part of the plant has some sort of use. It's really a multi-purpose crop, particularly in the West African. So, however, it's because of its diverse utilization, it's being considered as need-threatened species. So now effort is more gearing toward this conservation effort. But again, this is almost horticulturally undeveloped food crop. And part of that is just limited genetic and genomic data set. So for that reason, we have generated high quality reference genome for this staple crop to support the conservation effort, also the breeding pipeline. So we used PacBio Hi-Fi and OmniSeed, which I don't want to go into the detail as the previous presenter already presented detail. So the overall idea is the PacBio Hi-Fi and OmniSeed that we just combined, and we use them to reconstruct the haploid assembly of white terapole. So in detail, before going into the detail, what we did is we wanted to look into the haploid genome size of the terapole genome, and then also the heterozygous level. So we find out that it's diploid, and also the haploid genome size is around 823 megabase, and it's really homozygous. So that's helped us to kind of design the reconstruction or the assembly strategy. So based on that, we went for reconstructing the haploid genome size using this workflow you can see on the right side, and until we get in the final curated and chromosome scale assembly. So looking at the assembly statistics, so we managed to assemble almost more than 99% of the estimated genome size, and the largest scaffold, the largest chromosome is around 100 megabase, and then half of the chromosome is almost higher than 57 megabase, and then 90% of the chromosome is within 47 megabase size. So totally almost around 96% of the assembled chromosome, the assembled sequence is assigned to chromosome. And when we looked into the assembly completeness, basically looking into those evolutionary conserved single copy genes, we can see that almost 96% of those single copy orthologous genes is already conserved. A few of them are fragmented or missing, which is attributed to the fragmented scaffolds. And then once we clarified and sorted out the contiguity and the completeness of the assembly, we looked into the repeated gene content using the workflow you can see on the left side. So we found out that almost 59% of the terapole genome is highly repetitive. Almost 14% of that repeat content is attributed to retrotransposon element, and the remaining percent is attributed to a DNA transposon element, including T element, non-T element, and a line element. But the rest part is seems to show some sort of gene coding element, gene coding part. So looking at the distribution of repeat content, we can clearly see that there are some variation of repeat content across chromosome. Basically, on chromosome 8, we can see that the DNA repeat content of chromosome 8 is a bit higher. So we think that that might be something to do with adaptation of this species, which we will further look into. Once we identified and we have the repeat distribution of this species, we looked into the protein gene content. Basically, we only used homology-based approach using protein codings of closely related species, and also the inferring genes directly from the genome. So we predicted around 75,000 genes. Almost 3/4 of the genes have some sort of functional attribute. So we think this resource would serve as a baseline for conservation and breeding effort. And then this is just kind of to show the gene and repeat content. So in summary, this is basically the first genome which was sequenced on ground in Africa from PacBio Hi-Fi and OmniSeed for plant genome. And it's also effectively demonstrated collaboration among various institutions in Africa towards biodiversity genomics. And we think that this is a source of a source that can be utilized for future breeding, basically looking into the evolution and adaptive traits that can be exploited further for breeding purpose. So there are quite a lot of people who are involved in this work. I would like to thank Michael Landy, who is the lead contributor to this work, Laura Yedamon, Kisili Acclaim, and every other who actively participated in this work, and also various funders who contributed financially and through infrastructure. We already have the manuscript. We are expecting to come out in a couple of months, and we really look forward to have your feedback. And that's what I have. I'm really happy to take any questions. Thank you.
Thank you. A round of applause. Thank you very much, Sadiq. Again, you've said another first of this type of sequencing happening on the continent, first of the assembly. And we must thank the lead scientist in this, that was Michael Landy. He had to travel and he wasn't able to make this presentation, but you can see because it was a collaborative effort, we were able to very, very quickly ask Sadiq to present for us. So we're really, really grateful. So hang on to your questions, please note them down. That is our second AfricaBP genome. Our third AfricaBP genome, which is in a consortium of genes, a group of individuals, comes from South Africa. The presentation will be made by Sinebongo, but the lead of this group is Professor Olivia Mafoli Ntanga. She's an deputy executive dean in the College of Agriculture and Environmental Sciences at the University of South Africa in South Africa, and she's a full professor of animal genetics and has also served as the head of Department of Agriculture and Animal Health. She leads a big group of scientists that include Dr. Lucky Nsengani and also Dr. Sinebongo, who's the one who's going to do the presentation. And I would like to invite Sinebongo now to go ahead, and we thank your group for actually initiating this particular group of sequences, and we're excited to hear about this. Please go ahead. Thank you.
All right. Thank you so much, Prof. N, and thank you so much for the organizers of the workshop. My name is Sinebongo. I don't know if you can see my screen.
Yes, your screen is visible. You're also audible. Maybe you can just make it in presentation mode.
I think I've done that now.
That's great.
All right, so I'll be standing in for Lucky Nsengane. He couldn't be able to be here, so I'm happy to give the presentation. So I will be talking about the sequencing livestock genomes for food security and biodiversity conservation. This is just a case study of locally sequenced indigenous African pig, cattle, sheep, and goat breeds that we have done this side. This is in collaboration with the Africa BioGenome Project, and we are working within the pilot project, which is hosted at the University of South Africa. So Africa is quite rich in diversity, that is in species like cattle, your sheep, and the goats. And so the African livestock, it accounts for 1/3 of the global livestock populations. Cattle are mostly dominant in terms of numbers of breeds. This is followed by sheep, chickens, and goats. The rich biodiversity, it's in indigenous livestock breeds. They exhibit unique and rare adaptive characteristics. So as part of the African Agenda 2063, it's central to poverty alleviation and food and nutritional security. Beef and milk production, it's deeply embedded in continent's livestock production system. But although the beef and milk are mostly from the cattle, there is a potential growth in livestock production as well, which includes the small stock and your chickens as well. Then the livestock sector, it contributes significantly to agricultural GDP in the African continent. And this is related to different farming strategies, strategies, yes, production practices, and utilization of indigenous breeds. This is due to their hardiness and adaptation to harsh environmental conditions and climate change, with which previous speakers have alluded to. So the pilot project that we work in, it's called the Nelson Mandela Genomes Initiative for Conservation of Nature. And its main aim is to understand the genetic basis of Africa's culturally and economically significant animals and plants. It's also centered around building genomics capabilities for food security, biodiversity conservation, and biotechnology. So as the part of the pilot project, we have done so far 30 genomes. 15 of these have PacBio hi-fi reads only, and then 15 of these have hi-fi and OmniC reads. Those that have both hi-fi and OmniC, we've assembled them to the end, and we are just finishing off some of the processes. So the method that we use, we work with fresh samples. It can either be blood or fresh tissue samples, and we put these in dry ice, and then we just ship them off to Inwaba. And previously, we've processed these through the PacBio Sequel 2, and then we went into partnership with Inwaba, and we purchased the Revio machine. So the recent genomes that we have done, we process these through the Revio. Sorry. And then the methods that we use for assembling, we mostly utilize the Vertebrate Genome Project workflows, which make use of the genome scoping just to characterize the genome, then the hi-fiism and the scaffolding method. And then we finish it off with the manual curation process. So the genomes that we started off with, it was the Nguni sheep, the Nguni goat, and the Goldbrook pig. So you will note that the Nguni species or breed in both cattle, sheep, or goats is quite hardy, and it's very popular in smallholder farmers. This is due to its adaptation to different agroecological regions, and it's also very hardy, and it's resourceful to such farmers. And then it's the Nguni goat. It's indigenous to the Southern African region. It's small frame, multicolored, and it's got glossy short hair. And it also has exceptional tolerance to harsh conditions. Then the Goldbrook pig, it's adapted to harsh conditions and diseases that are endemic to the region, and it is regarded as one of South African indigenous pig breeds. So to scope out the characteristics of the breeds, and then to also sort of compare them to what is published already, we looked at the genome scope, and we saw that the length, levels of heterozygosity, and then also the error rates and duplications, they were quite comparable to what is published. So the length, it was around 2.9 for Nguni sheep and 2.8 for Nguni goat. And then for the pig, it was 2.6. And then in terms of looking at the completeness of the genomes, generally found that we didn't get anything that was less than 95%, which was quite comparable, and also proving that we have generated good quality genomes for our indigenous species. And then looking at the matrices, again, we compared our genomes to what is published already. Then if we're looking at the Nguni goats, looking at the scaffolds, and then the number of scaffolds, looking at the N50 and your L50 and the contigs, we saw that we have, based on these matrices, we have generated good quality genome for the Nguni sheep. This was also the same for the Nguni goat, comparing it to 5 other genomes, and looking at the matrices like the number of scaffolds, the N50 matrix, and the L50. So from these, we also concluded the same. I think the same thing for the Goldbrook, the genome length, the N50, then the total ungapped length. So we're happy with the genomes that we have produced for these breeds. Then we went on to do cattle as well. For the Nguni, we looked at the different ecotypes that are found in Southern Africa. We looked at the Bedi, the Venda, and the Shangaan. So between these three, there is, if we're looking at the Bedi, there's a distinctive coat color pattern, which is often featuring a mix of red, black, and white markings. And they are known for their good mothering abilities and the ability to thrive on poor pasture due to their efficient grazing behavior. And then for the Venda, this one is also known for moderate milk production and good mothering ability. It has distinct coat color pattern, which is high proportion with a high proportion of white. It's got good heat tolerance, strong disease resistance, and excellent adaptation. Then if we look at the Shangaan ecotype, it's got a medium body size, very varied coat colors with multiple patterns, and a docile temperament. If we take these three, we noted that there is quite a difference between them. And this is documented in a study where they looked at the winning weight. We noted that the Bedi ecotype had quite a high winning weight in comparison to the Shangaan and the Venda. And then looking at breeds, cattle breeds that are mostly favored within the commercial space in South Africa, we took the Bonsmara, the Thuli, and the Drakensberger. The Bonsmara, it's a composite breed developed from commercial beef production for commercial beef production. So it's composite, it's the Hereford and the Shorthorn and the Afrikaner breed. And then the Bonsmara are well suited to diverse environmental conditions, and they're tolerant to heat and tick-borne diseases. Then the Thuli, it's a dual purpose breed originating from Zimbabwe. It's adapted to harsh environmental conditions, and it has a good maternal qualities. Whereas the Drakensberger, it's native to South Africa, adapted to harsh conditions, and it is perceived to be a dual purpose breed, and it's tolerant to diseases as well. So the Bonsmara and the Drakensberger are also known for good meat quality. Hence, they are very popular in the commercial market. So again, we looked at the characteristics of the genomes, and we saw that they are quite comparable to what is already published. And then we sort of tried a different route with the Drakensberger. This was to help with resolving the haplotypes in the genome. So we did a trio. This was the first of our trios for this project. So we took the parents and the offspring. So the parents, we did short reads for them, and the calf, we did long reads for them. And then we also did the same for the ecotypes, the Nguni ecotypes, and we looked at the characteristics of the genome, and we were happy with how they were comparing to the genomes that are published out there. And then relatively the completeness of the genomes for all the cattle that we have done, we noted that the completeness was nothing less than 95%. And the missingness, it was sort of also averaging and comparable, nothing alarming, basically. And then looking at the statistics for the cattle breeds, we compared it to the Yunling, and the genome sizes, they were relatively comparable. And then looking at the scaffolds, we were happy with the outcomes. They sort of showed the quality, the good quality of the genome, the N50 and the L50 as well. And then to help polish the genomes, we also did manual curation. This was just to correct misalignments or misjoins. And then we noted that after manual curation, the genomes were better, the quality was a bit better. And that was the last step before annotation. So annotation, we've done it for the cattle, and then we did it for the sheep. and then we're still working on the other breeds as well. So we looked at the number of genes, the length, exons and introns. That was for the cattle breeds. We're happy with the number of genes. And then in the next slides, you'll see we were looking at the shared orthologs with other breeds. This includes breeds from South Africa and then the published genomes as well. So for this one, we only looked at the breeds that are South African. So that's your Thuli, your Drakensberger, your Nguni ecotype, and the Bonsmara. So that there was a lot of sharing of orthologs between the breeds. And out of these, the Thuli cattle had more orthologs in comparison to the other breeds. So we did the same with the sheep. And we compared it to the published Rambolet genome. Then we included the Tibetan and we had access to the Zulu sheep genome as well. And we did the comparison there as well. And we saw that between the Zulu and the Bedi, there was more orthologs for the Zulu in comparison to the Bedi. So in other genomes that we are working on, it includes the indigenous chicken, big blind snake. In terms of plants, we've done the bush tea, the woolly bottle brush, and then we've done the reed cormorant. In addition to the Nguni goat, we've done the boa goat, the savannah, and the speckled goat. Also not included here is the wildlife species like your lion and the southern white rhino. So the benefits and future prospects from this is that crossbreeding efforts, they place the indigenous species at a vulnerable space, and this runs the risk of extinction, prompting the need to establish conservation programs to save these unique species. So for this purpose, generating a high quality reference genome is a right step to realize the efforts and potential for future research to unlock more benefits from African indigenous species. This is also to link genomics, to the bioeconomy. Therefore, we need a continued investment and collaboration to realize these opportunities. So with all of this, I conclude with saying that having a high quality reference genome that is accessible to researchers is essential as a genomic tool to establish efficient breeding programs. It is to also improve the production traits and conservation strategies to guard against the extinction of indigenous genetics resources. I think as a bonus as well, and just to pique interest on the audience, it's if you are interested in reading more about the work that we're currently doing, we have recently published a paper. This was in collaboration with many colleagues from the Africa BioGenome Project. This was published on the 10th of July. So this doesn't end there with just the 30. We do have a target of 100 genomes for 2025. I know that we have about 4 months left in this year, but we're quite optimistic, and we also want to finalize OmniC for the remaining genomes that do not have OmniC. And so with that, I'd like to thank the organizers for the opportunity and also my team members for the work that they put into this. Thank you so much.
Thank you, Sinebongo. I hope you've had the applause that you were given here, and this goes to the entire team, and give our best wishes to Professor Ntanga. But please hang on. We have one presentation that will come after, and then we can have some group questions for all the panelists. And we're changing gears a little bit. Yesterday I mentioned that Africa BioGenome Project, in partnership with our collaborators, is offering fellowships that are free to African scientists for you to go and have hands-on experiences in top-notch labs. One of our first beneficiaries was Mr. Frederick Kebaso, who is a bioinformatician at the ISIPE, International Center for Insect Physiology and Ecology in Nairobi. He's studying his PhD at University of Nairobi, and his doctoral research is on identifying drug and vaccine targets for contagious bovine pleuropneumonia. So Frederick, before you and online, you have very, very young many early career scientists who are very eager to hear how you ended up getting a fellowship and what your experiences was. Thank you. Please go ahead. So we can't hear you. Maybe you unmute yourself and we'll perhaps be able to hear you.
Okay, what about now?
Yes, now you're loud and clear. Go ahead.
Okay, thank you for the introduction. However, I'll just make a few corrections to the introduction, though I had not communicated it. I've been a bioinformatician at ISIPE, but currently, starting from August, I'll be based at Cambridge, Walter Reed, Kericho. Now, as stated, I was a beneficiary of the
Frederick, you have frozen. As we are trying to reconnect with Frederick, we can take a few questions for the three panelists. We've had three groups presenting their work. If you have any question for any of them, please go ahead and ask the question, and we'll also be looking out for questions that could be online. Maybe you can check for us whether there's a question online. Yes, there's a question here.
Thank you very much. My question to Ansia and also the same to Sadiq is that you haven't shown in your presentation how much sequencing data you actually used, both for ONT, Hi-C, and RNA-seq for Ansia and for Sadiq in terms of PacBio, Hi-Fi, and Omni-C. And I also wanted to correct that it's not the first African genome sequenced on ONT on the African soil, because my group was first. We sequenced Africa yam bean genome some time ago. It was published end of 2024, and we sequenced that using Hi-C and Oxford Nanopore long reads also.
So you can go ahead, Ansia and Sadiq.
Good. Thank you, Prof. I think I will go first. Yeah, so that's, I think, a good spot. I didn't mention the amount of data sets we have used for the assembly. So it was about 16x PacBio Hi-Fi reads. The estimated genome size is around 800 megabase. So we noted that was around 16x coverage. The Omnis read is around 2.5 million reads. So that's the amount of data we used for the assembly. I don't know if that answered the question.
Any contribution from Ansia on this?
Yeah, so for our ONT, I think we had about 30x coverage of the genome, and for the high C reads, there was about 400 million raw reads. So we had pretty good coverage. And yes, Jean-Marc, you are right. We are not the first to sequence ONT on African soil. That is correct.
Yeah. So I think just to add to what Rebecca mentioned, this is thank God here. So I think for the PacBio, yeah, because I understand that for ONT and high C, there was the lab genome by Olu and persons in your team. But for the PacBio and OmniC, I think this has not been done before, because OmniC technology just actually came quite recently, and our partner in South Africa probably one of the first people, and this is the first time it's been applied. So I think, yeah, just for the clarification on both, essentially, yeah.
Maybe to add that the first plant genome, there might be an animal genome. I'm
yeah,
not sure.
The first, the focus is on plants, yeah.
Yeah, the first plant genome with PacBio Hi-Fi sequenced on ground, sequenced and analyzed on ground in Africa.
Thank you for that clarification. Do we have another question in person? And maybe, Paul, you could check whether there are any questions online.
Yeah, I think we have 2 questions online, Stephen and Talent. So do you want to unmute, both of you?
Hello, everyone.
Hello, we can hear you.
My name is Stephen Buru. I am a BSc student at Jomo Kenyatta doing biochemistry and molecular bio. And I will want to ask a question to the one speaker who did the white star apple. I have 2 questions. How feasible will it be to engineer a synthetic tissue or metabolite pathway in an organism whose genome has just been sequenced through AfricaBP? And also, what are the steps required before one could apply bioengineering into it? And also, the second question is, once a genome is assembled, what strategies do you use to connect the sequence data to actual gene functions, especially for the traits that might be useful in regenerative medicine or synthetic biology? Thank you.
Thank you. We can take the next question so that we can answer them together. Oh, thank God you can read the question for us. Please go ahead and ask your question. There's a question here. And then after this, we can ask them all together.
I've seen Magule. Your hand is up. You can ask your question, please.
Okay. Can you hear me? Thank you very much.
Yes.
Yeah. So thanks, Dr. Anissa Sadiq and Dr. Simbonko for excellent work and the informative presentation. I have 2 questions. One is if you guys are considering the AI and machine learning aspects to enhance the genome assembly, particularly to improve speed, accuracy, and also other aspects. And I have the same question also for you coordinators, if there is a subcommittee in ABP specifically linking the bioinformatics pipeline to AI, and if there is such a group, such a team, how do some professionals, African professionals, join that team? Thank you very much.
Okay, so we'll take a question from the floor. Maybe, thank God, you can talk about the subcommittees and the others will answer their question. And please go ahead and ask the question.
Hi again, my name is Ami, and I think I'll direct my question to Ansia. You talked about using ONT technology when you are sequencing your genome. A common approach is to integrate the technology with Illumina sequencing just to improve on the accuracy. Is this a plan for your genome, or are you satisfied with the accuracy of your sequence? Thank you.
Okay, so we can go around and answer the questions. Maybe we'll start with Ansia, then we go to Sadiq, then we go to Thangod. Go ahead.
Okay, I think I'll answer the last question first. So no, we didn't do Illumina short read sequencing or whole genome sequencing to kind of error correct for the genomes. I know that is a common approach to combine short read and long read to improve the accuracy. So we only used the ONT long reads, but we did do the super accurate base calling model, which does increase the accuracy quite significantly. And we excluded all of the low quality reads, anything below Q15. And then we integrated the high C data during the assembly step as well. So that is short reads and it can also help correct some errors. But the HiFiism assembler has a really good error correction step. And so that has been very useful, and it can still result in really high quality assemblies, even in the absence of, additional short read data for polishing. And then I think there was a question about AI. So we haven't integrated AI into the genome assembly steps from our end, but I think that it would be very useful in the future, especially since we've kind of gotten to the point where we've established some pipelines and, they work really well. And so we can test them on a few different species and then, use that for some machine learning, which will improve genome assemblies in the future. I think those were all the questions directed at me. So my vote is Sadiq.
Thank you. Sadiq?
Good. Thank you. I'll try to answer the question. So the first question regarding to metabolic engineering in the star apple, I think it's good to acknowledge that this is the first step, and we already have good assembly. Even the annotation is not that much really high quality. So the way we try to infer the protein is using protein homology and then just directly from the genome itself. So what we are envisioning is on the next step, we will be using high quality RNA-seq dataset to infer high confidence genes. So once we have that, the next step we probably validating individual genes and as gene of interest, how that can be probably functionally behaving. So that would be the next strategy what we are looking at. Again, with regard to AI, I would like to again point out we are more into using available tools. We are not heavily investing on the development side, but we still have a committee who dedicated for the AI, integrating AI part to the bioinformatics pipeline. Tangat can also back me up on that side. So that's what I would like to add.
Yeah, thank you, Sadiq and Ansia. So just to add a few things out, I'll just talk about the AI and then I'll talk about the sequencing aspect. My name is Tangod Ebenezer, and I'm helping to kind of coordinate some of this together with Professor Anne, a guy in Nairobi, Professor Bouabid in Morocco, Craig Yama in South Africa, and also Professor Julian Osuji in Nigeria. So two things, just to mention that the technology that Ansia used is not, so we used ONT and Hi-C, but the Hi-C was done using MGI G400 machine. Ansia, is that correct? So because previously we started out with Illumina doing the Hi-C, and then that was with the UNISA genome. So I can't find Professor Mugai Sinebongo. Is she being asked? I mean, I'm trying to spotlight her. Is she still on the call?
Yeah, I'm still here.
Yeah, so let me spotlight you so that you can. Yeah, so we started off with the data for the UNISA team, and we're also doing Illumina for animals. And when we started the sequencing with this SAMRC, we then tried to use the MGI G400 machine to actually test it. Answer if I'm correct, that's like the first time we tried to do the high C with such technology, if I understand, because we've been doing. Am I correct? You know.
No, the high C worked first time. I think what you might be thinking of is the STLFR
The
sequence.
STLFR, yeah, that's correct. Yeah, yes, absolutely. So the STLFR, so I think that is basically it, in terms of that. Then in terms of the integrating AI, just to mention that next year's conference or next year regional workshops will be focused on artificial intelligence dedicated to biodiversity. So perhaps whoever asked the question on the AI is kind of few months ahead or faster than us. Well, we will be launching the advert in October, for all of the thematic areas next year. So I will be focusing on AI, but we do have a data science and artificial intelligence subcommittee. We also have a paper coming out soon in that aspect. So, but as Sadiq mentioned, we are not actively investing on developing tools because there are so many genomics tools, and it's just for you to adapt them and kind of do your work. However, we are also working internally on developing tools like protocols and pipelines that we use. So an example is, for instance, we've had a situation where genomes did not, the sequencing or library prep did not quite work the first time, and then we optimize protocols and get them to work. Or we have a situation whereby the bioinformatics tool did not work the first time, and then of course Sadiq and Ansia, you guys have been involved in all of those things. So all those things could be a form of developing pipelines, but they are not production level or production ready pipeline. They are tools that, for instance, Sadiq can use or Ansia, or Sadiq can pass on to Ansia to use, but they are not there on GitHub, for anyone to use, because there are just so much that needs to go into it. For instance, if you're going to put it on Nextflow, then the documentation needs to bear and things needs to be captured quite correctly. And just to also mention that I think I don't know if anyone has any question for Snebongo, Professor Ann. But the sheep genome has also been quite interesting to see all of this that has been done. And just to mention that all this started off, there has been, when we started, the question is, can Africa do this? When I, going around the world, going around Africa, trying to make a case for this, the question I keep getting, in fact, in one of the conference in Germany is, can Africa do this? Then I said, even if we have one sequencer, if we have one bioinformatician, we have to do this. And I think this is what will also be a motivation for every one of us. I do believe that these things can be done. Sadiq and Ansia and Snebongo have also showed that, we can actually do this. And I hope that this will be a form of inspiration to all African institutions, because we want to see more African institutions doing this. We want to see more African institutions investing in this. And just to mention that majority of the investment that, that drove this presentation actually came from 100% African institutions, or at least 60-70% contribution from the African institution. An example is the data that Nebongo presented is 100% from their institution. There is no international funding. There is no funding from the World Bank and all of those things, and that is huge. The presentation from Sadiq is 100% local funding. And in one form, it was also kind of a crowdsourcing funding. The presentation from Ansia is, we had the SAMRA team, and also it's a partnership with the MGI-Tech company. And all of these, when I look at all of this, I see the possibilities, things that are possible that we can do. And that is one of the reasons why we have this workshop, to share these experiences, to provoke action, to inspire people to action, and also to drive everyone for us to continue to move in the same direction and kind of, have the Africa we want continentally. Thank you, Professor Ann, and for this, over to you.
Yeah, thank you.
And Snebongo, I don't think Snebongo has responded to her question yet.
Yes, I was going to invite her to respond, but just before Sinebongo comes on board, let me just say that the AfricaBP concept note was written by, thank God, he's the one who pulled us all together to and he made us buy into the vision, and thank you for that. And because of a dream of a young African scientist, we have AfricaBP present today. So Sinebongo, there were questions, and I'm sure you have some comments to make. And also, just tell us about the investment that your institution has made, and not necessarily in the amount of money, but it must have taken quite a bit to get your university to invest in a Revio, which doesn't cost $1, it costs many more dollars. And what can we do as Africans to sell this dream to our heads of institutions? Thank you.
All right, thank you so much for that. So I think I'll start off with the AI question. So when we went to PEC, we already started playing around with annotation, but I think for some time the accuracy of that we're not happy with that. Then we found researchers that were using like deep learning resources or methods. And a colleague of mine who's very invested in our annotation, Tendo Chilate, he adopted that. It's Tiberius. I think it is on GitHub, the workflow. So whoever is interested in that can go and look at that. And that's what we currently using moving forward with the annotation. And then secondly, I think I saw a question on how do we connect the genomics to improving breeding? So that one, we go back to the smallholders and look at the improvement of traits, which is something that we haven't done, but it's definitely what we will venture into once we are settled and at a sweet spot with the genomic work and genomic assembly that we're doing. So definitely we believe that it will pour into our smallholder farmers and improve their breeding strategies. And then the investment part, I think it has helped a lot partnering with with the commercial sector. And in this instance, it is Ingaba. I think everybody is familiar with that. So our institution, there was a lot of convincing between them and then having them invest into this project. And I think having the basis and the institution seeing the work that has started, and this is thanks to the collaboration with AfricaBP as well. And then they also saw that it's quite touching into many other institutions, the collaborations that are there. So I think that's how they came on board into investing. And then they also saw that it drives down the expenses of sequencing compared to the previous years. So that's how we got to where we are at the moment.
Thank you. This has been an excellent session. We've been able to showcase that what we've been talking about can actually be done. It started off with one genome, and now you can see it's escalating. We're talking about, at the end of this year, 100 genomes coming out of University of South Africa. And as the other workflows and processes get stabilized and optimized, I'm sure that will ripple into the 100,000 genomes that we've been talking about. So let's give a round of applause to this particular session. Our AfricaBP project fellow, Mr. Kebaso, has a session in the afternoon, and he'll combine his presentation together with the one that he's doing for the practical. So right now, I'd like to move on to the next session, which is
Professor Mugai, just one moment, please. And just to
Yes.
add that, I think, just to kind of support what I mentioned previously, that hopefully when we come back to the East and Central Africa Regional Workshop, we would have institutions, sequencing with us as we've done, essentially, more genomes from Kenya and Uganda. And hopefully this could be something we could maybe discuss further. I did know that we have like several practicals coming out from, for instance, MKU. This is one of the platforms we can begin to look into. Because when I look at all of these local investments by African institution, and I say that it's not quite a lot. It's quite a lot of money, but it's sometimes you look at it, you're like, okay, some African institutions can do this. So perhaps let's have that discussion and carry it ongoing. Hopefully in 2026, we will be presenting more genomes from Kenya and by Kenyan institutions from Uganda, Tanzania, and Congo DRC. Sadiq has one more comment.
Go ahead, Sadiq.
Yeah, sorry, Target. Yeah, I mean, I just want to highlight that if there's anyone who would like to contribute to this endeavor, just please email to info@africabp.com, so you can just send an email, and then express your interest, which committee, which part of the work you would like to involve in, because I saw that part coming out in the conversation.
And indeed, you're right. Dr. Thank God, we are having those conversations. And indeed, MKU is one of the institutions that has invested heavily in getting sequencers. And we will be beginning our initiative in Eastern Central Africa. This is a rallying call to scientists in Central Africa. We are looking for an institution where we can anchor the African BioGenome there. We are also excited by the new partnership and interest from Somalia, and we're looking forward to more institutions. In Ethiopia, we have 2 strong partners, and we're looking for even more institutions, individuals, and projects to come on board and partner with us. And indeed, if you go to the web and just type in African BioGenome Project, it will take you to our website. In the contact form, fill it out. Those emails come to me. And we said this is an African initiative. So wherever you are, there is a role for you. Whether you're a student, a scientist, a graduate fellow, there is a place where you can act. And we're especially looking for scientists who can even volunteer their time to work in the subcommittees of the African BioGenome Project. So thank you very much. Let's give a round of applause to these. It was a great session, and I really do thank you all, because in 2023, when we started the workshops, it was a dream that we will sequence, we will, but now we can actually show that we are sequencing and we are publishing. So we're changing gears a little bit. We are not only interested in having scientists coming and giving presentations. We are. That's how the science moves. But in addition to that, we're very, very keen, as you have seen, in mentoring students. And we have posters as you're walking out of the corridor. Please step by the posters that you see displayed there. And we also want to mentor the students in this thing of science engagement and science communication. So we have a presentation from Gitaka Lab, which is a lab based at Mount Kenya University. We have students from the lab coming to present their work and tell us what they do. And this session is going to be coordinated by Dr. Kuja. Welcome.
Thank you, Prof. Migai. And the session now to know more about the Gitaka Lab at MKU. And MKU is one of the top partners to AfricaBP. And today we are going to listen to some of the projects that are ongoing. In addition to what you experienced yesterday from Kanoi and even Mark. So today we have Alfred Kibungei. You have 1 minute, Kibungei, to tell us why you must stop by your poster and listen to you. Welcome.
So I'll be the timer.
Yeah, thank you so much. Good morning, everyone. Yes, my name is Alfred Kibungei. I'm a master's student here at MKU. Additionally, I'm also a wildlife conservationist. So currently, I'm working on a project to look at what is the epidemiology of pathogenic organisms among the African lion. So this is inspired by the fact that in some parts of the country, specifically in Laikipia, people live very close with wildlife, particularly lions. And the fact that lions are very important species in the ecosystem, they are the apex predators. They additionally are charismatic species. And I'm sure everyone, if you're taken to a safari, you would want to see a lion. So because of their importance, it is worth studying what are the threats to this population. And IUCN lists these species as vulnerable. Some of the threats include human-wildlife conflicts, diseases, and illegal wildlife trade. So most efforts have been put into addressing other threats, but diseases remain lagging behind. So my aim of this project was to look at what are the diseases that form threats to this particular population, and are these diseases also found among other species, say, domestic animals and humans. So I have a poster just by the entrance of this hall, very beautiful, to show preliminary results from metagenomic analysis of eDNA from 96 samples to see what are the pathogenic organisms found in these lions. So pass by, let us discuss what this means to the lion population, the health of people around these lions, and the rest of us. Thank you.
Thank you. You've taken exactly 1 hour and 46.
1 hour?
Oh no, 1 minute 46 seconds. So
1 hour. That's almost 2 minutes.
That's almost 2 minutes.
Wonderful, wonderful project there.
Yeah, it should be
usually,
under 1 minute.
Usually in science, at times you have 1 hour to present, 45 minutes, 30 minutes. Sometimes for defense, 15 minutes. At times, 5 minutes. And at times, you have to do 1 minute. And if you can fit in within that time frame, then you're a super scientist. And we can feel that from Gitaka Lab now. Next, you have Rachel Kimani.
Good morning, everyone. My name is Rachel Kimani. I'm a postdoc fellow at the Gitaka Lab. My work was on AMR, and in future, all of us here are at risk of dying from very simple infections. Maybe you can imagine getting hospitalized because of just a boil. In this context, patients who are very ill or immunocompromised in the hospitals are the ones that we are very concerned about. Simple bacterial infections means that they stay longer at the hospital, increasing the cost of healthcare. So my work was at Thika level 5. I isolated bacterial isolates from clinical samples and also from the environment. I did antimicrobial susceptibility testing, and the resistant phenotypes were subjected for whole genome sequencing through the ONT technology. So if you walk down, you'll see my poster, the findings. Apart from measuring AMR at our local hospital, what else now? We have local data, which at the moment we are trying to push for policy change. Thank you.
Thank you. 1 minute 17 seconds. So
Wonderful,
we are doing well.
Wonderful. Dr. Kimani, wonderful. Next, you have Samuel Nganga.
So, good morning. I am Nganga Samuel, and I'm pleased to stand before you this morning to present findings of a recent study on wastewater epidemiology in the context of SARS-CoV-2. So we know so well that traditional surveillance systems are usually time-consuming, labor-intensive, and in resource-limited settings like in Kenya, usually we have limited resources for the same. And so wastewater-based epidemiology is usually non-invasive, is also scalable and powerful. And this has been mentioned before in the context of One Health. So we sampled wastewater from hospital, the level 5 effluent, some estates or other apartments, MKU, and also the municipal wastewater treatment plant. And we were able to, using quantitative RT-PCR, we were able to detect SARS-CoV-2 RNA, that is 2 gene targets, N and also ORF, ORF1AB. And we found out that 16.5% of all the sampled wastewater was positive for the same. This wastewater was also, we also carried out NGS, wastewater metatranscriptomics, and through that, we were also able to get some viruses such as rotavirus A, which was predominant in all the wastewaters, and stand by my poster, the last one, the corridor, as we engage more. Thank you.
Thank you. You've taken 1 minute 46 seconds.
So in about 7 minutes, we've had a lot of science, very technical science. We've talked techniques, we've talked objectives, We've talked, the reasons why some studies are being done. We are not taking any questions because I want you to go ask the questions at the poster. So please, let's give them a round of applause. I was joking with somebody yesterday that by the time you're leaving for a conference and boarding the plane, you've been given 30 minutes to present. By the time you're reaching your hotel, you see an email saying you have 20 minutes to present. Then when the time comes, just before you're entering, you're told, I'm sorry, you only have 10 minutes. So you've left your country with a 30 minutes presentation, but just when you're about to present, you have 10 minutes. So you have to be versatile as a scientist. You have to be ready to give the 1 hour talk, because they could also say, oh, that presenter has not come. So you have 1 hour and then you only have a story for 10 minutes. So you have to be versatile to either shorten your talk or also increase. And I think this is a great example. And Gitaka Lab is really, really out there doing it. So let's give them another round of applause for that. Yes, and we're going to take a 30 minutes break now. So those of you who are online, please join us in 30 minutes. Those of you who are here, we can just walk out. Let's go view the posters as we take some refreshments. Thank you, and let's be back here in 30 minutes.
Diseases. Analyze genomic variations in tumors. Project outbreaks through surveillance monitoring. Combat drug resistance. Discover mutations for disease monitoring. Identify analyze RNA disease drivers. Develop simple. With breakthrough simplicity, our fastest runtimes yet, everything in one footprint with onboard Dragon analysis and significant sustainability advancements. This is the highest standard in benchtop sequencing, empowering every lab everywhere on a quest for discoveries that can't wait. This is the MySeq i100 series. This is sequencing made simple.
Welcome to Inkaba Biotech East Africa Limited. The company was started in the year 2010 on the 23rd of June. Over the years, we've grown into a fully fledged one-stop molecular biology shop. And currently, we're also running one of the best labs in the region. Join me as I take you through the tour of our laboratory. Welcome. Here we have our engineering department, whereby we service our equipments here, equipments which that we can carry from the client site to our offices. Basically, these are small equipment. For the large equipments, we prefer servicing them at the client's place. Here we also do calibration services. For all of you, those who have pipettes that need calibration, we now proceed to our extraction room. This is where we do all our extractions. Once we've received samples, this is where it all starts. Our scientists and qualified personnel will look at your samples, record them the way they are supposed to, and take all the details and also report on what they've received. From there on, they will proceed. This is our PCR room. As you can see, we have scientists who are also very busy doing their work here. We have timelines whereby we try as much as we can to give our clients results in the shortest time possible. As you can see, our lab is fully equipped and work is progressing, as you can see. So we are looking forward to having our clients approve our services and also happy at the same time as we continue to provide solutions, molecular solutions, and also solving research problems in the region. Here we have our post-PCR room. This is where we do our sequencing services, clients, after they've sent their samples, gone through the extraction, gone through the PCR, this is now where we do our sequencing. As you can see, our machine is fully running throughout because we have a lot of samples and we have a lot of samples coming in each and every moment. Here we have our data analysis room. As you can see, one of our researchers is also seated analyzing data, which has already been gotten from the machine. Hereby, we have our gel documentation room. This is where we do all our gels. As you can see, we have one of our researchers already trying to load the gels and also run them in the machine called the e-box. This is from a company called Wilbur. They have very quality gel documentation systems.
The core service we provide is distributorship and sales services. We sell our products across the extended Eastern Africa region. All the countries in the East African region, ranging from the DRC, we have Rwanda, we have Uganda, where we have a branch office, we have Tanzania, we have Burundi, Kenya, Ethiopia, Somalia. Our sales are basically for the molecular biology field and the general lab. In the molecular biology field, we provide products and solutions for all the processes, the entire workflow from sample collection, DNA, RNA extraction, PCR, amplification, both reagents and instruments, and sequencing and bioinformatics. In the general lab, we offer equipment. We supply equipment, including but not limited to freezers, to fridges, to biosafety cabinets, and many more. So you can always come to us for any reagent, consumable, or equipment need that you have for your lab. We also have a line of diagnostics, molecular diagnostics, both for plants, animals, and humans.
So we like to welcome all of you to come utilize our services here in 74 Riverside Drive. Our lab is open to students. Our lab is open to researchers who want to do their work here. We provide space, we provide the equipment, and we also provide the reagents and chemicals that you may need for your research work. I welcome you all. Thank you.
Study infectious diseases. Analyze genomic variations in tumors. Project outbreaks through surveillance monitoring. Combat drug resistance. Discover mutations for disease. Rapidly identify analyze RNA drivers. Develop simple. With breakthrough simplicity, our fastest runtimes yet, everything in one footprint with onboard Dragon analysis and significant sustainability advancements. This is the highest standard in benchtop sequencing, empowering every lab everywhere on a quest for discoveries that can't wait. This is the MySeq i100 series. This is sequencing made simple.
Welcome to Inkaba Biotech East Africa Limited. The company was started in the year 2010 on the 23rd of June. Over the years, we've grown into a fully fledged your one-stop molecular biology shop. And currently, we're also running one of the best labs in the region. Join me as I take you through the tour of our laboratory. Welcome. Here we have our engineering department, whereby we service our equipments here, equipments which that we can carry from the client site. to our offices. Basically, these are small equipment. For the large equipment, we prefer servicing them at the client's place. Here we also do calibration services. For all of you, those who have pipettes that need calibration, we now proceed to our extraction room. This is where we do all our extractions. Once we've received samples, this is where it all starts. Our scientists and qualified personnel will look at your samples, record them the way they are supposed to, and take all the details and also report on what they've received. From there on, they will proceed. This is our PCR room. As you can see, we have scientists who are also very busy doing their work here. We have timelines whereby we try as much as we can to give our clients results in the shortest time possible. As you can see, our lab is fully equipped and work is progressing, as you can see. So we are looking forward to having our clients approve our services and also happy at the same time as we continue to provide solutions, molecular solutions, and also solving research problems in the region. Here we have our post-PCR room. This is where we do our sequencing services. Clients, after they've sent their samples, gone through the extraction, gone through the PCR, this is now where we do our sequencing. As you can see, our machine is fully running throughout because we have a lot of samples and we have a lot of samples coming in each and every moment. Here we have our data analysis room. As you can see, one of our researchers is also seated analyzing data, which has already been gotten from the machine. Hereby, we have our gel documentation room. This is where we do all our gels. As you can see, we have one of our researchers already trying to load the gels and also run them in the machine called the e-box. This is from a company called Wilbur. They have very quality gel documentation systems.
The core service we provide is distributorship and sales services. We sell our products across the extended Eastern Africa region. All the countries in the East African region, ranging from the DRC, we have Rwanda, we have Uganda, where we have a branch office, we have Tanzania, we have Burundi, Kenya, Ethiopia, Somalia. Our sales are basically for the molecular biology field and the general lab. In the molecular biology field, we provide products and solutions for all the processes, the entire workflow from sample collection, DNA, RNA extraction, PCR, amplification, both reagents and instruments, and sequencing and bioinformatics. In the general lab, we offer equipment. We supply equipment, including but not limited to freezers, to fridges, to biosafety cabinets, and many more. So you can always come to us for any reagent, consumable, or equipment need that you have for your lab. We also have a line of diagnostics, molecular diagnostics, both for plants, animals, and humans.
So we like to welcome all of you to come utilize our services here in 74 Riverside Drive. Our lab is open to students. Our lab is open to researchers who want to do their work here. We provide space, we provide the equipment, and we also provide the reagents and chemicals that you may need for your research work. I welcome you all. Thank you.
Study infectious diseases. Analyze genomic variations in tumors. Project outbreaks through surveillance monitoring. Combat drug resistance. Discover mutations for disease monitoring. Identify analyze RNA disease drivers. Develop. Simple. With breakthrough simplicity, our fastest runtimes yet, everything in one footprint with onboard drag analysis and significant sustainability advancements. This is the highest standard in benchtop sequencing, empowering every lab everywhere on a quest for discoveries that can't wait. This is the MiSeq i100 series. This is sequencing made simple.
Welcome to Inkaba Biotech East Africa Limited. The company was started in the year 2010 on the 23rd of June. Over the years, we've grown into a fully fledged one-stop molecular biology shop. And currently, we're also running one of the best labs in the region. Join me as I take you through the tour of our laboratory. Welcome. Here we have our engineering department, whereby we service our equipments here, equipments which that we can carry from the client site to our offices. Basically, these are small equipment. For the large equipments, we prefer servicing them at the client's place. Here we also do calibration services. For all of you, those who have pipettes that need calibration, we now proceed to our extraction room. This is where we do all our extractions. Once we've received samples, this is where it all starts. Our scientists and qualified personnel will look at your samples, record them the way they are supposed to, and take all the details and also report on what they've received. From there on, they will proceed. This is our PCR room. As you can see, we have scientists who are also very busy doing their work here. We have timelines whereby we try as much as we can to give our clients results in the shortest time possible. As you can see, our lab is fully equipped and work is progressing, as you can see. So we are looking forward to having our clients approve our services and also happy at the same time as we continue to provide solutions, molecular solutions, and also solving research problems in the region. Here we have our post-PCR room. This is where we do our sequencing services. Clients, after they've sent their samples, gone through the extraction, gone through the PCR, this is now where we do our sequencing. As you can see, our machine is fully running throughout because we have a lot of samples and we have a lot of samples coming in each and every moment. Here we have our data analysis room. As you can see, one of our researchers is also seated analyzing data, which has already been gotten from the machine. Hereby, we have our gel documentation room. This is where we do all our gels. As you can see, we have one of our researchers already trying to load the gels and also run them in the machine called the e-box. This is from a company called Wilbur. They have very quality gel documentation systems.
The core service we provide is distributorship and sales services. We sell our products across the extended Eastern Africa region. All the countries in the East African region, ranging from the DRC, we have Rwanda, we have Uganda, where we have a branch office, we have Tanzania, we have Burundi, Kenya, Ethiopia, Somalia. Our sales are basically for the molecular biology field and the general lab. In the molecular biology field, we provide products and solutions for all the processes, the entire workflow from sample collection, DNA, RNA extraction, PCR, amplification, both reagents and instruments, and sequencing and bioinformatics. In the general lab, we offer equipment. We supply equipment, including but not limited to freezers, to fridges, to biosafety cabinets, and many more. So you can always come to us for any reagent, consumable, or equipment need that you need. you have for your lab. We also have a line of diagnostics, molecular diagnostics, both for plants, animals, and humans.
So we like to welcome all of you to come utilize our services here in 74 Riverside Drive. Our lab is open to students. Our lab is open to researchers who want to do their work here. We provide space, we provide the equipment, and we also provide the reagents and chemicals that you may need for your research work. I welcome you all. Thank you.
Study infectious diseases. Analyze genomic variations in tumors. Project outbreaks through surveillance monitoring. Combat drug resistance. Discover mutations for disease. Rapidly identify pathogens. Study disease drivers. Develop simple. With breakthrough simplicity, our fastest runtimes yet, everything in one footprint with onboard Dragon analysis and significant sustainability advancements. This is the highest standard in benchtop sequencing, empowering every lab everywhere on a quest for discoveries that can't wait. This is the MySeq i100 series. This is sequencing made simple.
Welcome to Inkaba Biotech East Africa Limited. The company was started in the year 2010 on the 23rd of June. Over the years, we've grown into a fully fledged your one-stop molecular biology shop. And currently, we're also running one of the best labs in the region. Join me as I take you through the tour of our laboratory. Welcome. Here we have our engineering department, whereby we service our equipments here, equipments which that we can carry from the client site to our offices. Basically, these are small equipment. For the large equipments, we prefer servicing them at the client's place. Here we also do calibration services. For all of you, those who have pipettes that need calibration, we now proceed to our extraction room. This is where we do all our extractions. Once we've received samples, this is where it all starts. Our scientists and qualified personnel will look at your samples, record them the way they are supposed to, and take all the details and also report on what they've received. From there on, they will proceed. This is our PCR room. As you can see, we have scientists who are also very busy doing their work here. We have timelines whereby we try as much as we can to give our clients results in the shortest time possible. As you can see, our lab is fully equipped and work is progressing, as you can see. So we are looking forward to having our clients approve our services and also happy at the same time as we continue to provide solutions, molecular solutions, and also solving research problems in the region. Here we have our post-PCR room. This is where we do our sequencing services. Clients, after they've sent their samples, gone through the extraction, gone through the PCR, this is now where we do our sequencing. As you can see, our machine is fully running throughout because we have a lot of samples and we have a lot of samples coming in each and every moment. Here we have our data analysis room. As you can see, one of our researchers is also seated analyzing data, which has already been gotten from the machine. Hereby, we have our gel documentation room. This is where we do all our gels. As you can see, we have one of our researchers already trying to load the gels and also run them in the machine called the e-box. This is from a company called Wilbur. They have very quality gel documentation systems.
The core service we provide is distributorship and sales services. We sell our products across the extended Eastern Africa region. All the countries in the East African region, ranging from the DRC, we have Rwanda, we have Uganda, where we have a branch office, we have Tanzania, we have Burundi, Kenya, Ethiopia, Somalia. Our cells are basically for the molecular biology field and the general lab. In the molecular biology field, we provide products and solutions for all the processes, the entire workflow, from sample collection, DNA, RNA extraction, PCR, amplification, both reagents and instruments, and sequencing and bioinformatics. In the general lab, we offer equipment. We supply equipment, including but not limited to freezers, to fridges, to biosafety cabinets, and many more. So you can always come to us for any reagent, consumable, or equipment need that you have for your lab. We also have a line of diagnostics, molecular diagnostics, both for plants, animals, and humans.
So we like to welcome all of you to come utilize our services here in 74 Riverside Drive. Our lab is open to students. Our lab is open to researchers who want to do their work here. We provide space, we provide the equipment, and we also provide the reagents and chemicals that you may need for your research work. I welcome you all. Thank you.
Study infectious diseases. Analyze genomic variations in tumors. Project outbreaks through surveillance monitoring. Combat drug resistance. Discover mutations for disease rapidly identify analyze study disease drivers. Simple. With breakthrough simplicity, our fastest runtimes yet, everything in one footprint with onboard Dragon analysis and significant sustainability advancements. This is the highest standard in benchtop sequencing, empowering every lab everywhere on a quest for discoveries we can't wait. This is the MySeq i100 series. This is sequencing made simple.
Welcome to PacBio Biotech East Africa Limited. The company was started in the year 2010 on the 23rd of June. Over the years, we've grown into a fully fledged your one-stop molecular biology shop. And currently, we're also running one of the best labs in the region. Join me as I take you through the tour of our laboratory. Welcome.
All right, welcome back to the session. For those who came in early and did not register downstairs while arriving, you can scan the QR code to register for day 2.
Kindly please register and put in your details the way you want them to appear on your certificates. Please.
Great, so we are ready for the next session. And we will start with Omar Mahadale, who joined us yesterday for the online panel. He'll be giving a talk on advancing genomic research in Somalia, opportunities and challenges. And as Prof mentioned, Omar is the first person from Somali to basically give a talk in the AfricaBP conference, East and Central Africa. So I don't know if we have him online already. Omar, is he online?
Yes,
Great.
okay.
We can't hear you. Maybe you can unmute yourself.
Yeah. Am I audible now?
Yes, you are. Are you able to share your presentation?
All right.
Great.
Mm. Mm. I think it says the host should allow for me to share.
Just a few moments.
All right.
Does he have the permissions to share now? Okay. All right, could you try again?
Still I can't.
All right.
I also emailed, so maybe you could share it from your side.
I think that would be okay. Maybe share it so that we can project it from this side. So in the meantime, maybe we can have another presenter go before you, if that's okay. Just 15 minutes, and then you can come after.
Definitely, that's fine, yeah.
All
Thank
right,
you.
thank you so much. So we'll have our presentation from AfricaBP Biotech East Africa, and AfricaBP has partnered with us since the inception of the Africa BioGenome Project. And today we will have a talk from Victoria, who will be representing William Nyakundi and Dennis Kivuva and the rest of the team. And we're very grateful for your support. So we're looking forward to hearing what you have to share with us today. Karibu.
So good afternoon. I'm Victoria Wambua, representing AfricaBP Biotech. If you're from the south, you know that AfricaBP has a click sound, which I can't do, sorry. So I'll be taking you through the introduction of our company, what we offer, and I'll also give an overview of what the participants for the workshop will expect when they come to our facilities. So if I may start. Okay, sorry. So in Kaba, we are Africa Genomics company. We do distribute for several manufacturers based in the US, Europe, and various places in the world. And some of our major brands are listed in that slide. So I bet you've used some of them. So yes, on to the next. Sorry. So our products are molecular biology, reagents, biochemicals. We also have molecular diagnostics and bioinformatics softwares for phylogenetics, DNA sequences, analysis, primer design, and many more. So we also offer products for cell biology, plastic consumables which are used every day in everyday lab usage, proteomics, and food testing. So I'm going to highlight some of our major products. So we'll start with the DNA and RNA purification kits. We do supply this from Zymo Research, and the kits are they do offer on extraction high quality DNA and RNA for any downstream applications, including PCR and next generation sequencing. They come in different formats. We have the spin columns for the manual extraction, magnetic beads for automation, and 96 well plates, and we offer kits for extraction from all sample types. Then we have for molecular reagents for DNA, RNA, and proteins from New England Biolabs. So these include DNA polymerases and amplification technologies, RNA reagents for cDNA synthesis, cleanup and isolation, protein expression and purification technologies, ladders and sequencing kits and reagents for the commonly non-sequencing platforms like Illumina, MGI, and Oxford Nanopore. And we also have the molecular diagnostics. We offer that from Seegene. We have all diagnostic assays in one platform. These kits actually use the syndromic assay approach, whereby the kits actually detect depending on a syndrome, the patient syndromes, sorry. Then we have oncology solutions. We offer from Gene Verify. This is a company that offers real-time qPCR kits for oncology, for cancer diagnostics, prognosis, and risk assessment. Oxford Gene Technology for FISH and NGS oncology panels for accurate detection and diagnosis. Then we have Omixon device and Celemics. These are used on the NGS solutions. Then we have vaccine delivery and proteomics. We have recently onboarded a company, the Quantum SI. It's the first NGS protein sequencing platform, and this is ideal for testing efficiency, RNA-based vaccine delivery mechanisms, and other proteomics applications at a fraction of the cost of the existing technologies like mass spectrophotoscopy. So we also do supply instruments from various manufacturers. These range from centrifuges, PCR thermocyclers, biosafety cabinets, and pipettes. We also, apart from the supply of reagents, consumables, and equipments, we also offer services. These services are as listed, so DNA synthesis, that is, we do synthesize oligos and probes, Sanger sequencing, next generation sequencing, bioinformatics, qPCR analysis, animal genetics, instrument and service repair and maintenance, SNP genotyping and methylation analysis, and Agena mass array services. So some of these services, we offer them locally, and some are offered in our lab in South Africa. So I'll just highlight some of the services. So we have the DNA synthesis and probes, primers and probes. So the longest oligo we can synthesize will be 90 mers, and the longest oligo that we can modify, that is, have the 5-end and that end modifications, will be 50 mers. Then all oligos are provided lyophilized. This helps us with the transport of the stability of the primers during transport. And so once you receive the primers, we do recommend that you do a quick spin, because most of the time the tube looks empty because of the lyophilization. Then the scale, we also have different scales and different yields. So we also offer different oligo purification methods with our standard bin desalting, and we also offer the cartridge purification. So I'll just explain the desalted one. So this is our standard purification method for the primers, and it's to remove the small molecule impurities, and it's good for many techniques, including PCR, and it's acceptable for oligos that are below 35 bases. So if your oligo is, like, let's say 40 bases, we'll have to do the next method, which will be the cartridge purification. And this is done, purify with different reagents in a column. Oligo binds to the resin, wash, and elute. And this is recommended for the long and modified oligos. This would be the probes and any oligo that is more than the 35 bases. And also, the oligos that have inosines, we also do the RP cartridge. So you'd know, most of you probably know about the HPLC purification method for the primers. So we do offer the cartridge purification in place of the HPLC. And it's actually, it's proven to be a good purification method. Then we have the stability of the oligos. So we do, for long-term storage, whether the oligos are dried down or resuspended in water or TE buffer, it's optimal to store them in frozen at -20. For room temperature of 4 degrees storage, oligos are resuspended in TE buffer. So for oligos, you can, once you receive them lyophilized, you can resuspend them with TE buffer or water. But we do recommend the TE buffer because they are more stable when resuspended with the TE buffer. Then we also, the degradation is also affected when they're stored in low pH conditions. So if your TE buffer is not the optimal pH conditions, then sorry, the primers will degrade faster. Then these are some of the modifications we offer for the 5-end. So this, when you modify the oligo to be a probe, these are some of the modifications we offer, the 6-farm, X, CAF4, and several others. Then we do offer the quenchers, the black hole quenchers, BHQ1 and 2. Then these are some of it's a table that shows the different dyes that we offer that are compatible with different qPCR machines. So we've just listed a few of the qPCR machines in the market. Then we also offer Sanger DNA sequencing. So for this, we do offer it locally. Our Sanger DNA sequencing facility offers DNA sequencing from various templates using the brilliant dye terminator, cyclin sequencing on the ABI 3500 XL genetic analyzer. We strive to deliver a flexible service to serve most needs. We have a TAT of 2 to 5 days once we receive the samples in our lab. And the services that we also offer for Sanger sequencing, we offer primer design and production, template preparation. This will be the DNA extraction or RNA extraction in this case, application and purification, cloning, data analysis once the sequencing is done, species ID, including fungi, bacteria, insects, plants, tissues, etc. Yes, so we've just have a nice picture of the systems that we have in the lab. Then we also offer fragment analysis, which this is DNA fragment analysis is a molecular technique that is used to identify, compare, and interpret DNA fragments. So it's often used for genotyping, microsatellite analysis, mutation detection, paternity forensics, and genetic mapping. Then we do offer next generation sequencing. For this, we offer it as a service, as well as we do sell the equipments for this. So the NGS applications that we offer are whole genome sequencing, that is for humans, bacteria, plant, animals, viral genomes, transcriptome sequencing, differential RNA expression profiling, human exome, metagenomics, genome reduction libraries, and the rest. So I'll just add I'll just talk about these systems a bit. So we do offer the NGS sequencing on our PacBio platforms. It's from our manufacturer, the PacBio Life Sciences. They currently have 3 systems in the market, the 3 ones in the middle. So we have the Revio, which is a long-read sequencer, and it's mostly applicable for people who have large-scale projects. Then we have the Vega. The Vega is a benchtop long-read sequencer. Then we have the Onso, which is a short-read sequencer. And they all sequence using the SBB sequencing, which is sequencing by binding technology. And the data that you get from this is hi-fi data, and the accuracy is really amazing. Then we have metagenomic analysis. So we do offer metagenomic analysis using the PacBio systems, and we also have an Illumina system in our lab in South Africa, so we also offer this. And for metagenomic work, we do recommend that you use the Zemobiomic kits, and they really give amazing quality of DNA. Then we also offer qPCR analysis. This we do offer probe and dye-based assays using one of our manufacturers, one of our brands, which is the NEB Luna product line. Then various applications like the SNP genotyping, gene expression, library quantification. We also offer instrument repair services. So we also offer pipette calibrations. We also do centrifuge calibrations, qPCR calibrations, but with a little bias to the instruments that we do supply. Then animal genetics, this is zoomics, and we do offer DNA profiling, DNA parentage for pets, and many more. Then that will mark the end of my presentation. Last but not least, so I believe some of you have been selected for our practical workshop. So we will be offering Sanger sequencing and also bioinformatics. So I believe that those who got selected for these programs, you got an email from us. So we're looking forward to interacting with you, and we hope you learn a lot from us. And yeah, our offices are at Victoria Office Suites, that is at Riverside Drive. So if you have any queries, just get in touch with us, and we are looking to assist. Thank you.
Thank you so much, Victoria. And if you have any questions for INCABA, they have a desk out there. You can ask. You can reach out to them. So I don't know if we've been able to give the rights to Omar to be able to present. Not yet? Okay. Sorry about that. Omar, please hang on. We are working on it. So we'll go to the next presentation from Lewis Karani, who is from Change Biotech. And just to highlight, the reason we are having our technologies companies present here is you realize we do not charge you to participate in this conference. One of the missions of the Africa BioGenome Project is to build capacity in Africa to be able to achieve the mission that we have, which is to be able to deliver these genomes for Africa by Africans. So it is good for us to hear what they have to offer. And again, they continue to support us in developing these reference genomes. So Lewis Karani, karibu, and I hope you will keep time.
So good afternoon, everyone. Like you heard, my name is Lewis Karani. I am an application scientist. I work for Change Biotech Limited. I had 2 presentations, so I kind of like merged them into one. I was told I have 20 minutes. So I'll begin by introducing my company, Change Biotech. Change Biotech is, they call it an astute. Astute meaning we understand the client. And we're focused on bringing impactful change to the life sciences industry across East Africa. It was founded in 2020, so that will make it 5 years old. But now we offer a very tailor-made solution. Those who've worked with us, you know, our solutions are not broad-based. We tailor it to your science, to your questions, to your objectives. We actively engage in clients across East Africa. That's in Kenya, Uganda, Tanzania, Rwanda, Burundi, Congo, the larger East Africa in general. Then I'll spend a bit of time here on our current partners. We do partner with QIAGEN. QIAGEN is, I think, one of the leading companies in life sciences. They offer a range of products, all the way from extraction kits, PCR kits, equipment, more for PCR, for fragment analysis. They offer bioinformatics softwares. Basically, anything you might need for life sciences, you can find them at QIAGEN. We also do represent Oxford Nanopore across East Africa. Oxford Nanopore, I think you've had a few presentations of people who've sequenced with Oxford Nanopore Technologies. We are their official channel partner across East Africa. We represent stem cell technologies. When you think stem cell technologies, think cell biology, culture media, cytokines, antibodies, all stuff. MTC Bio, more of your plastics, your consumables, your biohazard bags, more so the daily lab consumables that you use. You can find them at MTC Bio. Benchmark, it has to, Benchmark, Scientific, Hamley Benchmark, and Accurities are more an umbrella, 3 companies under one umbrella. And they offer scientific equipments from centrifuges to electrophoresis machines to gel docs to mostly vortexes, all the equipments you think you might need in a lab, we can provide. And because I was here to present on our upcoming training for Oxford Nanopore Technologies allow me to focus, narrow down kind of my presentation to the Oxford Nanopore Technologies going forward. This is our contact information. We also do have a desk out there, so if you have any questions after the presentations, you feel free to email us. You can come out at the desk, and me or one of my team members will be happy to answer any questions you have. So this was kind of like the meat of my presentation. I was called here to present about Oxford Nanopore Technologies. And my platform is one platform, endless possibilities, because that's what truly it is. It's one platform giving you boundless possibilities. This is more of a disclaimer. My presentation may have forward-looking statements for improvements your Oxford Nanopore Technologies is looking to make. So it's just giving you a disclaimer that these are forward-looking statements. We are not sure the timelines that they may be implemented in. So what is Oxford Nanopore? Oxford Nanopore is a sequencing technology, scalable. Scalable meaning it's the same technology across mid-throughput, low-throughput, high-throughput sequencing systems. By one platform, I mean it uses one technology, and that is the nanopore. You sequence a DNA by passing it through a nanopore. There's an electrical signal that's passing through the nanopore. As the DNA strand is passing through the nanopore, it disrupts the electrical signal, which is then converted into a base call through a process called base calling. And it's suitable for any biology. If you want to do, we'll see in the coming slides. It's also, it's also an end-to-end platform, meaning we give solutions from sample prep to sequencing to analysis. So from preparing your libraries to sequencing them to doing bioinformatics, all under one roof with Oxford Nanopore Technologies. And like I said, it's one sequencing platform for all your biology. So if you're doing single nucleotide polymorphisms, you're doing indels, you're doing methylation studies, in epigenetics, you're doing structural variations, you're phasing, you're assembling genomes, Oxford Nanopore is the technology for you. And I feel interacting with people, I ask them, what do they understand about Oxford Nanopore Technologies? Many of them tell me they understand it as a long-read sequencing technology. I want to demystify that and tell you Oxford Nanopore is not only a long-read sequencing technology. It can it's sequenced from 20 bases all the way to 4 million bases. So you can sequence any length from oligos to, we call it read length agnostic. As long as you can extract a DNA of that fragment, we can sequence it. It's scalable, meaning you have personal sequencers. I think I carried mine. I'll show you. This is a sequencer, mind you. Amazing. This is kind of the journey of Oxford Nanopore. It was, I think it was founded in 2010, but the first commercial product was launched in 2014. And through the years, we've had co-improvements to our technologies. This was the latest one, the MK1D. So you can see we are committed to improving our technology, not only through giving more throughput sequencers, but also improving our chemistries. As you can see, we started with a Q8 quality score, able to sequence 30 base pairs. Now we're able to assemble genomes in the Q60 kind of quality frame. For human genomes and large karyotic genomes, we're in the Q20 plus chemistry, and I've kind of highlighted sequencers that you may need. We have personal sequencers, kind of like the one I have here. We call this the Minion MK1D, and we also have a PromethION. For Oxford Nanopore, we call it scalable because you only have two options, basically, two flow cells. We have a Minion flow cell and a PromethION flow cell, all with different data outputs. With the Minion flow cell, you get up to 50 GB of data. With the PromethION, you get up to 290 GB of data. So depending on the genomes you want to sequence or the sequencing depth you want, that informs the choice of the sequencer you go for. So we have personal ones, we have scalable ones, and we have high-put sequencers. If you're doing small projects, you go for the personal sequencers. If you are looking to centralize a lab, you go for the scale-up sequencers. And if you're doing population genomics or you have a project that needs very high throughput, you go for the PromethION family sequencers. So you might ask me why Oxford Nanopore? There are a lot of sequences here, Illumina, PacBio. So why Oxford Nanopore? Like I said, whatever the investigation you may need, we have the solution. I promise you, whatever you may feel you need to sequence, I can give you a solution. That's a promise. So with any technique, whole genome, whole transcriptome, whole exome, targeted sequencing, metagenomics, if you want to assemble complete genomes, if you want to do single cell sequencing, if you are doing cancers and you want to do fusion transcripts, you want to, the list goes on. We've made this biology accessible to anyone with end-to-end workflows, meaning we provide you a workflow for all the way from sample preparation to analysis, meaning you don't have to spend time optimizing protocols. We've already done that for you. You just tell me, this is the application I want, I give you a protocol. You start. All the way from research to clinical to applied genomics to industrial. Also something I should mention about Oxford Nanopore, we sequence native molecules, native DNA, native RNA, meaning you don't have to modify your DNA or your RNA, you sequence it as it's extracted. You've had a lot of sequencing by synthesis, sequencing by I don't know what, but for us, we sequence what is there. Like the DNA or RNA you extract is what we sequence. We don't modify it in any way, meaning any modifications in the DNA that already exist or in the RNA, including methylations, including any epigenetic modifications, we can tell you, and this is done in real time as well. So we offer gap-free coverage sequencing. We can do single nucleotide polymorphisms. We can do structural variations. We can check total DNA and RNA modifications, epigenetics. We can do end-to-end genes. We also scaffold chromosomes. I think the running theme of the Africa BioGenome Project is assemble genomes. You know, and when you assemble genomes, you need not only do sequencing, you need to do facing, you need to do scaffolding. We offer all this under one platform. So I've kind of highlighted the evolution of genome assembly. This is from Oxford Nanopore. In 2021, they were able to assemble a full telomere to telomere haploid sequence. Now look where you're at 2025, they've assembled a near telomere to telomere diploid assembly. Near telomere to telomere means, you know, DNA is packaged into chromosomes. And when you sequence whole genome, you need to kind of assemble it into chromosomes. Like humans, you have 20, is it 22 chromosomes? Scientists, 23, including the X and Y. So we are able to sequence chromosome 1 from end to end. One technology, one library prep. I don't know if you guys are understanding. So the kind of goal of Oxford Nanopore is to accelerate whole genome sequencing, making whole genome sequencing telomere to telomere, meaning when you're assembling genomes, you'll be able to assemble them from end to end without gaps. When if you've done bioinformatics before, you know, when you try to assemble genomes with short reads, it leaves a lot of gaps in your genome. So with Oxford Nanopore and long reads, you kind of like assemble it end to end, no gaps. And this, our aim is to make it the standard of sequencing. It's like we do prokaryotic genomes, we do eukaryotic genomes, including tetraploids, even plants. You know, plants have many, the ploidy of plants is very variable. You can have a plant with up to 6 septaploids. So we can sequence that and we can assemble it for you. For prokaryotic genomes, we're able to resolve it up to strain level. And this, remember, I told you, like the record for Nanopore assembled metagenomics is Q59. It's the gold standard for metagenomics, if I can say so myself. And like I said, we detect methylations, epigenetic modifications without extra library prep steps. If you want to detect something like methylation on an Illumina, you need to do bisulfite conversion. That's an extra library prep step. But with us, we detect it natively, meaning if it's there in your DNA, you don't have to do anything else. You just click a button, say, I want to know if my base is modified or not, and we'll be able to tell you. and this gives you deeper insights in your data. We also offer something, I only mentioned this, this is a technology that we've launched recently. It's called adaptive sampling. It means you can choose what to sequence. If you want to do targeted genomics, if you want to enrich for a certain region, you just tell, you give a bird file to you upload a bird file in your sequencing, tell it, if you see reads that match to this, don't sequence. If you see reads that match to this, sequence. That allows you to enrich for your target genome without doing any extra library preps as well. And this has a lot of applications. Like you can do whole exome with it, you can do cancer panels targeted, you can deplete for host. If you're extracting DNA from a microbe that is in a human blood, you can deplete for the human and only sequence selectively your host, your target genome. I mean, and like I said, it's scalable, meaning if you want to start small, we can start here, and if you want to do population-based genomics, we can scale you up to the Promethean families. I think my time is up, so again, all this in one sequencing run. To get the same kind of data, you'll need at least 3 library preps, 2 sequencing platforms, 3 separate data analyses. But with Oxford Nanopore, 1 library prep, 1 sequencing run, 1 data analysis. Thank you. For any questions, you're out there. Finally, as Change Biotech, we have the pleasure to kind of like show you this technology in action. Starting tomorrow, we'll be hosting a sequencing, a whole genome assembly for the African bee, the honey bee at TAGCC. That's the African Genomics Consultancy, the African Genomics Center and Consultancy at Westlands. If you've been selected or you're interested in seeing how this technology can help you as well, please visit our booth or feel free to stop me. I'm very social. I'll not frown. Thank you.
Thank you so much for that presentation. I think again, you can get to your questions answered at their desk. And I think we've been able to get Omar to be able to present. So we'll have Omar go before the others. So Omar, are you there? Great.
Yes, Cecilia.
Great, thank you. Are you the one sharing your presentation?
Yes,
Perfect.
that's mine. Okay,
Okay,
thank you so much, Cecilia. And hi, everyone, once again. So I would like to share, you could say like a personal journey or establishing how that, can you see that now?
Yes, yes, we can see that and we can see you.
All right, thank you. So I would like to talk about advancing genomics research in Somalia. For the past 15 years, I've been living abroad, so just seeking knowledge and in terms of marine science and all other fields related to the maritime and oceans. So what I was always thinking was where I'm gonna go back after I finish my studies and all of that, where I'm gonna go back since Somalia is a post-conflict country. So that's where come the idea of building partnerships back home before I go back and work in Somalia. So I started last 4 years. I've been traveling home for work, starting partnerships and connecting Somali scientific diaspora to the local institutions. So here, as you see, Somalia mainly um, rely on livestock, which supports 65% of Somalis, and also 80% of our exports are also livestock products, and 45% of the GBD as well. In terms of fisheries and marine biodiversity, as you might know, Somalia has the longest coastal line in mainland Africa, which has a lot of potential and also host for the Somali current, very rich in nutrients. So that's why we have a lot of fish in Somali coastal waters. So what I was thinking was always understanding the biodiversity in general will really improve the resilience of our people and also the productivity of our resource. So here I started, communicating with local institutions there, people who work on several institutions, government, and also private institutions. So to talk to them about the opportunities of genomic research in terms of livestock, pre-selection, and also drought tolerant camels, and as well in agriculture. I was also talking to institutions that mainly do marine research. I mean, it's just starting after the Civil War. They're also in their baby steps. So talking to them about the potential of genetic mapping of fish stocks and other works, and also establishing collaborations within the region, African BioGenome Project, and also for training and expertise, and also leveraging, since we have a lot of Somali diaspora abroad, so for funding and also support. Actually, they have supported a lot of my travelers back home, a lot of people who really like science and want a lot of science work to come from Somalia. Here I am with the director of the National Museum, the picture, and we discuss also the natural history and how museum specimens will be very beneficial for research in terms of the genomes. So the research landscape is really growing back home. We had a lot of conferences, the research conferences past few years. Also, a lot of people are trying to pioneer the health action. They started doing like local research on agriculture and health, and also a lot of work on supported by the UN and other agencies on focused on the livestock. But there's always limited studies on marine and other ocean creatures, specifically few genomic studies. So we're really trying to change that. Here also I go and travel along the coastal areas and talk to people, see their perception. So there's a lot of also challenges back home. The infrastructure, for example, the place I am now here in other picture is really remote and very isolated, and you need really to travel almost 2 days to reach there. without paved roads and all of that. So also limited, the labs are limited, the infrastructure in general is very limited. The human resource also is limited back home, but also we have a lot of diaspora who are really successful work doing abroad. So for funding also, Somalia mainly relies on donors. So, and then there's no mainly interest in research specifically, and everyone can understand why, because all the political problems and also conflicts. Since now the country is really getting better, so that's why we're getting the energy to establish some work there. Also, the climate change and all other environmental threats that are affects the country. So here, for the strategies that to advance genomic research. So for capacity building, we were working with locals to train. Basic training on genomic data. So since I work with the Philippines Spire project, so I have a lot of data that I can really show them how this works to the young early career researchers back home. And also for policy, also I've been talking with parliamentarians. So one of the major problems we are facing is Somalia has already signed all the international agreements, but there's no national national ordinance that support these agreements. So we try, for example, to make all these to put them in their agenda, so they can really put that into the national plan. For conservation, also marine biodiversity and all the genetic studies that we are working now. For current work and future directions, we already have some achievements thanks to the team who welcomed me in Berbera Maritime and Fisheries Academy. So I already started talking to the Somali diaspora abroad. A lot of scientists who work in a very esteemed institutions in Europe, Norway, US, UK, many other countries. So just to tell them like, okay, maybe now there's a chance. That there's environment that will welcome you and appreciate the work you might do in the country. So we have collected some samples after the collaboration between Berbera Maritime and Fisheries Academy and also University of Tasmania in Australia. So this is my colleague Shafi collecting the fin clips from Gulliver sharks. So we're trying really to build a lot of partnerships within the region and also internationally. And for the medium term, also, we are planning to establish Somali Genomic Research Center that focuses on the livestock, agriculture, and also marine studies. For the long term, we are planning to integrate genomics into our national policies. As I mentioned, we already talked to the parliamentarians. So to conclude, I think there's possibility that a country like Somalia, post-conflict countries like Somalia can really start from scratch and build up on that. So, and also strategic investments in the research infrastructure and human resource can really be a good step into supporting the resilience of our people, so Somalis. So I would like to acknowledge the team in Berbera Maritime and Fisheries Academy in Somaliland for their support during the stage that we are establishing the work, and all other people who really are willing to help. And thank you, everyone. These are my contacts. You can reach out to me. And thanks.
All right. Thank you so much for that excellent presentation, showcasing how building networks can get you to move forward. And for Mr. Omar, we'll take 2 questions. And thank you for your patience while we were setting that up. Do we have any questions for Omar? Online or here in the audience? All right, so we have your contacts displayed there. I think we can reach out for anyone who has questions or would like to link up with Omar for collaboration for any future work. Thank you so much. And we'll go on to the next presenter. Again, from Genomics Technologies. So we'll welcome Dr. Peter Nthiga from FNS Scientific, who will be speaking to us about the technology solutions that they offer as well. So 15 minutes.
Okay, good afternoon, everyone. Yeah, my name is Peter, as I've been introduced. So I've tweaked the title of my presentation a bit. So I'll be talking about, or rather we'll be exploring how Illumina is developing a genomic ecosystem for plants and animals to address the various challenges that we are encountering, especially in our region, which ultimately will lead to safeguarding our biodiversity and One Health. So we are going to review how genomics is a game changer when it comes to safeguarding biodiversity. We're also going to review the tools that we have from Illumina, and also we're going to look at the data analysis solutions and what are the innovations in store. And then I'll end with a high-level slide on the end-to-end solutions that we have from FNS Scientific. So just to mention, FNS Scientific is a channel partner for Illumina in Kenya. Okay, so why do we need a genomic ecosystems in the first place? So I think this has been mentioned a lot in the previous presentations. So we are dealing with a growing global population, yet we are feeding this growing global population with less water, less land, with unstable climate conditions. So it's a no-brainer. We need solutions. We need smarter solutions to be able to encounter, or rather to mitigate and solve these challenges. And so what has happened over the course of the years, and this is something that typically the audience is aware of. So even before we talked about DNA, we were still doing a lot of genomics through the traditional breeding methods. But what Illumina has done in the last decades is that it has advanced the technologies in genomics through key aspects, that is genotyping and sequencing. So Illumina, we are well known for sequencing platforms, but I don't know how many in the audience know that Illumina started as a genotyping company. So the solutions, or rather the foundation of Illumina as a genotyping company, is enabling it to solve the genomics from an end-to-end point of view, and I'll elaborate more on what I mean by that. So the continuum between genomics and genotyping, or rather between sequencing and genotyping, is essential because with sequencing, essentially what we are doing is we are discovering what are the new variants, what is the new information. But this information alone, sequence data alone, is not enough for us to address the different challenges that we have. That's why we need a genotyping solution. And I don't know how many in the audience are aware that we have an iScan instrument that is used for genotyping activities. So I'll be elaborating more on that in my proceeding slides. So these are the different instrumentations that we have when it comes to agrigenomic solutions or applications. So as I've mentioned, we require the sequencing to do our discovery of the variants and get this novel information that guide us in terms of what we really need to know when it comes to advancing that information and applying it in the field. This is where the Infinium early genotyping comes in. So we have quite a number of applications depending on whichever, or the solutions depending on whichever applications that you're doing, whether it's genome by sequencing or eukaryotic metagenomics. So we've quite a number of sequencing platforms to help democratize the whole sequencing and genomic aspects. So I won't dwell so much on the sequencing aspect for today, but what I rather want to focus on, as I've mentioned, is the impact that we are pushing agrigenomic applications when it comes to microarray genotyping. So what do I mean by microarray genotyping? So Illumina is leveraging on this Infinium technology. So this is bin chip, where we capture the markers in a bin chip, and then we scan these markers using our iScan technology. And what this Illumina microarray solution provides is it provides us with trusted data quality, with exceptional coverage of valuable genomic regions, with consistent reproducibility, flexible customization options. And as I mentioned, right, as of this moment, or during the course of my presentation, actually about 1,000 samples will be genotyped, or rather genotyped on our platforms. Just a testament to how popular this platform is, the Infinium early technology. So we have over 100 commercial and custom arrays, over 10 million plants and animals, for example, was genotyped in 2023. And the reason that makes this technology so popular with our market is the accuracy, the high level of confidence that you get from whichever diverse genomes that you're working with, the scalability, being able to do 100 and over 1,000 number of samples on our chips, the reproducibility of data across different labs, and also the cost effectiveness of our solutions. So these are the different applications that we leverage on the Infinium technology, the genomic selection, the marker system selection, and purity, fingerprinting, parity verification. So in essence, the reason why I'm iterating on the genotyping aspect is for us to be able to solve the challenges that we have, we really need to have impactful information. And this is where genotyping pushes over the sequence data that we get from our instruments. I'll quickly talk about the data analysis solutions that we have. So I'll hone in on the secondary analysis platform, the Dragon. So Dragon, I know many of you might have heard of it. So this is our secondary data analysis solution that we have from Illumina. So this is a collection of different bioinformatic pipelines that helps you perform your secondary line of analysis. What I mean by secondary line of analysis is beyond the sequence, or rather the raw data that you get from your instrument. We've incorporated some of these pipelines onboard our instruments, and and the rest are in the cloud options. So this is very important because it leverages the Dragon server, which I would say is a gold standard when it comes to secondary data analysis at the moment. So it can process data at 177 times more than our local HPC. So it's a very valuable solution and of utility when it comes to data analysis. Just to quickly mention about Dragon, so it handles complex genomes, whether you're working with repetitive elements, or whether you're working with complex animal genomes or plant genomes. This has to do with exceptional sensitivity and specificity that is incorporated in the algorithms of this particular platform. And also something to quickly mention is you can, of course, include your reference genomes to analyze your data. And also there's this significant reduction in cost because of the lossless oral compression that we've incorporated in the system. So regarding the next generation, or rather the next innovations that we have at the moment, so we have incorporated genomic AI into our data analysis solutions, especially the Illumina Connected software portfolio. This is, of course, to enable, give you more comprehensive data, accurate, and also real-time data to address the real-time needs that we are facing. And something else I should mention is the continuous R&D that we are doing to improve on our chemistry. So many of you may be aware that Illumina has been in this space for quite a number of years. But what they've been doing during that space is they've been improving the chemistry, the sequencing by synthesis chemistry, and some of our newer platforms, the sequencing cartridges are coming at ambient temperature, which is really significant, especially for our market, as it significantly reduces the cost and also improves in operationalizing the sequencing work. The other thing I should quickly mention regarding our Infinium chips for genotyping, there are constant improvements to ensure that these chips can be used to genotype a large number of samples, a large number of markers to serve the different needs that we have in the market. So I'll quickly end with this slide, which indicates our solutions from African Scientific. So African Scientific, as I mentioned, is a local company and headquartered here in Kenya, in Westlands, to be specific. So we provide end-to-end solutions. So for our local market, when it comes to doing your genomics work, we endeavor to ensure that we give you the local support from an end-to-end point of view, right from sampling all the way to data analysis. And we've partnered with different companies in this regard to ensure that we give you best value and best support, whether it's during your budget preparation for your proposals. Always feel free to contact us to guide you through the process and to provide you with what you really need to ensure that your work proceeds seamlessly. I'll quickly mention we have some practical sessions starting from tomorrow, following through the same workflow. So we'll be having DNA extraction workshop starting tomorrow using our Knowledge Kit. We also have a demo session as well using one of our fragment analyzers, the QSTEP, to take you through how to do fragment analysis and QC. We'll also be taking through the participants through our solutions and our platforms, that is the Illumina NextSec platform here at MKU, and also our newest platform, the MySec 100. So we're excited to take our participants through these solutions. And then on Thursday, we'll be having a session on Dragon data analysis solutions. So this is something that we've gotten a lot of requests on. So we'll be having a session on it to take through the participants to really understand what this Dragon is all about and what value it provides. Thank you very much for your attention.
Great, thank you so much for that good presentation and for keeping time as well. The FNS also has a desk outside, so if you have questions, you can reach out to them, to us. All right, so we'll have the next speaker from Tegani Fresh. This is Nathan Dongo, and we've heard quite a bit about genomic solutions, changing gears a little bit. Let's hear from Tegani Fresh. Nathan, you're welcome. You'll introduce what Tegani Fresh does.
Thank you. Hello, everyone. My name is Nathan Ndongo. I'm from Tegani Fresh. We're a Kenyan startup, and we're focused on functional nutrition and sustainable agriculture, sustainable food systems. So today I'm going to talk about how genomics and nutrition and agriculture are connected. And I'll start by introducing the One Health concept, which I think a lot of you are familiar with. So it recognizes that human health, animal health, and environmental health are very deeply connected. And if we fix one thing, we fix if we fix one thing, we fix all of them. And I think food is a really is where all three converge. So if we fix our food systems, we can fix our environment. We can fix the problems that we see arising from degrading our biosystems, our ecosystem. So at Agoni, we create personalized meal kits and nutrition plans. So meal kits are basically where you get all your ingredients prepped for you. So it reduces food wastage, and it allows you to create, to make your own meal really quickly. It reduces the amount of dishes you have to do afterwards. I know that's what we hate about cooking. To begin with, you're mostly addressing people who are on a medical diet, so people who have specific nutritional needs, and that's conditions like diabetes, hypertension, ulcers, and our long-term goal is to bridge personalized nutrition with sustainable agriculture. So what I mean by that is we can figure out what a particular population needs in terms of diet, and address that using agriculture, and we make it easy for people to stick to a particular diet. So why does genomics matter? So nutrition models are mostly imported, the imported dietary standards, but they don't necessarily suit our metabolic profiles. Africans, we generally have different metabolic profiles, gut microbiomes, and our genetic predispositions are very different. And this is where genomic data comes in. So insights from genomics can help us figure out what our populations need. Like, so our diets, like our nutrition meal plans are focused on an East African population. And we try and source foods that are available locally, and we source organically. And if we understand what our bodies need, specifically Africans, we can avoid things like lifestyle diseases, which are mostly caused by poor diet, if I might. The next thing is we're able to create the right crops. So for instance, with genomic data, you're able to grow crops that maybe have more bioavailable iron in the leafy greens, or you can have more folate in your greens. And these addresses nutritional deficiencies, for instance. And then lastly, we connect what we grow, because we also do a little bit of hydroponics for we farm our own leafy greens using hydroponics. So this allows us to tailor whatever we are growing to the demands of the population that we serve. So we have a double burden, which is we have under nutrition, but we also have like rising lifestyle diseases, like I mentioned. So we chose hydroponics because we usually see the highest prevalence of lifestyle conditions, mostly clustered around urban areas. And the idea is to help people grow on their balconies, for instance. So hydroponics uses less water in land. It reduces dependence on chemical input, works in urban spaces, and is much, much easier to control what you grow and what it contains. So genomics is where we get our data, and hydroponics allows us to tailor our crops to
provide the nutritional needs for the population, and the nutrition in turn gives us impact. So what does this look like in practice, like at Tagani? So somebody with diabetes, for instance, will sign up to our website. They'll answer some questions that will help us recommend a meal plan or a nutritional plan, and this is tailored to whatever conditions they have. So if they've got ulcers, we know which foods they should avoid. If they have diabetes, we know to avoid foods with high glycemic index. And because we're also trying to promote our local ecosystem, we will look for foods that do well in our region. So some of the next step would be we provide the meal kits if somebody wants that option, and the greens are usually provided by us. They're grown hydroponically. We know exactly what they contain. We know they're not going to cause inflammation. And the next step is to get feedback. So if they reacted maybe to like a particular food has helped them with managing their condition, then we know to do more of that. This is a continuous feedback loop that helps us improve our offering. So we're looking for like the opportunities would be for collaboration would be with researchers who are working with genomic nutritional data, crop scientists who have information about what crops give, what nutrients, health institutions that usually that are the first point of contact for people with chronic conditions, and would be able probably to recommend us as a solution. And then urban farming, people who are interested in doing a little bit of farming for their own consumption, and possibly commercially as well. So we're thinking about something called a distributed food system, where you've got small hydroponic units on many balconies, all working together to produce, to coordinate harvest, planting, all of that stuff. So basically being able to outsource, have a big farm, but in small units. Yeah, lastly, genomic solutions are not just for labs. The research that comes out of this information, I mean, out of the labs, is what we should apply. It can shape how we eat, grow, and care for ourselves, and as well as what we put into our bodies. And I think that if we can address that particular the nutrition problem, we'll be able to address some of the other issues that I've had mentioned around reduced biodiversity and increased disease risk. Thank you.
Thank you for that interesting presentation. And Tegani also is part of our sponsors, and they have a desk out there. So if you have any questions, you can reach out to them at their booth. All right, and then we'll move on to the next presenter. We have Dr. Frank Onyambu from CMB Genomics. Back to genomics. This session is back and forth. So from CMB Genomics, and karibu sana.
Good afternoon, everyone. Thank you very much for the opportunity to be here. It's actually our first time to sponsor an event, so I'm usually on the other side of this conversation, so let's see how that goes. So my name is Frank Onyambu. Do you want to start my presentation, please? So my name is Frank Onyambu. I am a member of faculty in Meru University of Science and Technology, where I teach in the Department of Medical Laboratory Sciences, and actually see some of our students in the audience. I have other affiliations, some of you might know, but for today, I'm strictly speaking for CMB Genomics. And we have a really big goal, and we are not doing this alone. Like, we are hoping to contribute to what is going on in the ecosystem. And I have titled my talk as Building a Biotech Ecosystem for Biomedical Research and Innovation in Africa. So it's a bit of a mouthful, but I'll say a few things about it. So we are not inventing anything. We are plugging into an ecosystem that already exists. We've interacted with a lot of people in this room before, and we think that there is a role that now entrepreneurship and startups could actually play in this conversation. And we've been here for 4 years. We actually started around 2021 at the height of COVID. We've worked with a number of you, and we have ideas. We'll show some of the work that we've done, and we have a number of ideas on how this could be taken forward if you basically take the bait and reach out to us, and we try to see how we can build on this. So if you think of the focus areas of what we do at CMB Genomics, I will broadly list the 4, but I will mostly spend my time talking about talent development and training, which I think is a good entry point for this. So we are in a genomics and bioinformatics conference. I'm sure you've heard since yesterday up to today the importance, the opportunities that are out there, and the potential as well. So I would like to also bring in the angle of also opportunities that could be there in terms of creating jobs and entrepreneurship. So I have a faculty position, as I said. We have some research projects that we do. Some of you have brought research projects to us to work with. So that is also a key part of what we do. And in our own way, we are trying now to plug into the part that we hear about startups and entrepreneurship. But mostly our entry point is basically a talent development and training, and I'll talk a bit about that. So if you were to ask what is the problem, I'm sure a lot of people have talked about a number of problems since yesterday. But the specific problem that for me I want to highlight is limited career pathways for our students. I am a lecturer. We teach a lot of theory most of the time, at least for where I teach. And I think it is not unique for my university. There is limited access to hands-on training that industry is looking for. And for those of you in industry, you will actually attest to this, that when hiring now, a lot of our graduates, even first graduates, you have to spend maybe up to 6 months to actually train them, for them to actually be able to work. I don't mean to say this in a bad way for our universities, but I think it's a reality that a lot of us will acknowledge that there is a gap there. Additionally, we have a gap that is dual in terms of infrastructure. So a lot of our universities don't have the infrastructure to do the kind of practicals you guys are doing from tomorrow. But at the same time, some of those that have them, they are severely underutilized. I won't mention names, but we know where there are NGS platforms that are hardly ever used at all, if they are ever used. So all these actually contributes to part of the problems we now see, if you want to call it the Gen Z phenomenon, or where people are severely underemployed and they are extremely talented and there are opportunities that they could actually address. So I hope I convince you guys that there actually is a problem. Then if you want to think of where the opportunities are, there are so many of them that we could list, but I'll maybe highlight a few, and at the risk of repeating what has already been said. So genomics is now affordable. We've heard from the industry partners in terms of the technologies that are now available. They are now in our labs. A lot of them are now affordable and they can actually be used. There is a lot of interest now in hiring people from Africa or really anywhere in the world, and we know of what they call the demographic dividend. There is an increased population, there are increased number of graduates graduating out of our universities, but this talent is not being tapped as much as it could. So it is our opinion that biotech could actually be one of the ways in which Africa could help leapfrog part of the problems that we have on the continent. And then now I'll tell you guys about the CMB Genomics model. So CMB started as a private lab, as a private company, mostly because it was easier to register a private company than an NGO when we were starting in 2021. But now we have actually ended up adopting a dual structure where we have a for-profit arm that offers services at commercial level, but for sustainability purposes, really. Those of you who've used our services, you will actually attest to this, that we hardly make any profit from that compared to what is on the market. But on the other arm, where actually our passion lies, is the non-profit part of what we actually do. And some of these are basically activities for bringing people together. I'll talk about some of those, like the Journal Club that we run on Fridays. There are some workshops that we've held before, and now increasingly we are signing MOUs with universities to actually go there and do hands-on practical skills. I know at times we understand the importance of DNA extraction and PCR, even before you go to the genomics. But I can actually tell you that a lot of us who actually teach have hardly done those things. I'm sorry to say this, but it's actually the truth. I'm sorry. If I offend anyone, it's for me. It's my experience. But I think there are gaps. There are gaps out there. The way we are doing this through research collaborations. Evelyn Mahugu is here. She is a PhD student in the US at Penn State University. We have a collaboration with our supervisors that have actually enabled her to come spend the summer with us, and there are many others that we could actually list. So I hope I have talked about the model that we have built basically at CMB. There are a number of things that I could say here on what we see as the opportunities. Maybe not all of them. Maybe I'll start with lab infrastructure. Maybe this is the wrong room to talk about lab infrastructure with JB seated here in terms of that we are going to offer infrastructure, but I have already highlighted the problem of idle facilities across the country. As in, think of any institution that you know of that has a sequencer that is not in use. Think of a place with a PCR machine that has never been turned on. So what we want to do is offer a platform as the CMB of bringing people together to actually leverage those facilities. So the goal really here is, it's not, I'll show you the infrastructure we have. It's a tiny lab. But we think that there is potential for leveraging other infrastructure that is available in the country. Then the partner network, we are now actively reaching out to universities and signing MOUs with them to actually have leverage. We are talking to Pwani University. I'll be there later. We have signed an MOU with Catholic University. Their faculty actually has taken a course in our lab. My mother university, Meru University, actually where I have a faculty position, is also signing an MOU with us. So we want to have a partnership of universities, then now use that as a platform to actually build this, what I'm calling the biotech ecosystem. Not as ourselves, but actually with everybody else, and hopefully contribute. But mostly what I want to emphasize on is on talent development. I have seen extremely talented undergraduate students leave university, then actually end up not getting the opportunities that they deserve. So we started in 2021. I threw in a few pictures to show you guys that actually we have a real lab. It's registered as a Class F lab by Kenya Medical Laboratory Technicians and Technologies Board, KMLTTB, that regulates clinical diagnostics in this country. Class F, I believe, is the highest level you could have for that. We actually have that lab. And these are pictures from 2021. With Leon, some of you might know him, and Zakayo at the bottom, and Brian. And we were actually sequencing COVID when the country was sending samples abroad, and at times to institutions like Hillary. We requested to be given samples to sequence 20 reagents unsuccessfully, but actually we sequenced quite a few of those. And that actually demonstrates the potential that our people have. These guys didn't have PhDs at the point at which we were doing this work. These were literally undergraduate students. And I have a more recent picture here showing Sharon, some of you may have met them, and Celestine, who've been here at our booth. So we basically have a lab. Out of all these things that we are saying, we've been around for 4 years. Actually, some publications have come out of our lab. We've collaborated with people from Carlo. I think there are 2 papers on draft genomes that we actually generated in our lab. I'll not go through them, but the point really of this slide is to show you guys that we are also participating in the research side, but now we are trying to bring in what we are learning from the entrepreneurship space into this conversation. And as I said, I think that one of the low-lying fruit that is out there is the talent that is coming out of our universities. And we are now proposing this model for a 6-month fellowship or internship, whatever you call it, which we've actually piloted for a while for probably around 50 people, and it's done really well. So this was done at a really, really small scale where an intern will come to our lab, They will basically take courses online at times with ourselves in bioinformatics, genomics. We will also, they will participate in our commercial activities. That is our way of learning entrepreneurship for this space, and we think it's gone really well. And the reason now we are coming out, and this is the first event we are speaking at, is to look for partners to hopefully scale this. I'm sure there are many iterations that this will take, but I think there is an opportunity for this. Some of the universities we talk to are very interested in us coming up with another certificate or a degree, which I think they're more than enough currently. I think now what people need are hands-on skills that will help them basically to bridge into the market. And some of the ways that I'll propose in the next 3 slides, I don't know I'm doing for time, are first at the workshops that I've talked about, and we are going really, really basic, because for me, I'll speak from experience. We don't want to take anything for granted, so we are proposing at least 3 workshops in around 10 universities, depending on the number of partners that we get, such that we will now go and target students, faculty, but these are actually hands-on workshops. And we are basically inviting those of you who will be interested to partner with us to provide your time, reagents, compute, and among other things, so that we can actually now start to build these, what we are basically, not really start to build, but contribute to the ecosystem that is basically growing. So another thing that we are basically doing is launching, not really launching, but encouraging students to create clubs that are voluntary, where they will be discussing such issues as we are talking of careers in biotech, skills that they need, and so on and so forth. Then this one is, I hesitate to propose it, but we know Kenya is known for fintech. We know people have startups whereby if you wanted to start a startup today for something that you just need a laptop, you could just sit somewhere and do that. However, if today one of my students in Meru University or elsewhere wanted to start a startup in biotech, they can't do that in their room with a laptop. They'll actually need access to facilities and the people who are mentors and such. So I'm throwing this out there. I know there are many participants here. I think that it's time now we start to link with people like NILAB and others who are talking about entrepreneurship. I'm sure they have a really good playbook for building startups in that space, but also now to infuse that back into now those of us who are in the biological sciences, but with a bias for genomics, myself. So I've already alluded to this. Basically, what we are asking for is collaborations with those who are interested to participate in this. You could sponsor a workshop, you could give equipment to a university, and JB seated here, actually, I'm sorry, I hope I don't embarrass you, donated to us a server that we're actually now using in Meru University for bioinformatics through the International Livestock Research Institute. You could contribute kits, and also for our commercial part of what you do, you could bring work to our lab. So that is really what I had to share with you today. I don't do this alone. There is a very active team. Some of you have met them out here, and I have listed basically some of them here. So thank you very much. I think I'm short for time.
Thank you so much for keeping time as well, and for that great initiative. So anyone who'd like to collaborate with CMB Genomics, they also have a desk out there. Do reach out to them. For our next speaker, we are going climate action, and we'll have Mr. Kevin Muriuki from Taka Earth Limited present to us about what we are doing in Kenya in the space of climate action. Karibu.
Hello everyone, how are you doing? Yeah, my name is Kelvin Muriuki. I'm CEO and co-founder of Taka Earth, and we're purely a climate action company. And what we're focused on is converting waste into data and then transforming that data into actual action. So I'm going to try and summarize it as fast as possible. But ideally, so where our conversation starts is usually in informal economies, which is where we operate. So ideally, the global south, and the biggest focus for us is usually the fact that informal economies drive the whole world. So there's a lot of money running around in the economy, but at least 60% of that money is driven by informal economies. And informal economies is basically work that is not formally structured. So it could be gig workers, it could be Joakali artisans, it could be just people who are not formally employed, but they drive 60% of that workforce globally, and this is according to the World Bank. So our focus within that is to then understand how do we, first of all, bring the problem within Africa, so ideally localizing the problem. So for us, within Africa, which is where we're operating, at least 70% of consumer spending. So Africa as a whole, we spend $2 trillion annually in consumer spending. So that's paying for services, buying goods, all that type of work. We spend $2 trillion annually. 70% of that is informal, meaning that there's no receipt, nothing happens. There's no way of documenting that trade. So ideally it goes from a manufacturer, if, for example, it's a product, like let's say you're buying a soda. So it'll leave the manufacturer's table, go to a distributor or wholesaler, and then that's it. There's no other information downwards. But yet you and I are consumers, we're consuming those products, but there's no way of the manufacturer to know what you like, what you don't like. So it's a whole informal structure. Now, on top of that informal structure, you then have waste being generated. So what we've focused on is there's a massive black hole of data, but a massive and a bigger problem within waste for the whole of the global south. And as an example, one of the things we like to use is, do you know anyone who collects waste for your grandmother? Exactly the point, right? So we're building all these big cities, we're trying to expand, but then there's no structure of circularity systems outside of the urban areas, right? So there's no one who's collecting, there's no one who's recycling, but we are sending a lot of waste down there because we send it as products. So what we've done as a company is come up with a solution that allows us to then combine those two problems. So you have a data problem, and then you also have a waste problem. So if you can bring those two together, we think we can provide a solution that allows us to then elevate those informal economies, because that's where a lot of the spending is happening, but then there's no structures for them to be able to grow. And our product as a company is we provide consumers economic incentives, meaning that every time you drop off waste with us as a company, we give you money back, and that's direct money. So for every item currently within Kenya, we're giving 2 shillings per item that you drop off with us. So that's a plastic bottle, glass bottle, or even a can. And then on top of that, what we then do is and this might make more sense in this room more than any other room, is we phenotype the waste. So ideally, we are trying to understand all the different properties of the waste. So what's the waste type? Is it glass? Is it plastic? Is it none of those things, right? So is it non-recyclable waste? Is it organic waste? And then once you understand those properties of the waste, we then ensure that that waste ends up with the correct recycler. So for example, you'll find that within plastic, you have 7 types of plastic. So PET, there's a specific recycler who does PET, and that's purely the bottle, but the bottle top is a different waste type. You have a different recycler for that. So the idea is that if you can build a devolved or decentralized waste capture system, which is what we've done with the machine that you're seeing on the side, which is a smart bin. Ideally, what you do is you drop off the waste as a consumer, we give you money, and then we get that waste to the individual recyclers. But as we do that and as we phenotype the waste, we then generate insights. And those insights allow companies to uptake data from us. And that's how we're making money as a company. So the recipe of this conversation for us is to first be able to show what we're doing from an entrepreneurial perspective, but then also to show what's the long-term effect and how does this room join in that conversation, right? So what we're doing, as I've mentioned, is we have our own AI system that we've built, and you may be familiar with this, where you don't have a lot of data from the global south or within Africa to be able to train on specifically for AI. So we have to build our own data set, which is waste-based data set. And then after building that data set, then we have to deploy the data set to then understand all the waste that we're collecting. So we're doing this in different countries, and we're then, at the end of the day, still automating so that those informal economies still get money directly. So our biggest value set as a company is we do direct economic empowerment, meaning that we are directly affecting all the people who are interacting with us from an economic perspective, but at the same time being able to generate sustainability insights. We've been able to build this over a period of time. So we have active users, we have waste that we've diverted from ending up in landfills, and then the focus of that is also the companies that we've been able to onboard. Companies like Coca-Cola, Pernod Ricard, Rainforest Alliance, and Melanin Capital. To be able to scale this as a business is you have to consistently move away from thinking about waste and start thinking about the data. And it's maybe something that would be important for this room as well, right? Because you're doing a lot of data as you sequence genomes. So can you actually create and scale profitable business out of that, right? And then how do you still ensure that you're retaining a lot of data privacy within that structure? So our main structure of revenue earning is 3 verticals. So we lease out our smart bins. We have a sustainability subscription as a service model where companies uptake data from us. And then we also have a waste management platform that we provide to waste management companies. But the longer term focus of what we're doing is being able to deploy this for the global south, because that's where waste management systems do not exist. But the biggest goal for us is to be able to have direct impact within the global south. And this means that we need to reach those people, directly impact them from an economic perspective, and at the same time, be able to transform that waste into valuable resources. And that's where this room has a lot of effort to offer in terms of we want to be able to research the waste that we cannot manage. And that's from a molecular perspective. So what we are doing is purely data-ended from a non-scientist perspective, but also being able to collaborate with scientists will then allow us to achieve a lot more in transforming waste into valuable resources. Thank you.
All right, thank you for that presentation. So this is a One Health themed conference workshop, and we have a lot of information from different spaces. So that's climate action. And if you know what to do with some of that waste, reach out to Taka Earth. You might make, did he say $2 trillion? All right. So the last speaker of this session before our lunch is Ms. Fatma Mohammed from AgriGuard. And I think she'll also talk a bit about AI and how AgriGuard is applying AI in Africa, in Kenya, to transform our agricultural knowledge and information. So you're welcome, Fatma.
Hello, everyone. Hi. I know it's lunchtime. Anyway, we've been talking about genome editing and genetically engineered crops throughout yesterday's presentations from people like Professor Runo and Dr. Florence. But today we are going to talk about how we are going to use AI to be able to bring about public understanding in terms of these technologies. So let me start with a case scenario where, say you've attended a rally and a very prominent politician there tells you food made from GM crops causes hormonal imbalances, whereby you find that maybe he tells you the male can be able to grow female organs and things like that. Now imagine if millions of farmers who have attended that rally make decisions just based on this kind of information. This is very sad, and it's happening in our society currently right now. Anyway, my name is Fatma Mohammed Omar. I'm a subject matter specialist for AgriGuard, and I'm also a youth advocate for biotechnology, and I'm here with my colleague Ronald Okech. He's an AI innovator and a software developer. We are here to introduce AgriGuard, which is Africa's AI shield against agricultural misinformation. The billion-dollar problem here stems from misinformation. It's not only annoying, it's actually very expensive. It's costing our African farmers about $1 billion every year because of loss of yields and poor investments that are brought about by misinformation in GMOs. This is actually contributing to food insecurity in Africa and making Kenya one of the worst hit countries globally because of misinformation in terms of GMOs and agribiotech. And in Kenya, what is happening right now, GMO is being suppressed by about 40%. So you can imagine the kind of crisis we are undergoing right now. So my team and I, we asked ourselves, what can AI do for us? And we were able to come up with it's a 3-in-1 ecosystem, AgriGuard. It's a 3-in-1 ecosystem that works in real time. It comprises of Viral Farm. Viral Farm is an AI scanner for social media in real time. It detects trending myths in real time. The other one, we have AgriFactCheck. It is an instant SMS/WhatsApp chat box that debunks these false claims using verified facts. And lastly, we have Mythbuster AG, which is an AI-generated debunking videos that educates farmers before these myths go viral. I have a problem with this, I guess. Sorry. Yes. So our technology deep dive is using a lot of NLP models. Viral Farm uses BAT and Whisper. AgriFactCheck uses Trilio, GPT, and N8N. And the Mythbuster AG uses Sunos, GPT, and N8. All these are national language processing models that are being used to be able to develop our solution. Our solution is aligning with various UN SDGs, SDG 2 on zero hunger, 9 on industry, innovation, and infrastructure, and 13 on climate change. So we are basically trying to leverage AI to solve food insecurity, to use it to support digital agriculture, and also to support climate resilient crops and practices. Our product, we are trying to integrate 3 streams in terms of our business models. We'll offer free premium to farmers where they'll be able to receive free WhatsApp SMS messages using verified facts. The other one is business to business. We want to partner with businesses like Syngenta, where they'll be able to pay to access our myth detection APIs and analytics. The other one is business to government. We are offering licenses to government institutions like the Kenya's National Biosafety Authority and NGOs. We've actually had some partnership with NGOs like ISA AfriCenter and AATF. Our mission is basically to become the Google search for agricultural truth to protect 100 million farmers from misinformation by 2030. And our mission is to empower our African farmers and agri-stakeholders with verified real-time information on biotechnology, GMO, and agri-tech. So we are basically using AI and local language integration to defeat misinformation at a scale. And before I leave, I would like to give some notable achievements we've been able to have. We won a specific hackathon under the ISA AfriCenter, where we were among the top 3 finalists, and this gave us an opportunity to be able to attend a very grand summit called the FINA Summit 2025. We actually rubbed shoulders with Honorable Musalia Mudavadi there, and at least some policymakers are seeing what the youths are doing out here. And something else, we were also able to present in a conference called ACAT in Kigali, and also this one here, Africa BioGenome. And again, we are presenting again later this month in the ABBC symposium in Lusaka. I believe Dr. Florence mentioned ABBC yesterday and was talking about how ABBC is being used to fight misinformation around agribiotech and GMOs. So that's basically what we've been doing. And in short, I would like to say we are saying no to misinformation. We are saying yes to AgriGuard. We are trying to help our farmers to farm facts and not to farm fears. So to finish up, I would like to introduce my colleague who will give a very short demo of what AgriGuard is all about. Thank you.
Good afternoon, everyone. My name is Ronald Dodoyo Kech. I am an artificial intelligence innovator and entrepreneur. So we were able to come up with this solution, which is called AgriGuard, which is a mythbuster software that combat agricultural misinformation and provide farmers with science-based facts about modern farming and technologies. So AgriGuard has, as she said, it has 4 features. The first one being viral farm and agri-fact check. The first one is viral farm. Second is agri-fact check. Third is mythbuster AG and integrated ecosystem, where it consists of all the 3 other features. So beginning with agri-fact check. So when you want to start fact checking, you just come to agri-fact check and type your question or misinformation or any doubt you have. For example, do GMOs cause cancer? And within minutes, you'll get, not even minutes, within seconds, you'll get your answer. For example, let's see how many seconds it'll take. 1, 2, and 2 seconds, you have your answer. So numerous studies have found no credible evidence on linking GMOs to cause cancer. This was from National Academy of Science, which was published in 2016. So AgriFact Check also has, we have integrated multiple languages, around 30 languages across the world, so it's easier for anyone to use it. For example, let's try use Kiswahili. You can see it's auto-detect. You can just type any language you want. So, je, viua wadudu vya mimea husababisha madhara kwa binadamu. So again, within seconds, you get your answer. So this is the answer. Viua wadudu vya mimea vinaweza kuwa na madhara kwa binadamu ikiwa vinatumika bila kufuata maagizo. This was from source of World Health Organization. We also have frequently asked questions. This is another feature which is called Viral Farm. It has real-time analytics. It scans misinformation spreading online and social media. So for example, this was from 24 hours ago, GMO cause cancer. You can see the severity is very high. Another one, organic food is pesticide free. You can see the severity is medium, and it's from which source also. So we also have a chart here. You can see which social media does misinformation spreading. We also have mitigation tips that can help you navigate our viral farm. How does viral farm works? So it works by continuously monitoring social media and messaging apps 24/7. It also works by AI analysis. Advanced algorithms classify content as potential misinformation. We also have early response. You get immediate response on emerging misinformation before it takes its route. So another feature was mythbuster AG, which is AI will automatically generate educational video. You can choose your topic, and then, or you can enter your own topic. You can choose the video length you want, and then you can also choose the avatar of your choice, and then generate the video. Another feature I said is it's called, it's an integrated ecosystem where it consists of all the 3 other features. This is still under development. We also have our news and resources where you can learn about other agricultural facts and that are spreading. For example, how you can use what is called, you can use drones to map your land. You can use drones to scan crops, and it's AI-powered drones. So we are actually trying to partner with some of drone providers in Kenya to install those AI. So it's still in the early stages of development, and it's we can say it's MVP. So this is the solution we were able to come up with, and we are planning also to integrate voice flow that can help people who are not learned, like Mzee with Atushago there, who doesn't know, who is illiterate. We will integrate voice flow, so with just a press of a button, you can ask any information. So finishing up, we also have a company that teaches, that offers AI consultants to businesses. For example, we have heard here that how people can implement, are trying to implement artificial intelligence in doing genome sequencing and genome assembly. We can also tailor our AI services to that. We also offer AI education to anybody who wants to learn AI, even if they don't have any tech background. So you can visit our website called Atron Solutions. Thank you.
All right, thank you for that. Agrigad, sorry. Agrigad, thank you for that great presentation and teamwork. And I believe you're around, so any questions to Agrigad, you can field them in the course of the day. So I think, go ahead.
We have a few questions online, and thank God you can manage that space for us.
Mm-hmm.
I think I'd seen some people had raised their hands. Dr. Thangod?
Yeah, just carry on with the people on site. I'm just trying to curate the questions now.
Okay. So from this session that we've had, do we have questions that we can take before we go for lunch? Any questions?
My name is Shali from JQuat. My question goes to Fatma and Ronald. What is the biggest challenge that you faced scaling up your app?
The biggest challenge was language barrier, but right now we have integrated NLP that offers that language barrier, and also funding. So we are looking for funding to be able to scale this to other countries in Africa.
Right, we have another 2 questions.
Thank you for the wonderful presentation. My question is, have you considered in future maybe using USSD code? Because this system, you've created it for farmers, and you find most of the farmers, especially in rural areas, do not have the smartphones and such issues.
Thank you very much. We're also planning to include USSD. I actually forgot to mention that. Sorry.
Yeah. Okay, I think we're ready with the online questions.
Go ahead.
Yeah, so we're going to take about 4 questions. So 2 people have raised their hand, and also with about 2 questions. There are quite a lot on the chat, but I'm just going to read them out afterwards. So Faria, could you please unmute and ask your question? Faria Ndungu, could you please unmute? Okay, otherwise we'll go to Fekado.
Hello.
All right, go ahead, please.
Can you hear me?
I can hear you.
Yes, we can hear you.
I wanted to ask, how does the natural language processing softwares they've used in their tech factor in that most Kenyans use Swahili and Sheng language, and knowing AI and machine learning, it requires a lot of data that hasn't been captured, factoring like African languages, you know? So how have they circumvented that? Thank you.
Thank you very much. We also use API integration from GPTs like ChatGPT, Google AI Studio that have actually integrated African languages, including even Sheng.
The second person is Temesgen Mangu Olango. Could you please ask your question on mute? All right, carry on.
Thank you, Vianney, sir. So my question goes to all the presenters who used the AI models. So one is about the explainability of the models. So because I think both in the detection of this waste recycling, the models were how explainable were the models? And related to our last presenter, you are using ChatGPT and other large language models. And what about the GDPR and the data ownership and all those things? So do we have some regulations in Africa in general and in Kenya specifically about those things? Thank you very much.
If I got your question right, if it's about data protection, we're using Kenyan data regulatory systems to protect people's data. Could you kindly just pardon your question, if you don't mind?
Hello, can I continue?
Yeah, continue, please.
Yes.
Thank you very much. And my question also goes to the last presenter, and I really appreciate the initiative. It is very interesting. And what I want to understand is that this website platform shows that the source of the information, right? So is there any way to check whether the information is reliable or not?
Yes. As you had seen when we were presenting, for example, AgriFactCheck, we are getting our data. We have integrated with a lot of agricultural institutes like Calro and World Health Organization. So we are getting APIs from them. So when you search there, you get information that are actually proved, and it will tell you where the information you have gotten it. For example, as you had seen, the information that we asked in Swahili was from World Health Organization.
Sorry, just to add on it, our verified facts come from a lot of institutions, from AATF, ISA AfriCenter. These are institutions that basically are trying to demystify information that is surrounding agribiotech. We are also looking at others like the WHO that Ronald has mentioned, and also we are also trying to integrate peer-reviewed journals. So basically, I can say our information is actually verified and it's trustworthy.
Okay, thank you. I think for me, it's more, I have some questions before I go to the chat box, Cecilia. So my question is mainly to the mainly the industry folks, those doing this genomic service and technology providers, So one thing I'm thinking about is how do we kind of inspire people to sequence genomes locally, more end-to-end, 100%, because this is one of the things we do in AfricaBP that we aim to do. So as you heard previously in the presentations, where, for instance, the ONT and the Hi-Fi were done locally for the first time. I think I don't know if that aligns with Jim Baker. I think Jim Baker did mention that they've done it, but I wasn't sure if that was sequenced locally in Africa or it was sequenced outside Africa. So, but to CMB Genomics and others, how do we do this? Because we have some of these opportunities and some of these technologies locally everywhere, but how do we kind of inspire those action and ensure that we use the facilities and the opportunities and the infrastructures available? So perhaps it could be a panel between all of the industry folks that spoke earlier on, including CMG Genomics and the rest.
All right, so CMB Genomics will go first.
Okay, sure. Thank you, thank God for that question. I think those are really good intentions, but also at the same time, I think we need to look at the realities. My lab has an ISEC 100. it's now becoming obsolete, extremely expensive to run anything on that. You go to the next lab, it has a MySec. But then if people send samples to Macrogen or elsewhere, it's being run on a NovaSec. So the straight answer really is to consolidate a majority of the work being done locally to go to a central service. Is I think the short and straight answer to this, in my opinion anyway. Thank you.
Yeah, I do agree with you. Maybe can we also give other, so that we kind of give the other industry the opportunities?
All right. So I'll take the question on behalf of FNS Scientific. And I completely agree with CMB Genomics. I think a lot of the time we are acquiring this expensive infrastructure and thinking that we're able to operate these instruments at capacity within our own small silos and kingdoms, let me call it that, because you find that the capacity that MySec and NextSec needs to utilize, if you run 5 samples on that, you're really underutilizing the system. The cost per test becomes very prohibitive. So it makes sense to work together, to consolidate our efforts as Africa. The same way the Africa BioGenome Project has come together to, bring together all these initiatives. As local partners, can we work together to make sure that we're investing in infrastructure that is going to be utilized fully? And the reason why the external companies are able to bring down their costs to that level, in my opinion, is that we are actually providing them the samples to reach their capacity. They can project their samples from Kenya, Uganda, Ethiopia, wherever, and they can say within September we'll get 3,000 samples from Africa. So this means they can utilize their systems to full capacity. While we sit here with our instruments and we've only been able to do 2 field works, and we are not able to run at full capacity. So how do we come together as a scientific community to make sure that we're utilizing the infrastructure that exists to capacity, and then we're building a case to even expand beyond what we have? So that's, I'd say that.
So Change Biotech is here, and I've just asked them the question, what's the cost and the benefit of doing end-to-end sequencing locally, and is there a benefit in investing in this?
So thank you for the question. In terms of cost, I would say when it comes to driving capex down, I don't think any other sequencing technology offers a more flexible solution like Oxford Nanopore. Our capex is really low, our OPEX is very low. So when you're setting up a lab, in terms of cost implications, it won't be that much. And like I said, it's scalable. So depending on your budget, you can choose to go from low throughput sequencers all the way to high throughput sequencers. The other question was benefits of investing in this technology. Like I said, you've seen, I shared a slide of improvements through time, like from 2014 to 2025. And you've noticed most of these improvements are in our kit chemistries and in our flow cells. Meaning anyone who bought a technology even back in 2015 still gets the same updates. We won't ask you to buy a new machine just because we've changed something. And I was to share something, it's just that time ran out, but Oxford Nanopore is actually working on protein sequencing, meaning you'll be able to do RNA sequencing, DNA sequencing, and protein sequencing on the same platform. It's still in development, but it's coming.
Yeah, so that now fits into my, okay, do we have another company? Because I think this session is very important to kind of lay that foundation, because one thing I see, especially within the Africa ecosystem, is, maximizing the use of infrastructure. As you mentioned, for instance, we've had these technologies, but, not many, you don't often get to see so many genomes published by African institutions sequenced in Africa. As I mentioned previously with the presenters, they did the Oxford Nanopore and Hi-C end-to-end on African soil. To the best of my knowledge, I haven't read any paper on that before. And the other one, PacBio and OmniSeq. So those are the things I really want us, I would really like to see in terms of, how do we do that? And I really also like the response by CMB Genomics and also others. Pulling resources is very important. If I look at the genomes that have been sequenced by these teams, it has always been pulling that resources from one institution, from one network to the other, and bringing it together. So, Cecilia, do we have any other company that would respond on this?
Yes, we do have two more comments
Okay.
from JB and from Inkaba.
Okay, go
Yes,
ahead.
sorry, I'm not running a company, but I have a point to make here. I agree with Change Biotech. I'm not as commercially aggressive as them, but I agree that ONT is a great platform offering flexibility at very low capex costs, and that's very important to understand. My point is that also we have to lobby our politicians to have exemptions for custom tariffs on everything that pertains to scientific research. All the consumables you import for lab work, that's very important that at the EAC level, we have a reduced or zero tariff on all these. It exists for books, so because EAC was aware that for educational purposes, printed matters, books for school children are extremely important, and education is key to the development of a country. So now, if our same political leaders can understand that scientific research is key to advancing the country, then we have a big win there.
Yeah, so sorry, Jim, just to do a follow-up on that, I think that's a very important point. So who in ESC, taking ESC as an example, who do you think could take this up? Will it be, for instance, there has to be some people pushing pressure, or, I mean, which organization do you really think could be in a better position to really drum up that support or actually make that happen?
Well, I don't know. You have political leaders, including presidents, saying that they really see the value of scientific research, and some of them are pushing also the AU to put some of their own money into the development of research. So we have to talk about these, to talk to these same political leaders. I remember President Kagame at some point made some strong statements saying that African countries should not rely on the Gates and so on receiving money from outside to do the development of African countries. And I think it's time to put them to act in agreement with their talks.
All right, we have Inkaba Biotech.
Yes, so regarding the costs of sequencing on the PacBio system, the end-to-end sequencing, so with the PacBio systems, we have the market, the capital for buying a system is very high. The systems are expensive, but on the brighter side is the cost of the sequencing goes down. It's very cheap. So if we can have institutions just agree to put money on the systems, like in East Africa, we currently don't have a system. And the good thing with the PacBio systems, the data is amazing. The quality of the data is amazing. So if institutions can just come up with a way to get money that they can get a system, then the cost of sequencing would be cheaper, and hence we would sequence more genomes in Africa and East Africa probably. Thank you.
Yeah. So I think just to also wrap that up is that we, in AfricaBP, we provide all technology, genome sequencing and service providers the opportunities to work with us. So I must say that I have first-hand experience of kind of working with majority of the technology providers within, the ONT, like as you heard from the previous presentation, the MGI-Tech, the Inkaba Biotech, Revio, and also the Illumina system. So with regards to both, what the previous presenter just talked about, again, I'm not marketing any company here. So one example is, for instance, Senebongo did speak previously about the private-public partnership that they have with Inkaba Biotech to have the Revio system that they procure, and as the previous presenter mentioned, it's not cheap, but that was at the institutional level. That was what they did. It's huge amount of investment. I've also seen institutions that did the same thing on Oxford Nanopore as well within the Africa BioGenome Project. I've also seen institutions that are doing the same thing with, for instance, the MGI-Tech technology. So that is one way. Again, this goes down to the point of the previous point by Change Biotech that we need to pool resources, and our institutions need to connect with other organizations through public-private partnerships, because that is one way that I think this has actually helped several partners within the project. Over to you,
Great.
Cecilia.
Yeah, thank you so much. I think the fact that all these partners are here is that they're willing to work with the Africa BioGenome Project, willing to build these reference genomes and to get genomics progressing forward. So I'm sure we'll have a conversation where we find a way to make it work, because we have to make it work for Africa by Africans. So thank you so much, thank God, for that interesting wrap up to this session. So the online participants,
Sorry,
you'll allow us.
we have just one question from Tobias. He was raising his hand yesterday, and I think so that he can get the chance to speak. Just Tobias, keep your question very, very short. Could you please unmute?
Yes, please.
Please unmute. 10 seconds.
Okay.
Yeah.
Thank you, Dr. Tangod. I am happy to see you here. Mine was, I was actually raising hand when you posed that question concerning how to, put resources together in terms of equipment. Although by the time I'm now to speak, you have already said something that really wrapped up my intentions, because you have already talked about the African Bio Project group, which I was actually saying should be the hub that should organize these things in terms of assemblage of equipment to this for central use by most African countries, depositing their thing. Instead of sending them to the Western world, we now have a central point in Africa where all these demands for this and that test will be put together, and thereby reaching the high potential of samples that is needed to bring down the cost. Then in terms of political will, which you asked, who should make that pressure? I think the African BP too, which you, being the heart throb of African BP, I know you have the capacity, galvanizing all of us, all the African players, to reach out to the governments. I mean, African BP has become a household name, which gets respect from even AU. So you champion the cause under the banner of African Bio Project. It will be a very, very strong voice to cause our governments to act in the direction we want it. Thank you very much.
Thank you, Tobias. That's a big one. We have to think about it. We have the chair on site. So, and a big job for you.
Thank
so,
you, Tobias.
And just to mention that, Tobias, in response to infrastructure, we have 2 papers published already from the Africa BP that sets out our roadmap, both in terms of sequencing infrastructure and bioinformatics infrastructure. So all of those things are in the pipeline. And I agree with you. For us in Africa BP, we don't send samples outside Africa. We have not sent any sample. If it takes us 12 months to get the analysis done, sequence, we will wait until that time. until that 12 months. Thank you so much.
Thank you, dear.
All right. Thank you. So we'll take a lunch break for 40 minutes. So online participants, in case you've already had your lunch, you can prepare for the next session where we'll be having some talks about what we'll be doing in the practicals. So thank you so much. I'll allow the team here to take a break and see you after lunch. So 40 minutes, strictly 40 minutes break. Thank you.
Hereby, we have our gel documentation room. This is where we do all our gels. As you can see, we have one of our researchers already trying to load the gels and also run them in the machine called the e-box. This is from a company called Wilbur. They have very quality gel documentation systems.
The core service we provide is distributorship and sales services. We sell our products across the extended Eastern Africa region. All the countries in the East African region, ranging from the DRC, we have Rwanda, we have Uganda, where we have a branch office, we have Tanzania, we have Burundi, Kenya, Ethiopia, Somalia. Our sales are basically for the molecular biology field and the general lab. In the molecular biology field, we provide products and solutions for all the processes, the entire workflow, from sample collection, DNA, RNA extraction, PCR, amplification, both reagents and instruments, and sequencing and bioinformatics. In the general lab, we offer equipment. We supply equipment, including but not limited to freezers, to fridges, to biosafety cabinets, and many more. So you can always come to us for any reagent, consumable, or equipment need that you have for your lab. We also have a line of diagnostics, molecular diagnostics, both for plants, animals, and humans.
So we like to welcome all of you to come utilize our services here in 74 Riverside Drive. Our lab is open to students. Our lab is open to researchers who want to do their work here. We provide space, we provide the equipment, and we also provide the reagents and chemicals that you may need for your research work. I welcome you all. Thank you.
Study infectious diseases. Analyze genomic variations in tumors. Project outbreaks through surveillance monitoring. Combat drug resistance. Discover mutations for disease monitoring. Identify analyze study disease drivers. Develop simple. With breakthrough simplicity, our fastest runtimes yet, everything in one footprint with onboard Dragon analysis and significant sustainability advancements. This is the highest standard in benchtop sequencing, empowering every lab everywhere on a quest for discoveries that can't wait. This is the MiSeq i100 series. This is sequencing made simple.
Welcome to Inkaba Biotech East Africa Limited. The company was started in the year 2010 on the 23rd of June. Over the years, we've grown into a fully fledged your one-stop molecular biology shop. And currently, we're also running one of the best labs in the region. Join me as I take you through the tour of our laboratory. Welcome. Here we have our engineering department, whereby we service our equipments here, equipments which that we can carry from the client site to our offices. Basically, these are small equipments. For the large equipments, we prefer servicing them at the client's place. Here we also do calibration services. For all of you, those who have pipettes that need calibration, we now proceed to our extraction room. This is where we do all our extractions. Once we've received samples, this is where it all starts. Our scientists and qualified personnel will look at your samples, record them the way they are supposed to, and take all the details and also report on what they've received. From there on, they will proceed. This is our PCR room. As you can see, we have scientists who are also very busy doing their work here. We have timelines whereby we try as much as we can to give our clients results in the shortest time possible. As you can see, our lab is fully equipped and work is progressing, as you can see. So we are looking forward to having our clients approve our services and also happy at the same time as we continue to provide solutions, molecular solutions, and also solving research problems in the region. Here we have our post-PCR room. This is where we do our sequencing services. Clients, after they've sent their samples, gone through the extraction, gone through the PCR, this is now where we do our sequencing. As you can see, our machine is fully running throughout because we have a lot of samples and we have a lot of samples coming in each and every moment. Here we have our data analysis room. As you can see, one of our researchers is also seated analyzing data, which has already been gotten from the machine. Hereby, we have our gel documentation room. This is where we do all our gels. As you can see, we have one of our researchers already trying to load the gels and also run them in the machine called the e-box. This is from a company called Vilba. They have very quality gel documentation.
Basically, the core service we provide is distributorship and sales services. We sell our products across the extended Eastern Africa region. All the countries in the East African region, ranging from the DRC, we have Rwanda, we have Uganda, where we have a branch office, we have Tanzania, we have Burundi, Kenya, Ethiopia, Somalia. Our sales are basically for the molecular biology field and the general lab. In the molecular biology field, we provide products and solutions for all the processes, the entire workflow from sample collection, DNA, RNA extraction, PCR, purification, both reagents and instruments, and sequencing and bioinformatics. In the general lab, we offer equipment. We supply equipment, including but not limited to freezers, to fridges, to biosafety cabinets, and many more. So you can always come to us for any reagent, consumable, or equipment need that you have for your lab. We also have a line of diagnostics, molecular diagnostics, both for plants, animals, and humans.
So we like to welcome all of you to come utilize our services here in 74 Riverside Drive. Our lab is open to students. Our lab is open to researchers who want to do their work here. We provide space, we provide the equipment, and we also provide the reagents and chemicals that you may need for your research work. I welcome you all. Thank you.
Study infectious diseases. Analyze genomic variations in tumors. Project outbreaks through surveillance monitoring. Combat drug resistance. Discover mutations for disease monitoring. Identify analyze RNA disease drivers. Develop legislation. Simple. With breakthrough simplicity, our fastest runtimes yet, everything in one footprint with onboard drag and analysis, and significant sustainability advancements. This is the highest standard in benchtop sequencing, empowering every lab everywhere on a quest for discoveries that can't wait. This is the MySeq i100 series. This is sequencing made simple.
Welcome to Inkaba Biotech East Africa Limited. The company was started in the year 2010 on the 23rd of June. Over the years, we've grown into a fully fledged your one-stop molecular biology shop. And currently, we're also running one of the best labs in the region. Join me as I take you through the tour of our laboratory. Welcome. Here we have our engineering department, whereby we service our equipments here, equipments which that we can carry from the client site to our offices. Basically, these are small equipment. For the large equipments, we prefer servicing them at the client's place. Here we also do calibration services. For all of you, those who have pipettes that need calibration, we now proceed to our extraction room. This is where we do all our extractions. Once we've received samples, this is where it all starts. Our scientists and qualified personnel will look at your samples, record them the way they are supposed to, and take all the details and also report on what they've received. From there on, they will proceed. This is our PCR room. As you can see, we have scientists who are also very busy doing their work here. We have timelines whereby we try as much as we can to give our clients results in the shortest time possible. As you can see, our lab is fully equipped and work is progressing, as you can see. So we are looking forward to having our clients approve our services and also happy at the same time as we continue to provide solutions, molecular solutions, and also solving research problems in the region. Here we have our post-PCR room. This is where we do our sequencing services. Clients, after they've sent their samples, gone through the extraction, gone through the PCR, this is now where we do our sequencing. As you can see, our machine is fully running throughout because we have a lot of samples and we have a lot of samples coming in each and every moment. Here we have our data analysis room. As you can see, one of our researchers is also seated analyzing data, which has already been gotten from the machine. Hereby, we have our gel documentation room. This is where we do all our gels. As you can see, we have one of our researchers already trying to load the gels and also run them in the machine called the e-box. This is from a company called Wilbur. They have very quality gel documentation systems.
The core service we provide is distributorship and sales services. We sell our products across the extended Eastern Africa region. All the countries in the East African region, ranging from the DRC, we have Rwanda, we have Uganda, where we have a branch office, we have Tanzania, we have Burundi, Kenya, Ethiopia, Somalia. Our sales are basically for the molecular biology field and the general lab. In the molecular biology field, we provide products and solutions for all the processes, the entire workflow from sample collection, DNA, RNA extraction, PCR, amplification, both reagents and instruments, and sequencing and bioinformatics. In the general lab, we offer equipment. We supply equipment, including but not limited to freezers, to fridges, to biosafety cabinets, and many more. So you can always come to us for any reagent, consumable, or equipment need that you have for your lab. We also have a line of diagnostics, molecular diagnostics, both for plants, animals, and humans.
So we like to welcome all of you to come utilize our services here in 74 Riverside Drive. Our lab is open to students. Our lab is open to researchers who want to do their work here. We provide space, we provide the equipment, and we also provide the reagents and chemicals that you may need for your research work. I welcome you all. Thank you.
study infectious diseases, analyze genomic variations in tumors, project outbreaks through surveillance monitoring, combat drug resistance, discover mutations for disease monitoring, identify analyze RNA disease drivers, develop simple. With breakthrough simplicity, our fastest runtimes yet, everything in one footprint with onboard drag and analysis, and significant sustainability advancements. This is the highest standard in benchtop sequencing, empowering every lab everywhere on a quest for discoveries that can't wait. This is the MiSeq i100 series. This is sequencing made simple.
Welcome to Inkaba Biotech East Africa Limited. The company was started in the year 2010 on the 23rd of June. Over the years, we've grown into a fully fledged your one-stop molecular biology shop. And currently, we're also running one of the best labs in the region. Join me as I take you through the tour of our laboratory. Welcome. Here we have our engineering department, whereby we service our equipments here, equipments which that we can carry from the client site to our offices. Basically, these are small equipment. For the large equipments, we prefer servicing them at the client's place. Here we also do calibration services. For all of you, those who have pipettes that need calibration, we now proceed to our extraction room. This is where we do all our extractions. Once we've received samples, this is where it all starts. Our scientists and qualified personnel will look at your samples, record them the way they are supposed to, and take all the details and also report on what they've received. From there on, they will proceed. This is our PCR room. As you can see, we have scientists who are also very busy doing their work here. We have timelines whereby we try as much as we can to give our clients results in the shortest time possible. As you can see, our lab is fully equipped and work is progressing, as you can see. So we are looking forward to having our clients approve our services and also happy at the same time as we continue to provide solutions, molecular solutions, and also solving research problems in the region. Here we have our post-PCR room. This is where we do our sequencing services. Clients, after they've sent their samples, gone through the extraction, gone through the PCR, this is now where we do our sequencing. As you can see, our machine is fully running throughout because we have a lot of samples and we have a lot of samples coming in each and every moment. Here we have our data analysis room. As you can see, one of our researchers is also seated analyzing data, which has already been gotten from the machine. Hereby, we have our gel documentation room. This is where we do all our gels. As you can see, we have one of our researchers already trying to load the gels and also run them in the machine called the e-box. This is from a company called Wilbur. They have very quality gel documentation systems.
The core service we provide is distributorship and sales services. We sell our products across the extended Eastern Africa region. All the countries in the East African region, ranging from the DRC, we have Rwanda, we have Uganda, where we have a branch office, we have Tanzania, we have Burundi, Kenya, Ethiopia, Somalia. Our sales are basically for the molecular biology field and the general lab. In the molecular biology field, we provide products and solutions for all the processes, the entire workflow from sample collection, DNA, RNA extraction, PCR, purification, both reagents and instruments. and sequencing and bioinformatics. In the general lab, we offer equipment. We supply equipment including but not limited to freezers, to fridges, to biosafety cabinets, and many more. So you can always come to us for any reagent, consumable, or equipment need that you have for your lab. We also have a line of diagnostics, molecular diagnostics, both for plants, animals, and humans.
So we like to welcome all of you to come utilize our services here in 74 Riverside Drive. Our lab is open to students. Our lab is open to researchers who want to do their work here. We provide space, we provide the equipment, and we also provide the reagents and chemicals that you may need for your research work. I welcome you all. Thank you.
Study infectious diseases. Analyze genomic variations in tumors. Project outbreaks through surveillance monitoring. Combat drug resistance. Discover mutations for disease monitoring. Identify analyze study disease drivers. Develop simple. With breakthrough simplicity, our fastest runtimes yet, everything in one footprint with onboard dragon analysis and significant sustainability advancements. This is the highest standard in benchtop sequencing, empowering every lab everywhere on a quest for discoveries that can't wait. This is the MySeq i100 series. This is sequencing made simple.
Welcome to Inkaba Biotech East Africa Limited. The company was started in the year 2010 on the 23rd of June. Over the years, we've grown into a fully fledged your one-stop molecular biology shop. And currently, we're also running one of the best labs in the region. Join me as I take you through the tour of our laboratory. Welcome. Here we have our engineering department, whereby we service our equipments here, equipments which that we can carry from the client site to our offices. Basically, these are small equipment. For the large equipments, we prefer servicing them at the client's place. Here we also do calibration services. For all of you, those who have pipettes that need calibration, we now proceed to our extraction room. This is where we do all our extractions. Once we've received samples, this is where it all starts. Our scientists and qualified personnel will look at your samples, record them the way they are supposed to, and take all the details and also report on what they've received. From there on, they will proceed. This is our PCR room. As you can see, we have scientists who are also very busy doing their work here. We have timelines whereby we try as much as we can to give our clients results in the shortest time possible. As you can see, our lab is fully equipped and work is progressing, as you can see. So we are looking forward to having our clients approve our services and also happy at the same time as we continue to provide solutions, molecular solutions, and also solving research problems in the region. Here we have our post-PCR room. This is where we do our sequencing services. Clients, after they've sent their samples, gone through the extraction, gone through the PCR, this is now where we do our sequencing. As you can see, our machine is fully running throughout because we have a lot of samples and we have a lot of samples coming in each and every moment. Here we have our data analysis room. As you can see, one of our researchers is also seated analyzing data, which has already been gotten from the machine. Hereby, we have our gel documentation room. This is where we do all our gels. As you can see, we have one of our researchers already trying to load the gels and also run them in the machine called the e-box. This is from a company called Wilbur. They have very quality gel documentation systems.
The core service we provide is distributorship and sales services. We sell our products across the extended Eastern Africa region. All the countries in the East African region, ranging from the DRC, we have Rwanda, we have Uganda, where we have a branch office, we have Tanzania, we have Burundi, Kenya, Ethiopia, Somalia. Our sales are basically for the molecular biology field and the general lab. In the molecular biology field, we provide products and solutions for all the processes, the entire workflow from sample collection, DNA, RNA extraction, PCR, purification, both reagents and instruments, and sequencing and bioinformatics. In the general lab, we offer equipment. We supply equipment, including but not limited to freezers, to fridges, to biosafety cabinets, and many more. So you can always come to us for any reagent, consumable, or equipment need that you have for your lab. We also have a line of diagnostics, molecular diagnostics, both for plants, animals, and humans.
So we like to welcome all of you to come utilize our services here in 74 Riverside Drive. Our lab is open to students. Our lab is open to researchers who want to do their work here. We provide space, we provide the equipment, and we also provide the reagents and chemicals that you may need for your research work. I welcome you all. Thank you.
Study infectious diseases. Analyze genomic variations in tumors. Project outbreaks through surveillance monitoring. Combat drug resistance. Discover mutations for disease. Rapidly identify analyze RNA disease drivers. Simple. With breakthrough simplicity, our fastest runtimes yet, everything in one footprint with onboard drag and analysis, and significant sustainability advancements. This is the highest standard in benchtop sequencing, empowering every lab everywhere on a quest for discoveries that can't wait. This is the MySeq i100 series. This is sequencing made simple.
Welcome to Inkaba Biotech East Africa Limited. The company was started in the year 2010 on the 23rd of June. Over the years, we've grown into a fully fledged one-stop molecular biology shop. And currently, we're also running one of the best labs in the region. Join me as I take you through the tour of our laboratory. Welcome. Here we have our engineering department, whereby we service our equipments here, equipments which that we can carry from the client site to our offices. Basically, these are small equipment. For the large equipments, we prefer servicing them at the client's place. Here we also do calibration services. For all of you, those who have pipettes that need calibration, we now proceed to our extraction room. This is where we do all our extractions. Once we've received samples, this is where it all starts. Our scientists and qualified personnel will look at your samples, record them the way they are supposed to, and take all the details and also report on what they've received. From there on, they will proceed. This is our PCR room. As you can see, we have scientists who are also very busy doing their work here. We have timelines whereby we try as much as we can to give our clients results in the shortest time possible. As you can see, our lab is fully equipped and work is progressing, as you can see. So we are looking forward to having our clients approve our services and also happy at the same time as we continue to provide solutions, molecular solutions, and also solving research problems in the region. Here we have our post-PCR room. This is where we do our sequencing services. Clients, after they've sent their samples, gone through the extraction, gone through the PCR, this is now where we do our sequencing. As you can see, our machine is fully running throughout because we have a lot of samples and we have a lot of samples coming in each and every moment. Here we have our data analysis room. As you can see, one of our researchers is also seated analyzing data, which has already been gotten from the machine. Hereby, we have our gel documentation room. This is where we do all our gels. As you can see, we have one of our researchers already trying to load the gels and also run them in the machine called the e-box. This is from a company called Wilbur. They have very quality gel documentation systems.
The core service we provide is distributorship and sales services. We sell our products across the extended Eastern Africa region. All the countries in the East African region, ranging from the DRC, we have Rwanda, we have Uganda, where we have a branch office, we have Tanzania, we have Burundi, Kenya, Ethiopia, Somalia. Our cells are basically for the molecular biology field and the general lab. In the molecular biology field, we provide products and solutions for all the processes, the entire workflow from sample collection, DNA, RNA extraction, PCR, purification, both reagents and instruments, and sequencing and bioinformatics. In the general lab, we offer equipment. We supply equipment, including but not limited to freezers, to fridges, to biosafety cabinets, and many more. So you can always come to us for any reagent, consumable, or equipment need that you have for your lab. We also have a line of diagnostics, molecular diagnostics, both for plants, animals, and humans.
So we like to welcome all of you to come utilize our services here in 74 Riverside Drive. Our lab is open to students. Our lab is open to researchers who want to do their work here. We provide space, we provide the equipment, and we also provide the reagents and chemicals that you may need for your research work. I welcome you all. Thank you.
Study infectious diseases. Analyze genomic variations in tumors. Project outbreaks through surveillance monitoring. Combat drug resistance. Discover mutations for disease monitoring. Identify analyze, study disease drivers. Develop simple. With breakthrough simplicity, our fast runtimes yet, everything in one footprint with onboard drag and analysis, and significant sustainability advancements. This is the highest standard in benchtop sequencing, empowering every lab everywhere on a quest for discoveries that can't wait. This is the MySeq I100 series. This is sequencing made simple. Study infectious diseases. Analyze genomic variations in tumors. Project outbreaks through surveillance monitoring. Combat drug resistance. Discover mutations for disease monitoring.
So good afternoon and welcome back. To our online panelists, I hope you are all back. So this afternoon sessions, we're going to continue in the same spirit that we had last before lunch. And right now, this is an exercise where we will have the people who are hosts to the practical sessions give the introduction to what they were doing. So you realize that the African BioGenome Project model for hosting regional workshops is a 5-day workshop. The first 2 are conference style, so that's what we had today and yesterday. And then the next 3 days, we will have the genomic companies and sponsors running practical sessions. So how do we run these practical sessions? During the nomination, during the registration process, during that time, the applicants get to fill in whether they're interested in doing a practical or not. And then you choose one practical session that you would like to attend. And those names are taken to the technology companies who shortlist people depending on the capacity that they have. Obviously, the bioinformatics online classes, most of them can take a larger number than the physical in-person wet lab. So sometimes wet lab can only take 10 people or 15 people, and we've had some labs take even 20, and that was a stretch. But the bioinformatics platform, and especially the online ones, that some of them are being run, the numbers are as many as your Zoom subscription. So to set off this particular session, we will have a presentation from Dr. Helen Igusi, who in partnership with Professor Kasoun Tesfaye and Dr. Zedu Edea from the BETIN, the Biotechnologies, Ethiopian Biodiversity Institute, will be able to run a practical on RNA-seq analysis. And Dr. Helen, thank you very much for partnering with Africa BioGenome Project for now. This is your 3rd year, and in all those 3 years, you have been able to run a practical session. And we really appreciate that, because that's how our students are able to get hands-on experience on some of the things that have been talked about during the last 2 days. So Dr. Helen, you can share your presentation, and the mic is yours. As we connect with Dr. Helen, I can invite Professor Rasmus Heller, who is a professor of population genetics from University of Copenhagen, all the way in Denmark. He has been a partner of African BioGenome Project now for 2 years, and for 2 successive years, He has run practical sessions. Last year, with his partner at Pwani University, they ran one practical. This year, with Pwani University, they are doing 2 practicals. Next year, who knows? The trend is you do 3 practicals. So, but welcome, Prof.
Thank you very much, Professor Ann. Yes, as you say, this year we have managed to contribute to 2 different workshops. We are not participating actively, unfortunately, in the one in Kilifi in Pwani, but there is one, and Professor Sami will be joining the meeting later to introduce that one. So what I will do is I'll introduce the other one, which is a new format that we are, let's say, experimenting with this year. And I will not share slides with you. Instead, I'll just share my screen with the course website and just quickly go through what is happening in that course. Let's see if that works. So you can see it here. I hope the online people can also see it. Yes, good. So this course is a little bit out of structure in the sense that, as Professor Ann just said, most of the practical workshops are 3-day workshops that come in direct continuation of the 2 days of conference. But what we have done with this course is that it's a whole week course. So it has been going on actually since Monday. So we hope that the participants that were interested in following both have been able to juggle that somehow through the first 2 days. The reason why we have this format is that actually this summer course in high throughput data for population genetics is something that has been running for several years in Copenhagen. It's been running, I think, now for 8-9 years, something like that, in Copenhagen. It's a very, very good course. It's been hosting some of the best population geneticists in the world, I can say, ever since it started. Some of the founding fathers, even of population genetics, have been visiting and teaching in the course in Copenhagen. Now it's more the younger generation of really strong population geneticists from everywhere in the world that are teaching. So what we have done this year is we've been able to offer it also as an online option for the participants in the AfricaBP regional workshop. So we have a number of students, I think about 25 ABP attendants who are following the online course this year. And I'll just quickly tell you what it's about, what is going to happen. So it's taking place the whole week in Copenhagen at the Department of Biology. And I'll just very quickly, I'll introduce the teachers, or just quickly show you the teachers. These are the instructors that are teaching this year. They are all from Copenhagen, but not all from the Department of Biology. They're from also Globe Institute, a world-renowned genetics and genomic analysis unit. So all of these young and very able people are conducting this course and teaching, instructing in it. I'll just quickly show what the content is, what the different things that people are learning in this course. So the first day is a general introduction to, I'll need to zoom, yes. Does that work? Yes, it works. So the first day is an introduction where the very basics of next generation sequencing data is being presented for those who are maybe not too familiar about that. I should also say in the general structure of the course is that it's a mixture of lectures, theoretical lectures, and a lot of practicals. So there's a lot of emphasis on hands-on activities. And at the end of every day, I can also say regarding the structure, there is a research talk by someone working within the field of population genetics. You can see, for example, Monday it was understanding genetic architecture of disease in Greenland using population genetics. Today it's going to be a genomic history of the Rapa Nui. So these are very, very, I should say, very competent people coming and sharing their newest research at the end of every day. That's how it goes. First day, introduction about the NGS data, the basics of it. Next day is how you actually then extract stuff that you need for population genetic analysis from the data. So estimating allele frequencies, SNP calling, genotype calling, and such things. Then there's something about imputation, which I don't know how many of you are familiar with that, but if you work within population genetics, you will know what that is. On Wednesday, that's tomorrow, the theme is population structure and admixture. There'll be lots of different things you can learn there. Also things like D statistics and inferring ancient gene flow, which is something that is very, I should say, popular to do nowadays. And the research talk is about ancient pathogen evolution and epidemiology at regional and continental scales. So this is actually using ancient DNA to learn something about pathogens in the past, something they do a lot at the Globe Institute. On Thursday, the theme is local ancestry inference and selection inference. I'm sure all of you have some kind of biological background, so of course you will know what selection is about. Local ancestry inference is about figuring out how what the ancestor of each little small bit of your genome actually is. So where each of the bit of an individual's chromosome comes from. And finally, on Friday, there is a theme about principal component analysis and demographic history, where people will learn something about using extant genetic data to hindcast population history back in time. And there'll be an interesting research talk from an associate professor, Christine Bormann, who has developed a very cool method where you can actually, and I'm not kidding you, you can actually get DNA out of air itself and sequence it and do actual analysis on it. So yeah, that is very briefly the course. We hope to be able to offer it continually every year in connection with the regional workshop, and I'm sure we'll figure out a way to make the dates even more compatible, so that it's actually possible to first do the whole conference here and then do the whole workshop also in connection with that. I also encourage you, if you get the chance, I know it's hard, but if you get the chance, there is also a physical component of this. It's a blended learning method course. So if you get the chance to go physically to Denmark, to Copenhagen, very welcome to apply for this course and go and take it in person. Of course, that's even better than following virtually. Yes, that's all I have to say about the course. I think I also spent my time. I don't know, do we take questions or? Yeah, if anyone has a question, can answer. Yes.
So are you exploring LCWGS, low coverage whole genome sequencing? I mean, in the past we've used markers, and markers spread randomly all across the genome, that's GBS. Mm-hmm. But now more and more people study how much information can we get from data possibly generated with long reads, but at just 1x or 2x coverage. Yes. The fact that you have long reads is interesting, and then you have to do lots of imputation, but there are statistical methods being designed at the moment. Yes. to deal with sparse whole genome data.
Yes, that is, yes. I'm glad you asked that question actually, because some of the instructors on this course, some of the course organizers are some of the people who developed some of the methods exactly for analyzing low-depth genome data. So I don't know if you're familiar with the Angst sort of software universe.
Yeah, that's the package. That
yes, so these are the authors of Angst that are teaching this course. So I would say this is very much something that we work on. There's not that much about it in this incarnation of the course. There has been in some previous versions, but simply just for practical reasons that, you know, there's a limit to how much you can actually fit into to 1 week of teaching. If you want to go all the way from explaining what the data is to that comes out of the sequencer to having, let's say, finished population genetic analysis. But it's something that we do all the time. And as I said, it has been in previous versions. It might well come in there again. Yeah.
Thank you. Do you have another question? Do you have another question? Don't think so.
I'll stop sharing.
Question online? I have a question. Maud Farhat. Maybe you can ask your question. Yes.
Can you read it to me, Paul?
No,
Yeah,
actually.
Maud, shall I give you the I will give you
Can you
this.
hear me, please?
Oh, yeah, speak now.
Can you hear me now?
Yes, please go ahead.
Yes, we can hear you.
Sir, please, I'm a Moroccan PhD student, and I want to ask you if we can still complete the course and get the diploma, please.
It's the registration is now finished, so you cannot join the course now this year.
I already registered, but I had an email problem. I couldn't receive emails. I just joined the course today in the morning.
Okay.
I talked to Dr. Sabrina about it.
Yes.
And
yes.
he asked me to ask you if I can still complete it and get the diploma, please.
Yes, definitely yes. We'll sort that out.
Thank you so much, sir. Thank you.
Sure.
Okay. Thank you. You can move on to the next one.
Yes.
So thank you very much, Prof. And AfricaBP will take a commission for mediating a registration issue for your course. But to the young students, don't feel intimidated that you should only attend these courses online. I think with a little initiative and long-term planning, you can be able to apply for travel grants. These are available. You can also apply for, the travel grant gives you air ticket and accommodation, right, in some places. And if you have good notice, you can be able to look for funders. So don't let that stop you from making those applications. And thank you for inviting them to actually visit Copenhagen and yes, please, the mic.
Yes, I just want to supplement that by saying that we actually decided to have to reserve a number of slots for specifically for African students to follow the course. So don't be, as Anne says, don't be scared if you are not entirely sure if your qualifications are exactly those asked for. Okay, we have a bit of echo going on here. But what I just want to say is really
Helen, could you please mute? Helen, mute.
Is this mic okay?
Okay, yes,
testing.
Yeah.
Yes, but I think the message may have passed, but.
Sorry, we had some technical issues, but what I was trying to say was that really apply next year when the application form for the ABP comes along, either physically or online. Don't feel scared that your qualifications or your background is not the required one, because we want to reserve a number of spots for African students. Because this is the whole idea of ABP, and we want to promote that with this course activity also.
So thank you very much to University of Copenhagen and Professor Rasmus, who has flown all the way to be here present, not only to affirm the commitment, but also to foster that collaboration that we have started. So it's through AfricaBP that this program that was perhaps unknown or not known, not very well known to African students is now readily available for you. So please go ahead and apply. So next, we're going to have Dr. Helen Igusi, who is a professor from University of Addis Ababa, who is running a practical at the Ethiopian Biodiversity Institute in Ethiopia, together with Professor Tesfaye and Dr. Idia. And Dr. Helen is another collaborator who has been with us since the beginning and was one of the first ones to host a wet lab practical. And we're grateful for your collaboration, and you have the mic now. Please go ahead.
Okay, thank you so much, Professor Anne. And I would like to congratulate you for the successful launch of this workshop. And we are also very happy to be part of African BioGenome Project and to host this practical session together with Bio and Emerging Technology Institute. Unfortunately, this workshop, the practical session, to be hosted at Addis Ababa University due to relocation of the office and HPC facility at Bio and Emerging Technology. We're going to host this practical session for the next 3 days from Wednesday to Friday. Our topic will be RNA-seq analysis at Addis Ababa University. This is why my presentation, I will talk some about introduction, where we select this RNA-seq analysis for this practical session, some overview about the practical session during 3 days, and the practical session organizers and trainers. While we select this RNA-seq analysis, this is the third East and Central Africa Open Institute workshop. We had it NGS data specifically for DNA sequence analysis for the last 2 years. So in this year, we select RNA sequence analysis because it is very important and it helps to study the whole transcriptome, which is a complete set of RNA transcripts in cells or population of the corresponding particular time or developmental stage from the beginning at the end, and it might be under different physiological conditions, so which really important analysis compared to the previous conventional RNA gene expression analysis, because it also gives us a comprehensive transcriptome analysis that is beyond gene expression. It will help us to discover novel features. It accurately quantifies the gene expression level. It also helps to understand cellular processes and diseases, especially if there is a change in gene expression, to model different disease modeling and the cellular heterogeneity. So that is why we select this topic for this year practical session. And so we're going to explore this pipeline during the 3 days of practical session. From the beginning, the understanding of the sequence read, we're going to see fast accuracy. And we also using Galaxy Cloud Computing Facility, the Galaxy project for our practical session, from the very beginning up to visualization and gene expression analysis and visualization. In the first day tomorrow, we'll start some theoretical aspect in the morning and make the participant familiar about exploring of this, the Galaxy server, which is independent, any program, no need of programming language, R or Unix. It is open source web-based platform for intensive biomedical and related research. And the second day, we're going to show them some parts of quality analysis and mapping with reference genome. And they will give them most of hands-on practical session and some theoretical background. And the third day, they will going to quantify the gene expression level and visualization. And at the end of the day, we'll have a course evaluation session. So I hope the participant will enjoy their stay during the practical session. When we say the past practical session together with bio and emerging technology, the first practical session was in 2023 at Betin. The theme was DNA sequence analysis and mapping from reference genome. In the second workshop in 2024 last year, we also have NGS data analysis from mapping up to visualization, and we did a very successful workshop at that time using the existing HPC facility at Betin. And this year, the workshop will be at College of Natural and Computational Sciences at Addis Ababa University in the Ratkilo campus. We are really excited to host this workshop in the collaboration with Betin. This will be the Ethiopian team, Professor Kasaun and me is a national representative node of African BP, and Dr. Zod also the organizer and head of the bioinformatics and genomics directorate at Betin. The trainer also, two of them, Dr. Johannes and Mr. Nuguse also from Betin. They are the bioinformatician, will give the course together with me. We really acknowledge African BioGenome Project to give us this opportunity to be the partner, and Addis Ababa University, especially College of Natural and Computational Science and Department of Microbial Science and Genetics to host this practical session in this campus, and partnership, the financial support of Bio and Emerging Technology Institute and all the team and the trainer. So I want to thank you. Asante, amesagnalo. I hope we will have a very exciting moment, and I would like to thank you, all the organizer of the General African BP workshop. Thank you so much.
Dr. Helen, I hope you can hear the round of applause here. And
thank you so much.
We appreciate the partnership. I think we're the ones who are very grateful. We are also grateful for hosting the AfricaBP regional node in Ethiopia. And we can actually see it's an ignition point where we're getting all the partners who are in this area of bioinformatics and genomics coming together. We are especially grateful to the investment that Betin is doing. And we're honored and humbled that your DG is fully supportive, that the Director General of Betin is fully behind it, and we can see you have assembled a big team of technical people to support, and we're very, very grateful. Please pass our regards, and we wish you well, but we know that you will run a very successful 3-day event, and we are running behind you. Thank you very, very, very much for that.
Thank you.
So another round of applause to this great team.
Thank you so much. Asante sana.
Asante, Dr. Helen. So we started off in Kenya, and then we flew on to Denmark. As we were going on, we peeped down on Ethiopia. We saw what's happening there. Now we need to cross back and come back to East Africa. And we have Professor Dr. Antoine Mafuila from the University of Kinshasa in the Democratic Republic of Congo. And he's running a practical on the NGS data analysis with Galaxy platform and Linux. University of Kinshasa is the ignition point for Africa BioGenome Project in the DRC. And we are grateful to the university for having hosted us for the last 3 years. And Dr. Aintoin there is a bioinformatician. He'll tell us more about the practical that he's going to host and give us an insight on what he has done in the past years. You're welcome to present in English or French, whichever language is convenient, and we assure you that we will follow you. Thank you.
Thank you, Professor. If you can allow me to share my screen.
Yeah, please go ahead. You can share, please. You have co-host rights, Professor Antoine.
Just share your screen.
Yeah, I see that is locked. It's not activated.
You
If you
have
can
co-host,
activate.
right? You have co-host, right? Okay, hang on. All right, you can do that now. Go ahead.
Okay, thank you. That's great. I can do that now. Okay, thank you, Professor, again. So, for the third time, the University of Kinshasa and the Laboratory of Molecular Biology has been solicited by Africa BioGenome Project to conduct an hand-on practical session, and we propose the NGS data analysis with Galaxy, and also we will use some Linux command. So the purpose of the session will be to enhance participant and skills and the understanding of the essential concepts like genome sequencing, what is FASTQ file, what is the content, describe the content, and to understand the FASTA file, genome assembly pipeline, FASTQ quality check, timing, what is config, what is scaffold, and also to assess the quality of genome assembly. So, through a real life scenario in Galaxy platform and with Linux also, using data from bioinformatics repository, attendee will actively learn how to get data from repositories or from sequence machine, use application for analysis, and gain practical experience that reinforce theoretical lesson that they got. So we aim to foster solid foundation for the future research and outcoming research in genomic. So the training, the session is set for the next 2 days from 9 a.m. to 4 p.m. Central African time, which include a 1-hour lunch break. And the molecular biology classroom will serve for the training, and attendees who register for online session also will attend by online. And the training will be in French, totally in French, because most of our participants that was registered will speak mainly French. So participants need to bring a laptop connected to internet. So we will use Galaxy server, and they can also download Linux operating system into the machine. And this ensures everyone is equipped for an effective learning. So I will be the instructor of this training. I will be responsible for launching demonstration, leading discussion, guiding hand-on activities, and offering necessary assistance during the whole session. And each attendee is required to engage fully in session and share difficulty we face, give constructive feedback to foster a collaborative learning environment. It is critical for all participants to ensure their own safety and that of the other during the session. And this is a platform of the View Galaxy platform is designed to empower users with a wide array of tools. This practical session will introduce its core functionality, enabling effective data handling for diverse bioinformatics analysis. And our participants are 11 that have been registered, but I know that some other participants will join us, and we will be able to accept them if with some other places. And they come from DRC Congo mostly, and one come from Republic of Congo will attend online, one for Central African Republic and another for Benin. Most of them are PhD students, other are postdoctoral researchers, and we have also one or two researchers. They are working on the field of molecular biology, virology, HIV, microbiology, in National Public Health Laboratory, university, including Faculty of Medicine and Agriculture School. And they are also coming from Wildlife Conservation Society. So that was the global overview of the training that we will provide tomorrow and until Friday. Thank you, Professor.
So let's give Professor Mafwila a round of applause. So what is very unique about Professor Mafwila is that he's a medical doctor who went into public health, and from public health now he's doing his doctorate and is using next-gen and sequencing bioinformatics tools basically to answer questions, and this will have direct impact on his patients and of course on the work that he does. And we thank you so much for partnering with us. Dr. Mafwila, are all your participants coming physically or is it an online course?
We, among the 11 participants, 3 will attend online and the remaining will attend physically.
Oh, congratulations for that. So if you're, and it will be in French? The instruction will be in French, I imagine?
Yes.
So, yes.
Nous allons avoir toute la formation en français. Donc, ça permet, c'est ça la plus-value que Africa BioGenome Project apporte en Afrique. Ça veut dire qu'il permet aux pays d'Afrique francophone d'avoir accès à des formations en bioinformatique. So I can translate. This is the add value Africa BioGenome Project bring to the African continent because it allows the French speaker country to have access to bioinformatic training that is difficult this day to get trained in bioinformatics when you are in a francophone country. Thank you.
And also, Professor Mugai, just mentioned that Professor Antonio is also with the Africa CDC. I don't know if he highlighted that, and it also, I think Professor Antonio, you with the Africa CDC in Congo, you kind of look after the central office, the bioinformatics. You are the head of the bioinformatics, right? Or yeah.
So
exactly.
Congratulations.
Yeah. Thank you.
Yes, but congratulations, and we can see again the One Health application, bioinformatics in humans and in biodiversity and in livestock genetics and the whole group of it. So we wish you well in the next 3 days, and thank you so much for partnering with Africa BioGenome Project. Next year, I'm sure you'll have some students from Kenya joining you there physically for the workshop, and we look forward to a long partnership. Thank you. Let's give him a round of applause and let's wish him well. So now we are inviting Dr. Samuel Wambua from Pwani University, who is running a practical there, DNA Diversity, a hands-on introduction to African wildlife genomics. Dr. Sami was here yesterday at a panel session. You remember him. He's working on the turtles, doing a lot of science in the marine parks. And last year, there was a collaborative workshop that was run with the University of Copenhagen. This year, Pwani is the one, Pwani in Kilifi. Kilifi is how many kilometers from Nairobi? It's at the coast of the Kenyan coast. It's about 600, 570, something like that. And Dr. Sami, you're welcome to tell us a lot more about the practical that you're hosting for us. Thank you.
Thank you. Could someone confirm if my slides are visible?
Yes, please, we can see it.
Okay, awesome.
Yeah, I'm glad to be here again and to be offering this workshop for the second year running. So this year we are basically just offering a practical session on genomics as applied to African wildlife, a workshop that we have dubbed DNA to Diversity. I thought I would start with a bit of motivation as to why this is necessary or where the whole idea may have come from. Of course, with the ongoing biodiversity crisis and the loss of biodiversity, the rich African biodiversity still remains unexplored, and much of the advanced technologies of genomics haven't been really used in a locally relevant way to address some of the challenges that we face with biodiversity loss. And therefore, the workshops, not just the one we're offering now, but also the others that we offer, were born out of the need to raise awareness for upcoming scientists to start using genomics in exploring and hopefully informing protection approaches to species of conservation concern using genomics, and encouraging multidisciplinary networking and creating a critical mass of upcoming scientists that can use genomics to address a number of questions within the conservation space. So this workshop that starts tomorrow is coming actually after we have finished another one, which is a longer version of the same that runs for 2 weeks, and now we're offering it for the third year running as an annual event at Pan University. Last year, as Prof has mentioned, we offered this 2 weeks training in collaboration with Copenhagen. The year before, we offered it with help from University of Glasgow. This year, we've run it for 2 weeks up to Friday, this last Friday, in partnership with Indian Institute of Science, but also with generous help from Change Biotech and Oxford Nanopore. But for the particular workshops that now will be running from tomorrow till Friday, the goal, like I have hinted earlier on, is to raise awareness on the utility of genomics in conservation, hopefully to inspire upcoming scientists to start using genomics as a tool to address conservation problems, and build a critical mass of professionals that are thinking the same way and thinking that there are answers within genomics. And therefore, collaborations and networking becomes very also important to us. We shall be using real data sets from some of my research, but also data sets from my co-trainers' research as well. So it's going to be real data, and we'll see how that goes. As to what shall be covered, the first day, which is tomorrow, will cover the basics of genetics with a bias to wildlife. And then we'll look at why would often be using next generation sequencing data rather than other data forms. You'll be introduced onto working on terminal, specifically Linux, but also some R, and understanding the different file types that you will be required to understand to be able to work sufficiently or competently in the bioinformatics space. Day 2, which is Thursday, we'll start diving now into real analysis of population genomics. So we'll try to estimate relatedness and population structure, measuring different matrices of diversity, including heterozygosity. And we'll try to use all these to see if we can make any inferences about population's viability. Day 3 will go into metagenomics in all its shades and hues, but I mean, I think it has been hinted on how metagenomics would be. Well, One Health is an important component, I think, of the ongoing conference, and I think metagenomics as a tool becomes very indispensable when it comes to being able to get into the field of One Health. So we will run through some amplicon metagenomics data or metabarcoding, and introduce you to some shotgun also metagenomics, and speak about the various applications that there may be. Of course, what I haven't covered there is the fact that we think networking is an important part of this, and therefore we try to always end this with what we formally call a social networking event, which really is just a party in the evening on Friday. And I hope, if not for the population genomics or metagenomics, there will be inspiration there for someone to want to attend. So I will stop here, but I want to thank the organization whose logos are on here for helping one way or another with the trainings. Asanteni.
A round of applause to Pwani University and Dr. Sami Wambua. We're really grateful for this partnership. And I may not be so excited about the metagenomics, but the party is a real motivation for people to actually come for the course. And so it's important for you who are all listening to look out for our advert for next year. And this is the sort of flavor in the terms of the practical session. So when you're selecting your choices, you'll only have the ability to choose one. So you'll be able to at least choose one that is really interesting to you. So thank you very much, Dr. Sami and your team. And we're sending over Paul overnight to join you tomorrow. And at least you'll be able to brief us on what has happened. So thank you. So next we are going to move to Frederick Kebaso. Frederick Kebaso is the first African BioGenome Fellow from Kenya. He spent some time in a lab outside Kenya. He will tell us more about it. And he'll also tell us how Africa BioGenome Project has transformed him to his... Oh, sorry. So the next one is Joel. After Joel, we will have Frederick Kebaso. So Joel Ogwang is running a practical session in Uganda at the National Animal Genetic Resource Center and Data Bank in Uganda. They've been a partner with us for the last 3 years. And Joel Ogwang is running a practical known as Tidying and Analysis of Biodiversity and Ecological Data in R. Joel, please go ahead.
Oh, thank you so much, Professor Anne, for that introduction. So my presentation is going to be two-tier. The first is I'll give you a brief introduction to my PhD proposal, which I'm pursuing at the University of Wyoming in the US. And then I'll give a brief introduction to the practical session that I will be hosting. So my proposal will be covering climate and human-mediated fate of lattice perches of Lake Tanganyika, and I will be inferring this using genomics. Lattice perches, the best example is the Nile perch. So you know how important it is, and the picture is right there. So the main question I'm trying to answer is, how are humans and climate change influencing the evolution of pelagic fishes of Lake Tanganyika? So for introduction, Lake Tanganyika is located in East Africa and it is shared by Tanzania, Burundi, DRC, and Zambia. And by size, Lake Tanganyika is roughly the length of Uganda, for your information. And this lake has a wide diversity of species, ranging from snails and the famous cichlids, and then the economically important fish species, which are the pelagic species. We talk of the darger and then the lattice, which are the perches. So the problem here is that there is a decline in the abundance of these economically important fish species, and this is attributed to climate change over many centuries. As you can see, a correlation between fish abundance and an increase in temperature. So don't let these, the graph may confuse a little bit. Sorry, I did something wrong. Yes, the graph of temperature is inverted. So it's an increase in temperature correlating with a decrease in fish abundance. And this, on the right is the same story. The next thing is overfishing, which is fishing pressure on the lattice species and other economically important fish species in East Africa, which is affecting, the availability of cheap protein, fish protein, for the population. And as a result of climate change and overfishing, 3 of the lattice species in Lake Tanganyika are currently endangered. So the question is, how has the community structure in these fish species changed in the recent past? I will be trying to infer this using fossil data. I'll be collecting tooth and scales of fish from the bottom of Lake Tanganyika. I will carbon date it, identify 2 species, and try to assemble the community assemblages over the years. So I expect to find distinct community assemblages and that this will correspond with climate change. And in addition, how has their effective population changed in the recent past? For information purpose, effective population size is an indicator of long-term survival of a species. So I'll be using the coalescent method based on the site frequency spectrum to determine how this has changed. And briefly, we can start with looking at the shape of the spectrum to see whether the population is constant, declining, or it is growing. So when the population is declining, you'll have an excess of rare alleles, as you can see in the middle graph, as compared to when the population is growing or it's constant. And we can use the Markovian chain technique to see the trend of the effective population size, as you can see here, or we can use the stairways plot. And some of the expected results will be that there will be a constant population, effective population size, or it could be declining. And then we can look at patterns between species, whereby if the pattern is the same as in like the orange and the blue patterns of the simulation, we know that the environmental effects are the same. However, when there are different patterns and the species are still in the same environment, we can attribute this to the different genetic backgrounds of the different species, something which is referred to as epistasis. The next question I will explore is what factors have shaped this recent evolution? And I will be using the landscape genomics approach to identify and profile varying environmental variables and then test the association with the genetic variation. So I'll be interested in identifying which genotypes are covering with the environment and whether there are any indication of signatures of adaptation, and eventually whether we can attribute this to climate change and fishing pressure. So in conclusion, this research will provide a refined population structure and evolutionary trends in response to climate change and fishing pressure, and will also profile signatures of adaptation in economically important fish species in Lake Tanganyika. This will contribute to a better understanding by both researchers and policymakers of population dynamics in response to climate change and the increasing human footprint in Lake Tanganyika. Eventually, this will provide evidence-based policies for the sustainable management of Lake Tanganyika fisheries, the African Great Lake ecosystems in general, and in other freshwater ecosystems. And we hope that ultimately this would result in increased fisheries production and increased incomes to the fishers. So this is what I'll be researching on. And I want to acknowledge my supervisor and the lab mates and my funders. At this point, you can find more about what I'm doing using this QR. And I will also welcome your questions, but I think after I introduce my practical session. So for the practical session, which would be hosted at NAGRIC, which is the National Animal Genetic Resource Center and Data Bank in Entebbe. This year, the workshop is going to be virtual, and it will focus on data analysis of ecological data in R. So why R? We know that R is very good for statistical analysis, and it has a comprehensive depository of packages. You talk of the CRAN or the bioconductor, and it is an open source language, so it's free, and it is very effective for collaboration and facilitating reproducibility of research. And it can handle large data sets, and it has extensive community support. And crucially, it is very exceptional in data visualization. In addition to that, it is really, it has a very friendly graphical user interface where you are able to see the code that you're writing, you are able to see the objects or the data that you already have in R. You can see the errors. You can also see the plots at the same time. So for this year's workshop, I'll be taking participants through a simple workflow, which includes importing your data to R, cleaning it, transforming it in different visualizing, and doing all sorts of things around it before you can effectively communicate it. So to read your data, you'll use various tools, including table delimited data. You have read delim, read table, etc. A long list, as you can see here. And when you hover your mouse over any of them, you'll see the details of what you should do to make sure that you read the file correctly. And you will not be surprised that, quite a number of people are not able to do this. It may sound simple. So I did a quick survey of the participants, and I will be showing you, their levels of understanding of this pipeline, and therefore the need for this kind of workshop. The next thing we'll be doing is dealing with missing values. And we know that they are normally denoted as NAs. Sometimes in the field, people will denote missing values so differently, N/A, dashes, and so forth. But how do you deal with all of those? In the first place, you need to harmonize them, make them one type. If it's NA, let them all be that. And then you can detect where they are. And then you can decide to remove them or, to mask them. Again, quite a number of people are not able to detect these missing values. And the same proportion cannot either remove or mask them. So an example of data manipulation is creating new columns, but you can do other things like filtering, subset, select, group, summarize, etc. So I generated a simple data set to demonstrate this. To calculate body mass index, you will need weight and height. But from the formula, you'll have weight and then divided by height in meters squared. From your original data, you only have weight and height, but you can add an additional column using this function of mutate, which is in the DeepLife package. And you can add BMI here. As simple as that. But you see that a good proportion of participants, right now there are about 20 responses, but the proportion has not changed. They can't do that. And neither can they properly do subsetting or summarizing data to calculate things like standard deviation or means. So during the workshop, we will also guide participants to do simple plots like this. You have a histogram or bar plot or box plot. And well, quite about half of people are able to do this. And then the next thing is we'll try to distinguish between plotting in base R versus using ggplot. Base R is inbuilt in R, so you will not need you will normally not need external package installations. It's good for quick data exploration and allows fine-grained control of individual plot elements. It can be faster for large data sets compared to ggplot2. It allows fine control. Rather, on the downside, the fine control that it requires makes it less intuitive and requires longer code. And the default plots are less, aesthetically, less appealing to the eye and not publication ready. On the other side, ggplot2 is based on the grammar of graphics, which allows for consistency. And the default plot is usually really good and almost publication ready. Plots can be easily grouped with the functions like faceting, and it's easy to create complex plots through layering using the various geom functions. However, this requires the installation of the package ggplot2, but you also have less direct controls, and it may be a bit slower due to internal processing. And of course, the initial learning curve will be steep, but once you grasp the concept, it is really, really fun. So a number of people know about ggplot2, but a very high proportion do not know the difference between plotting in base R versus in ggplot. So in the end, I hope that you should be able to make, more advanced plots like this box plot you're seeing here. You can also do principal component analysis, by, grouping things into regions or by sex. And then you should be able to also make some, make some predictions by building models. At this point, I want to end my presentation here and invite any questions. Thank you so much.
Thank you very much, Joel Ogwang, for that introduction into your practical session. And just to mention that Joel is currently pursuing his PhD, and therefore you must be very, very hands-on. So he gave us a talk on climate change and how he's applying his knowledge in predicting some of the climate change events that have taken place in Lake Tanganyika. And anyone who had a question, this is a good time for you to ask. You can also ask about the practical. So anybody in person, anybody online, maybe thank God you can check for us for online questions. Okay, I don't see any questions. So I wish you well tomorrow and the next days following that in your practical session. I'm sure you'll send us the photographs. It's virtual, I know. And again, I know you have participants from across the region. And I'm also very much aware you've been copying me onto the emails with the forms that you've been sending out, so I can see that you've really prepared for your practical session. So thank you for that, and let's give Joel a round of applause as we wish him well. Thank you.
Thank you.
So Frederick Kebaso is online now, and Frederick Kebaso is a first Kenyan fellow of the AfricaBP fellow, and he is going to give us his AfricaBP story. And following his fellowship with African BioGenome Project, he's now running a practical for us because of the skills that he was able to get and also because of the networks that he developed. So Frederick Kebaso, if you're online, please go ahead, give us your presentation, and after that, give us an insight into your practical session. Thank you.
Frederick, I don't know if you can hear us.
Yeah, Frederick, you're a co-host now.
All right.
Frederick, do you want to unmute? Could you please unmute, Frederick? You're a co-host.
Hello, Frederick, can you hear us? I don't know.
Okay, he has now unmuted now. Frederick, do you want to carry on? Frederick, can you hear us, please?
I think Frederick is experiencing some technical issues, so maybe we can have the next session and then come back to Frederick.
Okay, he's here.
So let me try. I think for the network bandwidth, I'm going to switch off my video. So I'll talk about the bioinformatics practical. And let me just inquire whether you can hear me well.
Yes, carry on, please.
Okay, thank you. So I will be hosting the bioinformatics practical. And in this practical, I have my colleagues who are going to help me in delivering it. I myself am going to lead the practical. Then we have Jackton Oyo, we have Purity Njenga. They are going to handle the different parts, and because it is 3 days, we are going to help each other on Linux and HPC basics, because for bioinformatics, getting to understand the commands, the command line, getting to know how to navigate through the directories basic. So we'll start with the basics, assuming nobody has any exposure before, and this will work for all of us, for the team. Then from there, I'm now going to focus on genome assembly. Genome assembly is one of the key genomics analysis that is done. And in this, we'll first start with the installation of the bioinformatics tools, and we'll focus on Conda. Then from there, assessing now the data that we'll be using to do the assembly. Then after that, we'll now start checking the quality of the data, even before proceeding with the downstream analysis. Then after that, we'll proceed and do some cleaning if there's a need. Of course, in most cases, there's a need to remove the adapters and also contamination, and sometimes to also remove the short reads that can sometimes hurt. Yes, simply, then we'll do the genome assembly itself. Then now on the third day, we now focus on checking the quality of our assembly and assessing now the quality of the assembled genome. And we'll be doing this hands-on, so it will be giving you the foundational skills that you need, and you are able to follow that even after the workshop. So that is the end for the bioinformatics practical, and I'll welcome any question.
Okay, I don't see a question here. Is there a question for Kebaso here? Maybe you can also transit into the other presentation on my AfricaBP story.
Okay, thank you. So if there is no another question, there is one I think I've seen. Where can we get the link to attend the online platform? So for the participants who did confirm that they are available for the practical, had already shared the information on how to go through the installations and the link. So in case you also want to attend, you can just send your request direct to my email, and I'm going to add you to the WhatsApp group, as well as share the link where we'll be learning from. Okay, so I'll go direct to the presentation that I was to do in the morning. And I'm sorry I was not able to do it due to network issues, but I hope it's going to be possible now. Okay, the sharing a blank one. Let me just stop sharing and open it again. Then I'm going to share. Okay, I'm trying to get it into presentation. Okay, so I was to talk about my AfricaBP story, which I started saying that I can describe it as a success. And success, people define success differently. For me, I define it as a point where hard work meets opportunity. And this hard work started somewhere. And this is where I had done my BSc, my undergraduate. I proceeded to learn computational skills through the online platforms and also internships. Then after that, I proceeded to do my master's in bioinformatics at Pwani University. When I was done with my first year, this is when I got to know about AfricaBP. Then I had now to follow up 2023 towards the end. Then I got to know they have different committees. And since my project for master's was on genome assembly and annotation, I got interested in one of the committees, which was the genome assembly committee, and I got to go to the website for AfricaBP. I joined during one of the meetings that we do have online. There was a fellowship that was coming up, so I kept following up when is it coming out, and also being active and participating. So when the fellowship came out, I did my application. Of course, I did get several people to review my application. And since it was also related to the work, was when the work that I was doing for my master's, I was given that opportunity to go to Morocco for a training. And in this training, I got to meet different people from the north, such as Nigeria. We had some people from Benin, some from Tunisia, and the other countries that I cannot mention. So in Morocco, we met with the African Genome Center, the scientists there. We got through the wet lab skills first, and remember, coming from a bioinformatics background, I had not interacted much with the wet lab work and what goes on with the generation of the data before I received it as a bioinformatician. So at this point, we got hands-on skills on first extracting DNA from Acacia gumifera. That's the plant that we aimed to sequence. It's an acacia plant that is common in Morocco. So we got to extract the DNA, and from there did the library preparation, loaded it, and then did the Illumina sequencing. So these are some of the colleagues that we were there with in the lab and also in the bioinformatics part of it. So that's now the short story of the fellowship. Now I would like to acknowledge UM6P, University of Mohammed VI Polytechnic. This was the hosting institution, and we hosted under the African Genome Center. I could also like to thank African BioGenome Project for providing the platform, and also ICIPE, which facilitated my movement to and fro in Morocco. Thank you, and I welcome any question.
So thank you very much. I think you have been able to see the connection that once you join the movement, you can find your space within it. And through this competitive process, we are able to link you with partners. We have partners in Ethiopia, we have partners in DRC. The partner that was hosting this particular fellowship was UM6 Polytechnic in Morocco. The African Genome Center in Morocco is a state-of-the-art facility, and they have high-throughput systems. So we were able to ship our African BP fellows, and they spent some time there. But what has it been like for you since? Did the fellowship help you in any way? Did it assist or upgrade or enhance your skills in any way? Or, well, you don't have to say it if it didn't, but was it the networks that you developed there, or what was it? Was there any learning from that? Thank you.
Yes, because could say it inspired me to now get back to the wet lab. There before, I couldn't really like to go to wet lab and get to know how the data is generated. But after this, I have had several interactions in the lab, whereby I've been yearning now to understand how is the libraries prepared, how are they loaded to the sequence machines, then the various parameters that are set to generate the data. So it has helped me to go back to the wet lab and to get to understand the process, which I previously thought it requires advanced skills. But after that fellowship, I've been getting now back to the wet lab. And also, could say in various opportunities. I could say in the different opportunities that I've gotten, could it be for PhDs and my current place here at Walter Reed's. It has been one of the key things that I point out where I got started with the wet lab and how I connect it with the bioinformatics skills.
Yeah. I'm curious to how much money you spent going there, and how long was your fellowship, and how many African fellows were you there with? And then I think Thank God also has some questions. You can answer mine. It was fully funded or partially
funded? So it was okay. It was fully funded as the fellowship. Okay. When I was offered the fellowship, I had to go now and approach ICP, where I was working, to provide the transport only to and from. Then everything was catered for by the African Genome Center at UM6P.
Thank you for that. Thank God.
Yeah, thank you, Frederick. Thank you, Professor Mugai. So just kind of adding to what Frederick mentioned, one of the biggest challenge as a bioinformatician that one would have, and I think this would also be applicable to everyone on the call and others on site, is that oftentimes we have a bioinformatician who is purely a bioinformatician without understanding or having the experience of the wet lab. And again, oftentimes we have a wet lab person who is a purely wet lab without also understanding the aspect of the bioinformatics. So during my PhD, I had the opportunity of doing both. So I did bioinformatics and did bioinformatics, genome assembly, annotation, and I also had the opportunity of staying in the lab, DNA extraction, running gel, doing tissue culture, all of those things. So it's very difficult and very stressful because your brain is trying to do two separate things at the same time and link them up. And I think which is also why, as Frederick was talking, I could appreciate what he was appreciating. And when you do it, you may not appreciate it at that time, but when you finish that exercise and you go out and you sit in meetings and you have discussion, you would be able to connect the two disciplines in your head mentally. So, and what that means is that oftentimes you can't do that. And I think that is also one thing we are hoping that within the AfricaBP in the future we could do. That's a different thing altogether. And it's also very challenging to see people that can be able to do both or understand both essentially. But I would actually encourage everyone, if you can, if you're a wet lab, then again, try to understand or even practicalize what bioinformatician does, such that when you have a bunch of Excel spreadsheets or CSV files to analyze, and they tell you, okay, this cannot just be done by clicking a button, that you understand. And also the same thing with a bioinformatician. If you're a purely bioinformatician, perhaps coming from computer science, and you're wondering why a wet lab experiment is taking long, or this procedure is doing this X and Y. If you work in that setting, then that would help you appreciate and of course see what you can contribute. And just also to point out, Professor Ann, also Frederick, is that we basically the fellowship was basically sponsored. Again, I'm going to talk about some of this much later. Through a partnership that we had with the Mohammed VI University. I'm also going to talk about other fellowships we've given. So they sponsored everything in as much as once the applicant, lands in Morocco at the airport. So from that point, they sponsor everything. So the only thing was just a flight that couldn't be sponsored. And I think that is also what Frederick and others. I also had one of the fellows who was part of that activity, I think, was either this fellow, either in your set or another set that went to University of Illinois, have gotten a fellowship position in I think in John Ennis Center or something in the UK, right? So people keep doing things, and I'm hoping we can scale this beyond what we're currently doing. Over to you, Professor Ann.
Yes, so what happens is that we are able to connect you because some of the partners and projects that we are working with, if you apply individually, they are not very confident that you will be able to achieve the objectives. But if you come and work with us and we get to know you and you're a member of our committee or our community, and we see you there, I can vouch for you. And I normally do those recommendation letters. And these recommendation letters give you an edge over many other applications. And that is how AfricaBP is also contributing to get you further training. So do your work, come and support us. We will support you. And together we will be able to jointly capacity build yourselves and your colleagues and your institutions. Dr. Thangod will speak a little bit more about the Africa BioGenome Fellows and the other fellowships that are going to come on board. Final comments, Kebaso, anything you want to say in closing?
Could just say that as you look for opportunity, you will always need a place to start from that gives you a starting point. And I could say that being interested in genome assembly and bioinformatics gave me a base to start on. And I would like to thank AfricaBP for starting this enormous project and initiative. It has been of great help. Thank you.
Thank you very much. Frederick, we wish you well in your PhD studies and in your new position. And I know AfricaBP will be there to walk the journey with you, as we will be for all the other young early career scientists. Thank you. Let's give this young scientist a round of applause. And now, as we're coming to the end of day 2, we have just 2 more talks. One is a panel session, and the other will be the talk by Dr. Thangod on the African Open Institute Fellowships, because I think that's where we have been able to stir up your interest, and I know you're very keen to know how you can also apply. So I'll invite panel members to come in front of us who are young scientists, and we will be discussing the question, what is a young scientist's role in One Health? So first, let me invite Fatma Mohammed Omar. A round of applause for her. She is a passionate young budding scientist. You heard her presentation on AgriGuard, and we're looking forward to hearing your role. Next, we will invite Dr. Rachel Kimani, the postdoc from Mount Kenya University, who is also a member of the Gitaka Lab and has been a coordinator for this particular project, and we're very grateful to you. And then I will invite Simon Irongo from the Gitaka Lab. Please give him a round of applause as he comes up. You have seen him around helping us with the logistics in the place, but is not a logistician, he's actually a scientist in the Gitaka Lab. And then finally, but never the least, I'll invite Karani Anthony from Ecosystem Restoration. He was here earlier and we discussed the rehabilitation of the Kenyan ecosystems and the different various species. So 2 ladies, 2 men, it just happened like that. It wasn't deliberate. But I would like to start with you, Fatma, because you've been very active in this place. How difficult was it for you, or how easy, in other words, was it for you as a young scientist to enter into some of the places where we're seeing you, rubbing shoulders with the number 2 person in the country, being invited, to sit on the front bench? How easy or how difficult was it for you, especially coming from the perspective of a young scientist?
Good afternoon, everyone. Thank you for this opportunity, Professor Anne, and thank you, Africa BioGenome, for allowing me to be here. I would say it wasn't an easy journey, actually, because it had a lot to do with the fact that it started from basically my interest in science communication. That's beyond the lab, what I've been doing. And I've been attending a lot of workshops in terms of early career scientists and how we are able to communicate science to the outside world. And I would say I've literally been involved in many workshops and trainings that have ultimately led me to where I am today. And I have had various ideas in terms of science communication and how we can be able to drive agri-biotech outside here. And in terms of having my ideas, I presented them to various people. They were telling me, it's possible, but they're not giving you a way forward. They're not telling you anything. But I once got an opportunity from, there's a lady from ISA AfriCenter. She knew about my idea and she was like, you remember the idea you told me about? We have a hackathon. Can you come present your idea? And maybe we'll team you up with a couple of people where you'll be able to come up with something substantial that you can use. And that's how I can say that was like an opening for me. And we got to present in that hackathon. People loved our idea. Again, we were ranked the top in that hackathon, and that gave us the opportunity to go present in the FINA Summit. And that's how it all came about. And basically in the field of science communication, I'd say I've been pretty much active. I think because of my proactiveness, I've gotten an opportunity to attend various podcasts. We have a science podcast called Africa Science Dialogue Podcast. It's basically, again, a podcast that is trying to drive misinformation, or rather to educate people about agri-biotech, climate smart innovations, GMOs and things like that. And I've also been able to been invited to interviews by Daily Nation under a section called Seeds of Gold, where I was also able to talk about youths in biotech and how we can encourage our youths to drive biotechnology here in Kenya.
So thank you very much for that. I'll come back to you, but now I want to go to Rachel, who studied very young, got your bachelor's, got your master's, got your PhD, now you're doing a postdoc. What does the society say about a young lady who's spending a lot of time studying, publishing, now being a faculty member at the university? Is it easy? Is it easier being a man in your position, or there's no difference? Maybe Kenya, maybe there's no difference for a lady or a man. But what is your perspective about that? Do you think it would have been easier for a male in your shoes? And what would you tell the young ladies who are present here?
Okay, thank you for the question. First of all, I would like to say that it's not always an easy path for being a lady in sciences because of family responsibilities.
And in addition to that, as a lady, you have to be more aggressive when you want to achieve career progress. Immediately after masters, some ladies want to start family or go directly into PhD, which again will delay family. But all in all, we have examples of ladies who have excelled. Professor Anne Muigai being one of them. We always look up to her and how she conducts herself. And therefore, having a great mentor, you get there at the end of the day.
So thank you very much. You're also in a great lab with great mentors there with the Professor Jesse and Dr. Kanoi. And what can you say about how that has impacted the standards? Because people do science, and depending on the sort of science that is happening, it could be at different levels maybe, but you're churning out international standard, world-class research. What would you say about that?
Thank you once again. So meeting my supervisors or the leaders of the Gitaka lab, that is Dr. Bernard Kanoi, Professor Jesse Gitaka, I may say was a miracle because from the moment I finished my MSc to now get into PhD, there was a lot of back and forth, writing proposals, not getting very good guide, but once I settled in Gitaka lab, I was able to quickly decide on PhD title, quickly collect data, get a lot of support from the other members. And I can say just being in that setup really made things go smoothly up to now achieving the PhD. So great mentors must be there. They allowed they allow the students to attend as many trainings as they can. Training on grant writing, training on proposal writing, training on research methods, data analysis, wet lab, genomics, and all that, which culminated to who I am today.
Thank you very much, Rachel, for that. But now I want to move on to Simon, also a member of the Gitaka Lab. What does One Health mean to you? And maybe we can hear from you what area of One Health you're working in, and what is the place of the youth in that particular sector?
Okay, thank you so much, Professor Anne, for this chance, and also the African BioGenome Project Organizing Committee. Okay, as you've heard, my name is Simon Irungu, a member of Gitaka Lab in Mount Kenya University. Currently, I'm working on antimicrobial resistance, that is detecting the beta-lactamase genes in the gram-negative bacteria. And actually, in this area of expertise, young people can play a role by maybe integrating and advocating for the policy making, because we are doing a lot of research, yes, but we are not politicizing it. Not that you can come with solutions. You know that there's a resistance for a certain antibiotic, but nothing is done. So what youth can do is to advocate for this, and as well, maybe to use the AI. We are a young generation. We can use the AI to generate the mobile apps so that we can inform the community on the resistance patterns. Thank you.
And what challenges have you faced as you embark in this area of AMR and changing existing policies?
Okay, the challenges that are there is that in AMR, you face a lot of challenges in hospitals. Maybe when you go to collect samples, you find that you have to collect the metadata, and maybe you had recommended a certain antibiotic not to be used and move on to another line of antibiotic, but you find that they are continuing to use the antibiotic that you had said already, out of use, resisted, Yeah.
So transformation of science into action can have some delays, and I guess that is an area that we need more advocacy. But perhaps we can move on to Anthony. And Anthony, as an environmentalist and as a person who's working in this area, what role is there for collaboration, especially with different sectors, but more importantly, those that are involving a lot of the youth? And how can we ensure that the youth are not left out as this science progresses?
So thank you so much. The first thing is that I believe that Africa needs a new face, and that new face is you. So to understand this context is that most countries in Africa are developing countries, meaning that so many things have not been done, so many gaps needs to be filled. A lot of things, like literally everywhere you go, you can have something new to do, you can have some impact to create, and that is an opportunity for everything. As I said earlier, I believe in passion for nature and love to humanity. That what we do to help the ecosystems, to help the environment, comes back to us. Rather, we are doing it for ourselves. And because of that, you realize that so many things are interconnected. You realize that the food we eat has something to do with the animals that are in the forest. Our agricultural systems all depend on other factors such as pollinators, such as the watersheds and everything. And therefore, when these things are all connected, means that we all need to work together towards some common goal of uniting this entire thing. So for environmentalists, I believe there is a chance to work with people in the biosciences, in the doing molecular sciences, to be able to understand whether some of these things that are missing in the environment are probably can be used, facilitated by these technologies. One of the things that I see, for example, when I'm dealing with frogs, you realize that there are so many kind of bacteria, kind of fungus that are affecting these species, and some of these things are found naturally in nature, and they can also affect human beings. And therefore, when we are talking about the health of these animals, we are literally talking about our own health. And one of, for example, I can say that one of the things we have in snakes, for example, we have leptile-associated salmonellensis. And some time ago, I cultured a bacteria that was from frogs and also from snakes, and clearly it was a salmonella. So you realize that these things, some of the antimicrobial resistances are not actually in humans. They probably start in those animals and then they spread. You realize that if talking about antimicrobial resistances, you realize that some of our wastes, some I've seen researchers talking about the presence of medical waste in our water systems, in the water that we drink. And this medical waste mean that if you are antibiotics, then those antibiotics are still present in the watersheds. So if those bacteria are resisting those antibiotics or are being exposed to those antibiotics, means that is where those things began, not in the humans, but in the animals. And therefore, looking at this entire thing being connected, then I believe that it is a chance for us to work together. Finally, I believe in talents and patents. So clearly, I am continuously day-to-day looking for talents. So if you feel that you are a talent, you can see me. I will need a place where we'll collaborate together someday to do something bigger for the sake of Africa and for the sake of the securing the future survival of our future generation. Thank you.
That's an excellent point that you have made, that it's not only for us, the scientists who can mentor others or who can collaborate with others, but you young scientists should also mentor other young scientists, because there's something that you have that you can always share with somebody else, and there's a network that you have that somebody else needs, and you don't need it. So share that network with somebody else, because there's always something for the other. But coming back to you, Fatma, you mentioned, actually Fatma and Rachel, both of you mentioned the importance of training and the importance of attending workshops and capacity building things. Can you elaborate about more about that as you look at your journey back and also where you're going? What is the role of capacity building? And I'm just wondering, all the panelists here are very confident. Is there a workshop where people go and learn how to be confident and to be eloquent? And if there is, can you share so that we can be able to share it across there?
I don't think there is a workshop that tells you how confident you can be. There was a time, actually, I presented in a certain conference, and my confidence, it was terrible. But I was like, I have to do this. I have to deliver this message, and it was delivered. Sometimes you need practice, as in terms of confidence. That is, you just need to practice. If you can stand in front of the mirror and talk to the mirror, that is fine. But yeah, basically that's in terms of confidence. But in terms of the workshops I've been attending, I've attended a lot of workshops in, because I had an interest in bioinformatics, yes. So I've attended various workshops in ILRI, IITA, where I've learned a lot of skills in Linux, analyzing, using various pipelines, and just apply. To tell my fellow youths, when you see any opportunity out there, just apply. You never know what that opportunity is opening for you. So just apply. It doesn't hurt. And again, in terms of science communication, it's an interest that I picked up sometime last year. I didn't know if I could talk science. I thought I was only meant for the lab and for coding. And it felt really good to be outside there talking to people about science, talking to farmers, talking to policy makers. Yes, and yeah, it's a good opportunity that opened for me. And I want to encourage again my fellow youths to not only dwell on the wet lab or the dry lab, but also go out there and talk about science to all these people out here. And just to add, we are, let's say youths, we are all digital natives. We know about tech, we know about everything and AI. And we can we are very good at social media. In Kenya, first of all, we literally can organize, maandamano in social media. So you can imagine what we can do in social media. So let us use it for the right reasons. We can use it to talk about science, to advocate for science. Whatever small thing you have done for science in this world, tell it outside there. There's someone who is listening. There's someone who might want you in their platform. There's someone who is interested. There's someone who can fund you. There is just someone who can even give you a PhD or anything. So don't hesitate to go out there and talk about science in LinkedIn or on your X hashtags, and even on TikTok. We want to become content creators. So yeah, thank you.
Thank you very much. What does Rachel have to say about that?
First of all, I'd like to say that for Gitaka Lab, we normally have weekly lab meetings whereby people are able to present their progress reports. So it's an avenue whereby you're able to practice a lot on presentation, communicating to others, and especially science. Previously, we've been told here that if you cannot summarize your work and communicate to a lay person, then you're not making much impact. Therefore, that is only attained through practice. Secondly, conferences that are designed like currently what we have, whereby it's not just theoretical knowledge, there is hands-on experience sessions, are very good, because when you attend such, you widen your knowledge base, and also you're collecting a lot of information which will help you to come up with research questions, become a better scientist and a grant writer. So about science communication, when I joined Mount Kenya University, the research department, there was an ongoing project on science communication. It was being ran from the social sciences aspect. At that moment, there was, that was back in 2017, there was the tetanus vaccine that was being resisted a lot because of rumors that it was contaminated with birth control hormones. And at that moment, it was the failure of scientists to communicate to the community. And so science communication is an art that we all need to become better so that at the end of the day, we'll have community impact. Thank you.
Thank you very much. Nothing to add about science communication, but as we move on to Simon, what is the role of innovation in your science? And the young scientists are very good innovators. Can you tell us a little bit about how those innovations are getting out there and how easy or how not so easy it has been?
Okay, thank you again. So the role of the innovations out there is so much. At first, I just mentioned about AI, developing the apps that are based on the AI. We've seen them out there. They are so much used, and some of them, they concern health. And actually, to be very, what will I say, tentative or very clear about it. For example, in our website, that is Getaka Lab, we have an AMR that is GPT, whereby that's an innovation that someone came up with, and it normally helps one to go through the AMR things and blah, blah, blah. And then another thing is that innovations may bring about change in the health sector. Let's see, for instance, I can say a quote that is saying, "We are not the leaders of tomorrow, we are leaders now." So youths has a step to make, come up with innovations that are needed in the economy, to raise the economy, and as well to improve the health of the Kenyans and the world. Yeah.
Thank you very much. That's very, very powerful. That there's a saying here that politicians also always say, "You, the young people, join us in this endeavor. You're the leaders of tomorrow." But you're saying, no, we're not the leaders of tomorrow. We're the leaders of today, and I really, really embrace that. But finally, let me just ask Anthony, what about the youth in the communities? What can you say about them, especially that you're in an area that is using applied sciences? You're taking real-time scientific data and then you're applying it in something that is very practical, like restoration of ecosystems and forests and studying frogs. What is the role of the youth who may not have reached the education level that you have, or may be looking for opportunities, and what is the place for applied scientists in those communities?
So thank you so much. It's a very good question. Sometimes people consider me controversial, but I give myself a tether. The thing is that about communities, about the youth, about innovation, it's all in our mindset. Sometimes I feel like we look for big things that are actually not there. Sometimes I feel like if we consider different things to be innovation, sometimes we tend to forget things that are simple, that are problem solving, that are critical to our survival. So for example, if we are developing a mobile app, if we are using AI to create some kind of solution, what happens to the vegetables that are growing in our neighborhood? What happens to the plants that are very endangered in the places that are very remote? Sometimes I feel like I would dare someone to build a city somewhere in the desert from scratch, because we have the bricks, we have sand, we have everything. We have mines that we have rented out to the west. We have facilities that are that are everywhere, but yet very few people who are committed to utilizing them. And the solutions that we are looking for the local community are not very far. They're actually things that are simply where we are. For example, how much would you need to build this house? Or rather, what would you need to build this house? You need bricks, you need concrete. And where does it come from? From America? Mostly it comes from around here. And some of the solutions that we are all looking for are not out there. They're actually in here. And it goes back to our appreciating our own self. I started by mentioning earlier about Okot Pipitek, who said that Africa to him is darkness. The guy was talking in some satirical language, talking about the personality, a man who doesn't love himself, a man who wants to do what other people are doing, yet he is very rich in his own making. And he even asked satirically in the same poem that what did you expect would happen on the days of Uhuru? Did you expect that the trees of the fig tree would become silver notes, would become notes for women to go collecting as they go to collect for firewood? Or did you feel that dew would become gold for hunters to go and collect as they go setting up traps? In other words, it starts with our own self, appreciating that being an African doesn't mean that you are inferior, or rather you should leave the solutions that are just next door and approach solutions that are quite far. I must admit that today I feel very civilized, and that is especially because I remembered to comb my hair, which would make me look like a bandit or some mau mau from the. The thing is, as I mentioned earlier, the solutions to local communities, to the youth, do not lie in solutions that sometimes we exaggerate, the things we believe so much in. Those solutions are just next door in our communities. I do feel that we should sometimes leave alone so many things that we are doing and go back to our communities and mobilize them to be able to build from the things that they have. Today, I may want to sequence a genome of some very critical endangered species from the forest. I come from just next to Mount Kenya forest, so I access forest as I go to graze my father's goats and stuff like that, and I sometimes encounter very endangered species, some of which have not been seen for a century, since probably 1914 also, when a white settler came and collected them and took them to Germany. And there's no collection known for 100 years unless you go back to Germany to see that collection that was collected. So for me to sequence that genome, I need a sequencing machine that was developed in America, and it's very high priced, and also the president, or rather should I say the government of Kenya, will tax on that same item. I will need reagents, and those reagents were actually developed in Germany, and while they come to Kenya, they will require to be taxed. But then I keep asking myself, where do these raw materials to make these items come from? Do they probably come from ores that are in DRC Congo, that we have rented out to some Chinese company to mine and export to Russia, and then Russians produce something small, and then they sell it to England, and then England produces that machine, and that machine through Ireland, because they have a very reduced taxes, they sell it back to America, and then America sells it back to Kenya at a very exaggerated price. Are we like hijacking the point by believing that solutions to our local communities belong in the West? And what about the future of your children? I keep asking people. Are you using our resources sustainably for the future of your children or the future of aliens, I believe. So thank you so much.
Such a powerful way to end this panel. Please give them a round of applause. Basically, what we have heard is that science communication is key. And if you can be able to get yourself some skills in communicating in science, it will take you a long way. We have heard that innovation and youth go together, and the youth are continuously innovating. We have heard that there's a role for the youth in all the areas of One Health. But as we finalized, we have heard that confidence is something that you build through practice. Time and time again, presenting your results, you'll be able to build practice as a young scientist, and you'll be able to have confidence. But the solutions, and we can end with that statement, the solutions to our problems are around us. We only need to go back home to our communities and look at those solutions and see how they can help our communities. So thank you very much to these young scientists. Let's give them a round of applause as they take their seats. Very informative. If nothing else, it should give you confidence that there is a future for you as a young scientist, and your future is now. So come and embrace and take over the science that is definitely yours. So we are now at the point where we will hear our last presentation of this regional workshop. And I'd like to invite Dr. Thangod Ebenezer, the founder of African BioGenome Project, and now he is talking about the AfricaBP Open Institute of Fellowships, building the next crop of African biodiversity genomic scientists. Dr. Thangod, you have the floor.
Thank you so much, Professor Mugai. So I'm just trying to open my slide. Okay, all right, I got it now. Okay, so can you see my slide, please? Yeah, so yeah, I won't be making much of a presentation per se. So I would be doing more of, so I'm just trying to, yeah, I'll be doing more of I would like to make it more interactive. It's going to be like a mixture of interactive and, talking as well. So how about now? Can you see the full screen?
Yes, we can, thank God.
Is it moving?
Yes, it is.
It's moving. All right. Okay. Thank you so much. My name is Thangod Ebenezer, and the co-chair, one of the co-chairs of the African BioGenome Project. There are 5 co-chairs, one per region, and East and Central Africa is being coordinated by Professor Anne, one co-chair. And I am the one of the co-chairs, mainly looking after the operations and more about the upstream coordination of each of the regions, as well as helping also with the scientific direction of the project. With this presentation, I would just try to make it interactive, and in between, I would leave the slide and talk and then come back to the slide. Just to provide a bit of a background, Professor Anne Mugai spoke yesterday about the AfricaBP, but I'm just going to briefly talk about 1 or 2 slides that overlaps with what she talked about, and that will help me provide context into the Open Institute. So the AfricaBP, again, as you saw here, our aim is to use genomics to inform the future of biological diversity across the continent of Africa. And there are 3 goals. The first goal is to coordinate all non-human genome sequencing activities, especially in the non-human space, sequence over 100,000 species. And I must say that there are many more species within Africa. So there's a chance that we could start discovering more species because not all the species in Africa has been discovered so far. And while doing that, we expect that will naturally build capacity. For example, we've had institutions who have worked with AfricaBP who haven't sequenced genome before, and then they have genome sequencing, and also doing so many things, interesting things, and building global networks. So we believe that the thought process of actually sequencing the genome is naturally built into capacity such that the capacity does not need to happen in an isolation. It's already part of the whole process. And that would mean initiating and developing, initiating new sequencing efforts, either by inspiring countries or inspiring institutions or provoking actions, developing and supporting activities. For example, in South Africa, there is now the 1000 South African Genome Project. In Morocco, they're also planning their own project, and also in several other African countries. All of these things are now springing up, mainly looking into non-human space, because the plants and animals and eukaryotic microbes have been for a very long time neglected, and which is understandable because with humans we are more concerned about humans and about ourselves, but living the environment. But COVID-19 has taught us that we cannot live in isolation in the ecosystem, and that if we do not take care of the environment, or live in harmony with the environment, or try to also understand the environment with which we live, then of course we all know what happened in terms of the COVID-19 pandemic. Then finally, we aim to then use the information we have to inform policy. A few years ago, I was invited to speak at the Food and Agriculture Organization of the United Nations, and again, I was able to bring in insights from the Africa BioGenome Project. A few years ago, also, my colleagues spoke at the African Union and contributed insights. So we aim that some of this will help inform discussions, either through our writings or through the workshops we had, or through other discussions that are emanating. So essentially, the idea is that the AfricaBP helps to kind of be a voice for especially indigenous people, local communities, and generally for voice of people that are not being heard. We have 5 grand challenges. The first one focuses on genomics and bioinformatics technologies for the agri-environment. The second focuses on crops and livestock improvement and health. The third focuses on genomics for conservation of endangered and endemic species. The fifth focuses on socioeconomics, ethics, and policy. So, and I mean, with this fifth one is very important to us because we are not just interested in the technology, we're not just interested in the science, we're also interested in how the science is done and how our activities help the African people and institutions. And ultimately, we're also interested in the technology development, the knowledge exchange, and in terms of commercialization and industry. And of course, all of this revolve around value proposition and capacity building. So we aim to sequence mainly non-human eukaryotes. So we do not do human genomes. We focus on the places where there are gaps, which is plant and animal genomes, and also other eukaryotes like insects, algae, other animals, and other vertebrates and invertebrates that are oftentimes overlooked within the scientific ecosystem. And so how does the AfricaBP operate? So the project started off quite organically from a Friday activity, and also with Professor Anne Mugai and others. And then, but quite recently, we've now had a governing document, which is a constitution. And here you can see the structure. And of course, if you have a phone that can read the QR code, you can also use the QR code to get the constitution. So the constitution kind of defines how the project is currently being run from this year. And it's again influenced by our experience within the project, and also ensuring that we have a distributed operationalization of the project. So what does that mean? That means, for instance, even though there is a coordinating center or a continental center or leadership, the region are also have their own regional leadership, and the countries also have their own national leadership. And anyone within that ecosystem can sequence a genome, anyone can contribute, anyone can do anything, but of course, giving a coordinated direction. So there are 4 programs within the project. The first one is focusing on animal species. And again, animal here does not mean human. The second one focuses on plants. I must say that there are over close to 1,000 plant species that has been sequenced. I think 3-4 years ago, it was around 800, but hopefully now it's like 1,000. And of that, only 20 are African endemic species, even though there are over 45,000 or so, or just about 45,000 plant species on the African continent that has been recorded. So again, of all those 20, as at that time, 2022, I think only none has been sequenced, and it's just quite recently that we've now started to see some communities. I think since 2022 or so, started to sequence a few, and even as at that, one of the things we're now pushing out is how can we sequence end to end, and not just sequencing one bit and then shipping the other bit outside the continent of Africa. So we want to do everything 100% on the continent. And the third program is on community of practice on biodiversity framework. Within this program, we talk about, for instance, the issues around ethics, regulatory frameworks, the Nagoya Protocol, the Kumi-Montreal Global Biodiversity Framework, the International Treaty on Plant Genetic Resources for Food and Agriculture, the Convention on Biological Diversity. So all of this falls under this program, and of course, there are also several papers concerning this. The fourth program is the Open Institute for Genomics and Bioinformatics. This program focuses on mainly knowledge exchange and in terms of how do we work with African institutions to do these things locally. And when we started AfricaBP, as Professor Anne mentioned, the idea was, okay, we're going to sequence, but we need to create a framework whereby that will happen, and not just we sequence genome and publish it and nobody knows about it. No, we sequence genomes, we come to a forum like this, like the regional workshops, and then we discuss it. And also, we also invite others to come and present their work, and of course, we learn from each other. So I'm going to zoom in more on the program 4, which is relevant to the discussion here today. And of course, we have other opportunities, coordination, fundraising, partnerships, and policy engagement. So as I mentioned previously, the project started off organically, unlike other projects where you have an institution and then you have funding and then you have all the necessary structure you have is just for you to run the project. But with the AfricaBP, it started in the opposite direction, and I think that could be a difficult start as well. So eventually we held a partnership meeting at the University of South Africa, hosted by University of South Africa, where afterwards we also signed a partnership document with the Science for Africa Foundation in Kenya. The program 4, in the next few slides, I'm just going to talk about the program 4. We've done quite a number of things within the program 4, and the program 4, which is the Open Institute for Genomics and Bioinformatics, focuses mainly on regional workshops, online workshops, training, capacity building, and infrastructure development. So we have a paper on infrastructure. So, and one of the first things we did in 2022 was to organize the first regional workshops, and we published that in Nature Biotech. And the title of the workshop is Bridging the Gap in African Biodiversity, Genomics, and Bioinformatics. And we also launched, of course, in that workshop, we launched the Open Institute. And then afterwards, we had around 700 or 800 applicants. And of course, after that workshop in 2022, in 2023, we evolved the activities and then established the regional workshops, which is what we have here today. And in that workshop, we had registered participants that participated remotely on site. And of course, they are from, I think, close to 70 or 80 countries with around 49 African countries represented. And we published that in, of course, there were up to 4,000 applicants and so. And we published that in Nature Genetics last year. And then again, we also have the 2025 regional workshop, because each year we publish these papers and kind of define progress, where we are. This one has been accepted, or rather it's been published already, actually last week, in the current paper, which focuses on bioeconomy for the African continent. So as part of the regional workshop, the Open Institute, we also launched fellowships. The first fellowship was the African Genome Center AfricaBP Joint Fellowship in 2023, and that is a fellowship that Frederick mentioned. So in this fellowship, we partner with the African Genome Center to organize I think a 1 or 2 weeks fellowship in Morocco. And then afterwards, we also partner with the University of Illinois and about 4 other African institutions to launch the African Biodiversity Fellowship for Emerging Genomics Leaders. So this fellowship kind of hosted 4 African scientists in a lab in Africa. They spent 1 month in a lab in Africa that is not their lab. And then they spent 3 months in the United States at the University of Illinois. So many of them are already involved in organizing regional workshops, just like Frederick. Many of them are already heavily, they are like some of the key people also, coordinating this workshop heavily across the regions. Then the final one, we also worked with the International Institute for Tropical Agriculture and Nkaba Biotech to actually launch the African Plant Genome Assembly and Annotation Fellowship. And this is again form part of the discussion that Sadiq presented earlier, the genome, whereby we use the genome, sequence the genome, and also use that data to train about 8 African researchers. So with this, I would like to thank you all for listening, and we'll be happy to answer any question you may have. And I would like to also thank everyone in the project. So it's a village, and also there are just so many other people to thank. And I will be looking forward to answering any question you may have.
So thank you very much. Thank God. Is there a question? Maybe you can check online whether there's any question.
Yeah.
And here.
So we have online, we have, so Tobias, I'm going to come back to you, but let me just read like once online. So people are asking, is microbial genomics not a priority? So we do microbial genomics if it's within the scope of, if it's under the eukaryotic taxa. So we don't do prokaryote, at least not for not yet, because prokaryote is quite another level of there are just quite a lot that are in there. So we don't do prokaryote, bacteria, viruses, and the rest. So we just focused on eukaryotic microbes, anything eukaryotic microbes downwards up to non-human. Then I think, yeah, Tobias, you want to ask your question?
Are there any questions in the room?
Thank you. Thank God.
Mm-hmm.
Yeah, you've done a great deal. That's why when I was talking before, I said you are the hub of the African BioGenetics program. For you through, including Mogaye, all of you that are the founding fathers of this program have done great.
And founding mothers.
Founding fathers and mothers. Yes.
And not just, the 4 of them as well.
Yes. And that is why people like me will be able to today stay in Nigeria and enjoying these beautiful lectures happening far away there in Kenya. I wish I had the opportunity of the practicals too, which I applied for. Anyway, I couldn't get them, but all the same, it's still good. So, but the question I'm asking now relates to, I've asked this question before during the West African project, that of last year. You see, some of us are faculty members, and we're all brigades, in quotes, so to speak. We are professors, and then the in now is this genomics in the biological sciences, making rounds and then creating waves. Yet we are not there originally. We are now trying to learn through the hard way, struggling with the youth to learn what is so difficult, so to speak. And I ask, all these opportunities of fellowship, because for me, you can see I've been following the trend. Wherever it is happening, I'm applying. Whether in West Africa, East and Central, then North, I do apply. South, I do apply. I stay online like this and then participate, hoping that actually chances will be given for the online, on the on-site practicals. Because I want to acquire the skill and then change my game, research direction. So I ask, why is it that we professors, are we written up? How can't we be involved to learn, to be part of this fellowship and have opportunity of being there in the laboratory to learn? Professor is only a title for me. For me, my heart is like the youth. I want to learn. And I want to be able to, you don't give what you don't have. Not until I learn to acquire it, I cannot actually give my student the practical. I can only talk the way I talked before. But I now want to acquire the skills and be able to help them practically too. You give them a guide from practice and they have confidence the more. So, yeah. Sir, how do we handle this kind of situation?
First of all, AfricaBP is open to everyone across all career stages. Early career, mid-career, established scientists, even scientists that want to make a shift in the science. So it would interest you to know that some of the scientists in sequencing and assembling our genomes weren't doing this before. So they got involved in it, and now they are publishing papers, and they haven't done this before. So the project gives African scientists the opportunity and the place of, will I say refuge in quote, to get involved in things that they feel is intimidating, that they feel that naturally they will not be able to do it. And ultimately what the project does is once you're involved in the next 2 to 3 years, you'll begin to see it. So I won't give you specific examples, but several both young and early, mid established scientists come into the project without, having done like all the genomics. What they do is that they work with us, we put them through the process, and they do fantastic things. And when I look at the results have actually keep inspiring me. So which is why sometimes I always look at possibilities in African science, because what I've seen by just working on AfricaBP, in fact, I don't even work on it. I just, I have people like Professor Muga and others. And what I've seen by just doing that, and those things that I've seen rekindles my hope for Africa. And which is why oftentimes I talk about, I don't talk about like, oh, money, technical. No, our problem is not money. Africa's problem is not funding, right? Africa's problem is also not technical. Given the experience I've seen, because I've seen, for instance, institutions that are not so well established and not, they don't have the big resources, but they partner with, for instance, other institutions to get our results. And I asked, why can't we do this at scale, right? And to answer your question, we also are looking forward to, for instance, other fellowship opportunities that have like established scientists. So in one of our papers, we talked about establishing professorial chairs positions. So that should be in the Nature Genetics paper. So just type Nature Genetics establishing African regional workshops on Google. We talked about establishing professorial chairs positions. So perhaps that could be one area that people like yourself already in the space could be, for instance, will compete for the positions and things like that. Again, that will require like resourcing and funding. So we are still discussing all of those things, but just know that we haven't left. We didn't overlook you guys. You are still in the radar, but it's just a question of, okay, we have these resources and this opportunity. Where do we fit it? And oftentimes the opportunity comes to, for instance, early career, mid career, and even though we have we have like a report on professorial chairs, so we're still kind of looking out for those opportunities. I don't know if that answers your question. I hope it does.
It does, yeah, thank you.
Yeah. Okay, we also have another someone. It looks like Professor Anne, a lot of people are raising up their hand and asking questions. So I hope if you indulge me to take them. Peter, do you want to ask your question, please? Unmute.
Good afternoon, sir.
Afternoon.
Thank you to organize the program. From Nigeria.
Could you please speak up? We can't hear you clearly.
Yes, I am Wilson Wapweze Peter from
Okay.
Nigeria, doing a PhD program at Northwest University in South Africa. So the issue is I've been part of this African BioGenome Project for 2 years now, but I've not had chance to participate in any of the hands-on practical sessions. So I'm asking, when is this workshop, our fellowship announced that I can apply? Is it that there are months of the year when they are made available? And what is the modus of application? Do we have a database or website where it's announced? How is the communication made? Yeah, I am a bioinformatician. I am training. Mostly, I'm training myself using resources available on YouTube and the gap for my professors my encountered. So, but I want to advance to global level. I can't let everything on my own. I need structured training. So, and I think this workshop and fellowship will help me a great lot. So, other researchers who equally aspire to access such training. So, please, I need information on how and when the applications are made available. Thank you so much.
Yeah, thank you, Peter. So first of all, are you on our mailing list?
Yes, yes.
Okay, the first point is our mailing list. We usually send out the call on our mailing list. And usually for Southern Africa, the call will be like around March, April. So just join our mailing list, and then you always get the information. So also I shared the social media communication platforms in the Zoom chat. Please follow us on the social media because they will always go out on those platforms. We also have a Telegram group. So please follow us on those group. You will be able to get one.
Yeah, let me just add that the AfricaBP does not, at the moment, we don't have the finances to sponsor accommodation and transport. And we realize that this would be a challenge if you are applying from South Africa for the East African Conference. So my encouragement to all is to apply for the workshop, for the practical. You can attend any of the other workshops virtually. But if you're in a region, apply for the practical hands-on training in that region because there's a greater chance of you being able to access it. So for instance, in South Africa, the South African workshop is happening in September. Go to our LinkedIn page or X handle and you will see the dates. Apply for that. Unfortunately, I think it's already closed.
It's closed, yeah.
But there's the one in Morocco. So next year, apply to go for the in-person attendance in the regional workshop that is in your region. You stand a higher chances of being invited to attend in person because the barrier is less. Thank you.
Yeah. So and just to add that we try as much as we can to provide a lot of opportunities. So every year we've trained at least 400 African scientists in genomics, bioinformatics, molecular biology, ethics, sample collection. So collectively across all the regions. So we published that in the Nature Genetics paper, which you saw. We trained around 407 or so in 2023. In 2024, we trained 401, and the paper was published last year. And we had a participant from over 50 countries. Again, we would like to take up the number we trained. For instance, we received close to 4,000 applications, right? If we had the resources, we would train everyone. We'll give everyone the opportunity, right? But sometimes you have to look at the resources and then, be able to say, okay, how many can you train? So as Professor Mugai mentioned, please, maybe perhaps you can keep an eye on the Southern Africa one coming up next year in 2026. Just follow us on our Google mailing list, social media platforms, Telegram. You always see that come through.
So for every application we receive to attend in person, there are 10 other people who want, because for 4,000, actually more than that, for 4,000 applications, we have trained 400. So you have to make a very good application. And congratulations to the ones who are in the room, you made the cut. In your a pitch for why you need the training, please put some effort there. Sometimes we see people who leave it blank and it just limits your possibilities of you attending the in-person training. Thanks.
Yeah, so Professor Mugai, we still have lots of questions coming through. Are you running sort of time? Maybe Stephen Mburu and one chat can come through.
Yeah, we have about maybe another 10 minutes.
Okay, all right, that's fine. Maybe we can take, because it looks like this is an interesting bit of the, one of the interesting. Others are also interesting. Stephen Mburu, please ask your question.
Hello, everyone. My name is Stephen Mburu from Jomo Kenyatta University. I take BSc Biochem and Molecular Bio. My question is, first I would like to thank you, Dr. Ebenezer and Professor Ann, for quite a good opportunity. So my question is, this year I attended Plant Genomics Symposium and I really got to find a different meaning of early scientists, and it doesn't involve undergrads. So how are you going to factor in undergrads who are interested in actually learning the skills and also applying? Because I know fellowships, if an undergrad and a postdoc and a PhD students apply, chances are the undergrad will be, yeah, you understand. So
okay, thank God. Let's take one more question, then you answer the two together.
Okay, all right. So is there another question on site? Because it looks like the question online.
No, there are no other questions.
Okay,
questions?
the question online is more around, so maybe I'll just answer them as I speak. So
okay, very well.
So so we, again, as I mentioned, we don't say oh, we are more into postdoc or established careers or things like that. But what does happen is we do have criteria for people participating in practicals. So perhaps what will happen is I will try and share it. We've published it before. If you Google, you'll find it. So one of those criteria is that we give priorities to people that have a project or already have maybe either previous project or ongoing project or are looking forward to having a project. And usually what would happen is that we would expect that they would describe it in their personal statement or motivation. So, and as Professor Mugai mentioned, sometimes if you don't describe it or you just think that, oh, you just submit and you will take in, then it becomes challenging because you are competing with 9 other people for each position. So I think that is one of the things. So it's all about, do you have a project? Have you had a project in the past? Do you have a current project or do you have a future project? So that is step 1. Step 2 is, were you able to describe it very well in the application? So these 2 things. And the final thing is, if you are in Southern Africa and you're applying for a workshop in Eastern Africa, ordinarily we will not be shortlisted because priority will be given to those in that region first. Simply because we don't fund travels. There's a chance that if someone is from Southern Africa attending Eastern Africa, they may not be able to cover the cost. So we often focus on, and again, we move around. So we'll always come to your region. So we'll often say, okay, we're going to come to your region. When it's time in your region, you can actually apply. So I think that is one thing that I see. Professor Mugai, I'm just getting some comments. If you give me 2 minutes, I will just, how does that sound?
Sure.
All
Go
right.
ahead.
So someone is asking if we exclude prokaryotes. So as I mentioned, we don't do prokaryote for now, at least for now. We are focusing on eukaryotes because there are just so many eukaryotes apart from human that haven't been interrogated. The prokaryote is a whole different word altogether. Then is there any public database established like NCBI to submit species? So I do think that in the human space, pathogen genomics, there is, in the Africa CDC, there is the Agari, if I'm not mistaken. There's a platform like that, like Agari, but there is no like a continental one. And we have proposed in one of our papers recently. We are working on that to mean that one that will focus on biodiversity. So that paper, again, I can share it much later. The paper is also, it captures all of this. We're looking at the infrastructure, the databases, the data sharing platforms and analysis platform, and so many other things that are in the paper. So I think this, Joel is asking if we should, if there's similar fellowship for vertebrates. Yeah, if the opportunities arises, we would have such fellowship. Okay, Professor Ann, I'm just going to kind of respond to all the comments via chat and then, yeah.
Yes, so thank you very much. But don't leave the Zoom yet so that we can be able to see the responses that Dr. Thangod will give. It was brought to my attention that there's one practical that we did not highlight, and I'd like us to share the practical from Hawassa University in Ethiopia. Hawassa is partnering with us for the very, very first time, and we thank Dr. Degu for coming on board. And we are showcasing on the screen now the practical that will be conducted at the College of Agriculture at Hawassa University in Ethiopia. The first day will focus on genomics and bioinformatics with a talk on introductory talk on what it is, and then this will be very, very quick move into the DART sequencing data in R, how the methods are utilized and applied. It will then move into the basics of R and how to install the DART sequencing packages. The second day will now then move on to SNP quality and informativeness, the analysis of genetic diversity indices, and the whole of the rest day will be spent talking about population structure analysis using PCA and structure and AMOVA. The third day and the last day of the workshop will talk about structure in more depth, and now go into some of the softwares that are used in interpretation of data that come out of structure. And then following that structure, there will be a dive into phylogenetic analysis, how to build trees, neighbor joining trees, UPGMA, and how to analyze data with the various bootstrap analysis and tree visualization. So thank you very much, Professor Degu, for partnering with us and Hawassa University to showcase this particular, your lab, and to also give these particular skills. So today, now, we have come to the end of the two-day conferencing style part of the regional workshop. We will have the final closing on Friday, but let me just take this opportunity to officially close this two-day conferencing style by thanking you all who have been with us for 2 days of just science talk. I hope we've not been just talking as scientists talking to scientists, but we've been talking to scientists in an easy to communicate and easy to understand fashion. To all the online attendees, thank you very much. We would like to appreciate you for being there. At one point, we registered about 400 people online following us. You will be able to rewatch the videos on YouTube. Just do a search of African BioGenome Project on YouTube, and you can be able to rewatch any of these talks that you wanted to listen again. We want to thank all the presenters who gave various very inspiring talks that were able to delve into this One Health subject. And I think we all really understand and acknowledge that we are all connected in whatever area that we're doing. We are connected. Let us stay connected and let's work collaboratively. We'd like to thank our exhibitors who came to exhibit, and our workshop sponsors, and our sponsors, the technology companies that are partnering with us to run practicals. And if you just give me a minute, I will mention them. Inkaba East Africa Limited, FNS and Illumina, Change Biotech, Tegani Fresh, CMB Genomics, Africa Harvest. Thank you all very, very much for sponsoring this particular workshop. Our participants have been able to access these premises and be able to learn a lot. We also want to recognize the universities and the other institutions that have partnered with African BioGenome Project. Addis University, University of Addis Ababa in Ethiopia, University of Copenhagen in Denmark, Pwani University in Kilifi County, Kenya, the African Genome Center that's running a practical for us, University of Kinshasa in DRC, Hawassa University in Ethiopia, Jomo Kenyatta University of Agriculture and Technology here in Juja. We cannot forget Mount Kenya University. Thank you, you've been incredible hosts. Thanks to the Gitaka Lab who actually coordinated this particular workshop. I want to thank most especially the Vice Chancellor and the Deputy Vice Chancellor who graced the occasion yesterday and opened the workshop for us. It's a great affirmation of the commitment that this university has to advancement of science and technology. To the service providers, our Wi-Fi streaming service provider, Firm Studios, who provided the live streaming and the PA system. You've done an excellent job, and thank you very much. Let's give them a round of applause. Thank you so much. And to the great, incredible local organizing council of Cecilia, Kuja, and Paul, and of course myself. Thank you for burning the midnight oil and working very hard. It is because of their effort that we've been able to have this workshop. If I've missed anybody, just accept our great thanks. I'm aware that you have traveled from Mombasa, from Alupe, from Machakos, from all parts of this country to be able to access this workshop. So give yourself a round of applause, and we thank you very much. So thank you all for being here on this second day, and I officially declare that this part of the workshop is closed. Next year we will be back, and our theme is AI. We've heard a lot about AI, and we've selected that to be our theme of next year. The workshop will be in August. use our social media handles to find out where we will be placed. Remember to put in a good application and you will find yourself here. Thank you very much and have a good evening.
All right, final housekeeping from me. So we'll have tea before we leave. Our hosts have made sure we have that available. And tomorrow, only the people attending the practical for Illumina FNS will be expected. So we will not be having this meeting here tomorrow. The guests coming tomorrow, we will meet at the lab. So have a great evening, and I hope this was a great conference for you.
Study infectious diseases. Analyze genomic variations in tumors. Project outbreaks through surveillance monitoring. Combat drug resistance. Discover mutations for disease monitoring. Identify analyze RNA drivers. Develop simple. With breakthrough simplicity, our fastest runtimes yet, everything in one footprint with onboard Dragon analysis and significant sustainability advancements. This is the highest standard in benchtop sequencing, empowering every lab everywhere on a quest for discoveries that can't wait. This is the MySeq i100 series. This is sequencing made simple.